p.m. and we'll have oral presentations like last 2 days. It seems very tough fight amongst all the presentations and also a tough task for all the evaluators. We can imagine how, I mean, it was a rigorous and vigorous academic activity, oral paper presentations. And I really appreciate these evaluators are working very since last 2 days. So the evaluators will be assessing your presentations and contents, and the results will be ready before the valedictory function. So oral presentations will be over before 4 o'clock, and we'll have the results of all 3 days in our hand before that, and we'll start with the valedictory function at 4 o'clock. So Shri Tanmay Mandloi will preside over the function, and where we'll declare the results of this oral paper presentations. We'll have total 3 prizes, first, second, and third. So let's have our fingers crossed till then. Our technical team, Dr. Chatpaliwar, Dr. Patil, Dr. Manoj, and the pharmacognosy people, pharmacognosy madam, they are working hard for this paper presentations and the scrutiny, and helping all the participants to encourage and helping them to participate also, present also, and improve upon their presentations also. So let's have finger crossed, but till then, let's enjoy the knowledge fest we have been witnessing since last 2 days. May I request our professor, Dr. Aman Upaganlawar, to please start with the scientific session, day 3, session 1. Over to you, Dr. Aman Upaganlawar. Thank you very much.
Dr. Aman, you are mute. Kindly unmute yourself.
Technical support, please allow us to unmute those who want to talk.
Okay, good morning to all of you. Technical team has muted me, so sorry for that. And welcome you all for this last day and first session of this SNJB PharmaCon 2022. And I have found one uniqueness of today's talk, both the talk, that we have a small virtual get together of all pharmacologists, because our convener is Dr. Upasni sir. He is a pharmacologist person. One of the coordinator is Dr. Swati Jadhav from pharmacology. Dr. Amit Zorapurkar sir, also pharmacologist. Dr. Vinod Tiwari from pharmacology. And myself, Dr. Aman Upaganlawar, is from pharmacology. In the next session, Dr. Thakur Desai sir from pharmacology. Dr. Vipin Dode, chairperson, he is from pharmacology. And Dr. Manoj Mahajan is going to introduce his pharmacology. So I feel very happy and lucky that we will have a good discussion of pharmacology people. So today the session is chaired by Dr. Vinod Tiwari, and it gives me an immense pleasure to introduce Dr. Vinod Tiwari. Dr. Vinod Tiwari is currently working as an Associate Professor of Pharmacology in the Department of Pharmaceutical Engineering and Technology, Indian Institute of Technology, Varanasi. Dr. Tiwari has completed his B Pharm from HNB Garhwal University, Srinagar in 2004, followed by M Pharm in the year 2007 from NDM VP's College of Pharmacy, Nasik. He has, after completing his M Pharm, Dr. Tiwari has worked in Nicholas Piramal Research Center, Mumbai, where he contributed immensely in the drug development projects related to pain, inflammation, and arthritis. In the year 2008, he joined University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh, for pursuing his doctoral studies. After completing his PhD from Punjab University, Chandigarh in the year 2012, he moved to United States to carry out postdoctoral research in the Johns Hopkins University School of Medicine, Maryland, USA. Dr. Tiwari is working in the field of pharmacology and neurosciences from past 16 years, and his research interest is focused on behavioral and molecular neurosciences, decided to understand the various neurodegenerative disorders, including pain biology. His postdoctoral research work with Professor Srinivas N. Raja, Director of Pain Research at the Johns Hopkins University, USA, was focused on dissecting the key neuronal circuits involved in pain relief and drug abuse so that better analgesic drugs which are devoid of drug addiction can be developed. He also served as visiting professor at Johns Hopkins University School of Medicine, Maryland, USA. Dr. Tiwari has delivered several invited talks and presented his research findings in more than 30 national and international scientific meetings. He has supervised 8 PhD students and 21 master students. Dr. Tiwari contributed immensely in the field of neurosciences with more than 53 research publications, 22 research review articles, and 4 book chapters. In peer-reviewed high-impact international journals, including Natural Neuroscience, Proceedings of the National Academy of Sciences, the Journal of Clinical Investigation. His research work is highly cited by researchers all over the world. His citation index is more than 3,860, with H index 34s and I-10 index is 61s. I congratulate Dr. Vinod Tiwari for this great profile. He is serving as a member for editorial and reviewer board committee for many leading journals in the field of pain and neurosciences. He is currently working on different collaborative projects with investigators from Johns Hopkins University and with pharmaceutical industries. His lab is well funded with grants and funding by SERP, ICMR, MHRD, and industrial consultant projects. Dr. Tiwari has delivered several invited research talks and presented his research finding in more than 40 nationals and international scientific meetings. Now, I request Dr. Tiwari to chair the session and introduce our today's speaker, Dr. Amit Zorapurkar, sir. Over to you, Dr. Tiwari. Dr. Amit. Hello.
Yeah, may I start?
I think Dr. Tiwari have been again muted.
So Chatpalwar sir, kindly you introduce Dr. Amit Zorapurkar, sir. He has some problem with connection. Yeah.
This is an extempore. I again feel very privileged. This is the second time I have been, or third time in a row, I have been asked to introduce speakers from Dr. Saranjit Singh. Yesterday also, Dr. Sundramgoda Patil.
Chatpalwar sir.
Yeah,
Chatpalwar, sir.
he is there.
Yeah.
Yeah, he is there. Okay.
Doctor.
I'm really sorry for the technical glitch today. There's a lot of internet issues. I'm attending this meeting from my home today, so I'm really sorry about it. So yeah. So I think I can go ahead with introducing.
Yes, sir.
First of all, thank you so much, Dr. Aman, for your kind introduction, for introducing me to the audience, and it's really a matter of great pleasure for me, to be here today and see Upasni sir is also there, and so many dignitaries are present, whom I know, but didn't get a chance to meet. So yeah. So first of all, I would like to thanks the organizer for arranging this wonderful conference. And, this kind of conferences are very, very important for the participants, And so, yeah. So, okay. So it's a matter of great pleasure for me to introduce the speaker of our day, Dr. Amit Johapurkar. So, one second. So, Amit Johapurkar is a general manager at Zydus Research Center, the research unit of Zydus Life Sciences at Ahmedabad. He is an M.Pharm and PhD in pharmacology and is involved in the new drug discovery at Zydus in the new drugs like desidustat as the preclinical pharmacologist. He has a very, good profile with more than 60 research articles and 18 patents to his credit. He is also the recipient of CSR Award for Excellence in Drug Discovery in 2018 and 2019 Indian Society of Chemists and Biologists Award for Industrial Scientists. So, it's really a matter of pleasure for all of us that Dr. Johapurkar is here today with us. Now, I would request, Dr. Johapurkar to kindly enlighten the participants with his talk. Thank you.
May I add to it with your due permission, Dr. Vinod Tiwari and
Yes,
Dr.
sir.
Amit?
Yes, sir. Please, sir.
Dr. Amit has also been awarded for his research, for his efforts in research by a reputed and renowned biochemist, world-renowned biochemist, Dr. Nityanand, whose textbooks we used to read since our degree days. It was his dream to have meeting Dr. Nityanand in person. If I am not wrong, that must have been most satisfying feat achieved by Dr. Amit Johapurkar. Am I correct, sir? Again, he has been muted.
Am I audible now?
Yeah.
Yeah. So thank you, Dr. Tiwari and Dr. Chatpalliwar for your kind words. I am also thankful to Dr. Upasni, my senior, whom I respect a lot, being a very good administrator and also a very good scientist, for inviting me to this conference. And I am also thankful to the organizers, Dr. Zadhav, Dr. Ganlawar, for allowing me to share something which I feel is worth discussing amongst the fellow pharmacists. I hope the slides are visible.
Yes, sir.
Okay. So what we are discussing today is what could be the regulatory pathways for repurposing drugs. So whether we have some incentives left. To start with, I'll try to share two quotes. And these two people I really respect, and I feel every person on the earth should respect these two people. The first one on the left is Dr. George Washington Carver. How many of us have known about him?
Almost everybody.
At least in Maharashtra, those people who are literate, I would say, they should know George Washington Carver for a very famous book authored by Veena Gavankar, who has written Ek Hota Carver. I believe, and those people who can read Marathi and have not read that book, I would say very boldly that they are illiterate. So this guy, George Washington Carver, was a black scientist. We can't say nowadays Negro, so we can say African American scientist who started his life as a slave and then rose to become the first scientist who had around 250 patents to his credit. And he is the one who found around 250 uses of peanuts, which were considered to be a crop, which was considered to be a crop, which was to be given only, to be used only to feed pigs. and not to be consumed by humans. And he's the one who actually brought the southern part of United States from slavery and I would say the malnutrition. He is and will be a great scientist. So being grew up himself from slavery and the racial discrimination, poverty and everything. He was a great teacher as well. And this is a lesson which he gave to all his students and the humanity in general, is that start where you are with what you have, make something of it and never be satisfied. And I think that is a lesson which all the people who are involved in drug discovery, especially in the era when we have seen the golden era after 95, when the US markets open for Indian pharma industry, and now when they are fully saturated and the generics has been fully saturated, and when we want to do something new, there are very less opportunities, but still there are opportunities. And I think repurposing is a way by which we can use those opportunities to our benefit. And we can see today, we can discuss today rather, how can we optimize them. The second person who is depicted in this slide is Sir James Black. And I think those people sitting here, again, I would say if they don't know him, then again, they are, despite not being Marathi, because first one was a famous book in Marathi, but second one is Dr. James Black, who was a medicinal chemist as well as a pharmacologist. He started his life as a physician, and he rose to become a kind of the first person who got a Nobel Award for a real drug discovery, not that some basic sciences which leads to drug discovery. So he is the inventor of propranolol, the wonder drug which saved millions of cardiac patients in the middle of 20th century. He's also the inventor of cimetidine, and he's the guy who started eventually the discipline of medicinal chemistry in the middle of the 20th century. So these two people, they say more or less, they echo each other. When one is saying, start where you are with what you have, and the second person is saying a bit more closer to what we are discussing today and what is our profession and passion, I believe, is the most fruitful basis of drug discovery of a drug is to start with an old drug. And this is what is exactly the repurposing. So before trying to see what is a repurposing, we have to start with obviously as a what is a purposing. Now purposing means or rather what is called a drug discovery. Those people, as Dr. Upkanlawar has rightly mentioned, most of the people are pharmacologists in today's session, and they must have read Goodman and Gilman. Now in the introduction, they give a very good idea of where is the drug discovery today and how it was. The drug discovery earlier was something where you find a herb or an extract and then find something which shows that it can be useful for some disease, and then you use it. So that was kind of drug discovery, means you find something out, right? Now the drug discovery is not more, no more discovery, it is an invention. Now when you invent something, that means you already have a goal and you are trying to make something towards that goal. Now that goal here is a protein in our body. And mostly our discussion, I'm sorry for my limitations, but I will be discussing that which is a small molecule, not kind of monoclonal antibodies or biologics or vaccines. We will limit our discussion to small molecule drugs, means most of the generics are small molecule drugs even today, and that's the easy thing to work on for even a B Pharm or any Indian scientist. So we will limit our discussion to NCEs or drugs or small molecule drugs or formulations which are small molecule drugs. So when these small molecule drugs are, I think my internet connection is slightly unstable. So if I'm not audible in between, please correct me. So the small molecule drugs are typically invented against a protein. For example, say any drug like an adrenergic agonist or antagonist or a beta blocker or beta agonist. These are the drugs which are invented, not discovered, invented against a protein. You know a protein, you know its structure, and you make compounds that will go and bind to that protein, so that what happens after changing the structure, or which in a sophisticated terms called conformation of that protein, will lead to some changes inside the cell if this protein is on the cell surface, or if the protein is in the nucleus or somewhere in the cytoplasm, then inside the cell, so that the cell changes its function. So if it is a myocyte, it starts beating more, or if it is a, say for example, a gland, it starts secreting more or secreting less, and likewise. So this kind of drug invention requires two major things: a very good handle on medicinal chemistry. That means you should be able to synthesize compounds which are scalable. You cannot be content with say 1 mg or 2 mg of compound. You should be able to make them repeatedly up to the level of say kilograms or tons. And at the same time, when I am saying I am saying the team. Means no drug discovery can happen without a team. No single person can do it. And if any person thinks he can do a drug discovery himself, then he's a fool. Escalating that to a broader term, if an institute thinks it can do it on its own, then again it is a fool. So a drug discovery is basically a collaborative science. So when we are saying we need a good medicinal chemist, we also need a good biologist. What for? Because we are eventually targeting a protein, which is a biochemical, and we should know what happens if we touch. शुद्ध भाषा में अगर हम उस प्रोटीन को छेड़ते हैं, तो वो प्रोटीन हमारे ऊपर खुश हो जाएगा या हमसे नाराज हो जाएगा. So whether it is going to get agonized or antagonized. So this requires a very delicate and deep knowledge of protein biochemistry, and also cell signaling, which is also eventually protein biology. So typically, very deep understanding and delicate understanding of chemistry as well as biology gives us a handle on new drug invention. But even if we know that, and even if it is in the hands of the best minds and the richest minds of the world, still it takes 12 to 15 hours of time, despite all the latest technologies at disposal with millions of dollars to spend. Still it takes 12 to 15 years of years to start with it. Why so? Because it requires a lot of issues. You start with a compound, you make say approximately 10,000 compounds. Then you put it into preclinical studies. That means you test it in animals for pharmacology and toxicology. Then you go to clinic, which is roughly, I'm saying, around 100 healthy volunteers. Then you go to say around 500 patients, and then you go up to 5,000 patients. I'm talking of something where out of 10,000 compound, one compound comes in actual clinic. That means it can be prescribed by a physician and it can be sold. I'm not talking of kind of research article drug discovery, where we published one research article and we declare that this drug can be useful in say this kind of patients without any aim to it, means without any actual validation of that in clinic or through regulatory. Now this is very tedious and it is very costly. But this is what is drug discovery, and that is what we want to do. Means everyone who is here listening to this or and studying pharmacy, they want to do this, right? We at Zydus have done this in terms of two drugs, two NCEs, I mean totally new drugs. One is saroglitazar, which is marketed as Lipaglyn, and one is desidustat, which is the newest one, which is marketed as Oxemia. So these are the two NCE drugs, and this is exactly how we started with, say, we didn't do 10,000, but at least 5,000 compounds, and we reached in market after, say, 12 to 15 years. So this is the theory and this is the practical. So, but mind you, this is what we achieved in India. We are trying to replicate that in US, and it will be at least 10 times more difficult and costlier in US. So we are in phase 2 for these both these compounds in US, which is somewhere in between here. So why I am saying it is difficult, and why I showed this is not to pose that what we have done, but to give some kind of practicality to what I'm talking, and not that I'm talking only textbook science. So it requires a lot of efforts and a concentrated and collaborative efforts to make a new drug. And eventually there are two aspects when you want to have a new drug. Obviously, we want to benefit the mankind so that they get a remedy for something in which they are suffering, and that's the primary aim of even Zydus. When we say our tagline is dedicated to life, we want to make life better in terms of wellness as well as in terms of drug therapy. But at the same time, you cannot do that unless and until you get money out of it, because this, as we have seen, this requires roughly $1 billion. That is exactly what we spent so far. Now, to get $1 billion, you need to get money, and these compounds are not going to give you that money in 1 or 2 years. So when you want to spend on research, you want to earn money as well. And when you want to earn money, you have to find ways by which you can ethically earn money by doing drug discovery and development. And obviously, when you have to do that, we obviously go towards something called generics. Generics, where there is a compound already in the clinic, there is a formulation in the clinic, you want to make something different out of it. And then you come out of the original formulation, you make a new and improved formulation so that you can sell it and make money. But that is also getting difficult because there are very few compounds which are available now as generics. So still, means despite all this, that there being so many generics and there being so many NC research projects going on, there are still 95% of the diseases which have no treatments. And we have seen that very clearly when we had coronavirus pandemic. We have seen that there were no treatments, but people found out. People repurposed the treatments. Nobody knew that remdesivir could be used for this. Nobody knew that paracetamol could be a wonder drug for at least 75% of the patients. Nobody knew that we would have a vaccine which can be actually made from an old MMV vaccine, which can be repurposed by some modification for coronavirus. So this has shown that new treatments so far take too long to develop, that 10 to 15 years. But in terms of emergency, we could actually repurpose drugs. We didn't make a new drug. There was no new drug, that is a new chemical entity which was not there in the clinic and anywhere, and we made it for COVID epidemic. No. we use the drugs which were already there, and that is precisely what is called drug repurposing. So we are not talking of something which is a fancy. We are still talking of real service to mankind, where 95% of the diseases where we have no treatments. So we are still trying to find out treatments where we need to have drugs. And the best way to have those drugs, as the coronavirus epidemic has reinforced our belief in, is we can use the original drugs which are already there. And that's why the stalwarts like Sir James Black taught us 50 years ago. Now, before going to how can we repurpose a drug and what are the challenges, let's understand what makes a great pharmaceutical product. Means, and that eventually, obviously, the accessory to it and a side product to it is that it sells also very well. But eventually, to start with, it has to be a great pharmaceutical product. So these are few ideas which make a great pharmaceutical product. And those people, now everyone sitting here has used a pharmaceutical product, be it a sunscreen or be it a cardiac drug. So if you rethink on your experience, you will agree with me that the first thing a good product has to be is well designed. That means it has to have a proven mechanistic basis of drug action. Every diabetic in India has experienced an Ayurvedic drug which claimed to be a very good anti-diabetic compound, anti-diabetic mixture and all that. Eventually, finally, they settle down on metformin and sulfonylurea, or for that matter, gliptin. So a well-designed compound has a proven mechanistic basis of action. It is not that in some book it is written and someone has experienced it somewhere and it is very good, it tests very well. No. For a pharmaceutical product, it has to have a proven mechanistic basis of action, that this is how it works. It goes, for example, if it is sitagliptin, it goes in the body, it goes and inhibits DPP4 enzyme so that GLP-1 is released. GLP-1 goes to insulin and stomach. It causes reduction in food intake. Also, it increases your gastric emptying time, gives you a feeling of fullness. And at the same time, it releases insulin only when your glucose is up. And by that way, it causes eventually a decrease in glucose without increasing your body weight. At the same time, the compound has to be easy to synthesize. A peptide is difficult to synthesize, and a small molecule is easy to be synthesized. And that is why GLP-1 agonist is difficult as a drug, say Exendin or liraglutide, but DPP4 inhibitor, which eventually does the same job, is easy. So sitagliptin sells more than Exendin or exenatide or liraglutide. But obviously, they have their own pros and cons. The next one is it should be best fitted for the intended purpose. That means it should demonstrate better efficacy for indication with less associated side effect. Giving the same example, if you go for say liraglutide or exenatide, you have issues like immunogenicity, you have issues like nausea and vomiting. These issues are not there with sitagliptin, or at least lesser with sitagliptin. So it has a better efficacy with lesser associated adverse events. Then whether it is well positioned in the market, yes, it is. Means now even I have seen that when sitagliptin got off patent and got generic, there was a news in newspapers like Bhaskar or Lokmat. So when local and native means I would say native languages newspaper, they talk about a drug getting off patent and generic, that means people talk about it as a product that really works. Means people are interested that this is a drug which most of the people consume and it is going generic. That means the price of the drug is going down, and that is good for us. And why they discuss that? Because that product works. Nobody discusses a a product which doesn't work, right? Means nobody, you haven't seen people discussing about a new typewriter in the market or a new bicycle in the market. They talk about nowadays an automobile. They talk about nowadays on, say, a new laptop or a new computer. It's as simple as that. Now the compound also and the product also needs to have a long shelf life. So it should have a stable formulation, which should be stable in say 50 degrees under the shade, what we have typically in India in summers. And we don't want a product that has to be kept and delivered only in refrigerated conditions. Nobody would like that, at least in India. And it should be socio-economically acceptable. There are two things. Economically means fair pricing strategy, and they should be accessible. Unfortunately, we live in a society where asking for a sanitary pad is also a taboo in a medical store. So these are the issues which actually don't make a good pharmaceutical product to reach the desiring and deserving and needy people. And the last and most important thing is that whatever product you are making, that should be patentable. So those aspiring drug discovery scientists, I'm not talking to the self-proclaimed stalwarts, but those people who really want to do anything in drug discovery, this is actually a roadmap. You think of any product, means you consume a product and you use a product. So you are the best person, and you are students of pharmacy like me or anybody sitting here. So we all are equally equipped to do to make a great pharmaceutical product only if we find and use whatever product proposal we are making, whether it fits in most of this sieve, whether it is a well-designed product, and what can we do to make it a well-designed product? What does it really fits for the intended purpose? For example, if you are feeling that the currently used regimen of, say, sitagliptin, I'm giving the same example so that it makes sense, is not you think that once in a day can also be made, say, 6 times, means once in a week, and that would be best. You can use your knowledge and you can see literature. How can you make it? And that makes it for best fit for intended purpose. Which diabetic will not be happy if he or she has to take only 1 pill in a week and be happy? How can you well position in the market? You feel that still you don't get a proper formulation or a proper drug in your nearby drug shop, and you want to make it available. What if you want to make it a long shelf of life? Means we know many drugs which expire early. What can you do to make them last long? There are ways to do it. How can you make it socio-economically acceptable? How can someone go to a medicine shop and ask for, with a proper prescription, obviously, ask for a say tadalafil or sildenafil or a sanitary napkin with grace? So this is how we can change anything for a pharmaceutical product, which makes it better in any or more than one aspect of this sieve, and that will make a best case for a 505(b)(2). That means a repurposed drug. So when we are talking repurposed drug, unfortunately in India that kind of things don't work. But obviously, like any like a sunflower, which always goes and changes its direction according to the sun's direction, Indian economy till today changes its direction and goes up and down based on the power of the world, which is US. So because US and Europe, because that is the most regulated market in the world in terms of pharmaceutical products. You can make money in a regulated market better than, and you can make a business better in a regulated market than in non-regulated market. like India. We won't say India is a non-regulated market. Many people will feel bad about it. But unfortunately, we still have many off-target prescriptions. That means if a drug is given for some indication, nobody asks, at least the routine physicians, they don't see whether can they prescribe the drug for the same indication or not. They prescribe the drug for any indication. But in US and in Europe, means in most regulated market, there are three kind of drug applications, which obviously we also have in India nowadays, and that's a better way of doing regulation. But when we are talking in this presentation, we are mainly discussing US and Europe markets, and because that's finally where drug repurposing works, is that there are two kinds of application. One kind of application is called, and all these three applications are new drug applications. The first application is 505(b)(1) application. That application is that contains full report of investigation of anything which have been conducted by the investigator. That means you have, means either you have acquired it from somewhere, or but you are the total owner of the compound. I'm talking of a small molecule. I'm not talking of the formulation. So that small molecule, or say even a peptide, you have invented it, or you have acquired the rights of that invention. You have done all the preclinical toxicity and clinical studies up to phase 3. And then you are going to submit an application, which is called a new drug application, to the FDA. And in toto, this is similar in regulation to India. So regulatory wise, what we are talking is similar in US and India. So what it is going to give you is an authority and approval that you can sell this compound to in the market, in regulated market, obviously, for the indication for which you have submitted the application. For example, we have submitted this compound, desidustat, for the patients who are suffering from anemia, which has occurred because of chronic kidney disease. So we cannot use this drug, at least we cannot sell it. A clinician can easily give it, and that's the loophole in India. But we cannot sell this drug even by prescription to the patients who are suffering from general anemia. So we cannot give it to a pregnant lady who is suffering from anemia. Though it works, but we cannot give it because we have not done clinical trials for that effect. But we have done full 1000 patient clinical trials in patients who have done it, who are experiencing, they are either dialysis or not on dialysis. In both the patients, we can use these drugs comfortably, safely, and effectively to treat the anemia. That means the decrease in hemoglobin, which they experience. And this compound perfectly improves and normalizes their hemoglobin without any side effect in that range. So this is how an FDA or NDA gives you a successful and approved NDA, gives you permission to market a drug with a proper prescription. Now, so 505(b)(1) is a new drug, which requires extensive clinical and non-clinical studies, and obviously to this range, and 12 to 15 years to give a product in the market. But there are other drugs which most of us in India, they do. For example, just I have given an example of sitagliptin, or for example, paracetamol. These are the compounds which are called generic drugs. Now, what is a generic drug? I've just given an example of a new drug. That means, for example, desidustat. Now, eventually, Zydus will lose the patent protection on desidustat, say in any time in say 10 years or 15 years. After that, this compound will get generic. Means what? Any other company will be able to make a formulation and sell this compound than Zydus. And Zydus can also make it. Means any company can do it. Now, what any other company will be able to do it is that they will have to make a similar compound, means same compound. They have to make a similar formulation. They have to show that a formulation which they have made with the similar doses shows a similar bioavailability or bioequivalence in human subjects, means in humans, compared to the innovator product, that is in this case oxemia, and then they can sell it. with proper permission. Now, for the application which they will do to the FDA, means Indian in terms of CDSCO, that application is called as ANDA, that is abbreviated new drug application, because what you need there is not the full clinical and non-clinical efficacy, safety, formulation research and all that. You require a formulation research only with a bioequivalence and bioavailability study showing that your formulation is also similar, if not inferior, to the original formulation of the inventor. So this is the generic drug, which 90% of Indian companies do, whether they work in India or in US. We are talking of something today is a new drug which contains similarly active ingredient as a previously approved drug, but that is something different from a generic or a new drug. That means, for example, if someone comes with a potential that desidustat is a drug which is used for anemia, but we can use it for, say, Alzheimer's disease. And they find out using preclinical or clinical studies that it can be useful to for Alzheimer's, and it is safe at that dose, and we want to repurpose it for Alzheimer's, then it becomes, then you require very less studies, a bit more than the generic studies, but very less than the 505B1 studies, so that you can market the drug which you have not actually developed, you have not actually invented, you have not actually done preclinical, most of the preclinical studies or clinical studies, but you can use the studies done by other people to cite that this works, we have developed a formulation and we want to sell it for that indication. And FDA, and in that sense, if we want to parallel, CDSCO can approve that study and that application so that you can market the drug for that particular indication. Or you come out with another formulation which is saying that this formulation is what currently it is being sold. Say, for example, tablet, it is not a good formulation. We want to make a capsule or a tablet in capsule formulation, or altogether we want to have a topical formulation. You can apply it on the skin and it will have the same effect without any side effect, if any. Then you can show that you have a candidate for 505B2 NDA. And this is what a drug repurposing is about. Just to give you an example before going into the detail, is these are some examples of drug repurposing. So, for example, posaconazole or estradiol or daptomycin or cyclosporine. I can realize that desidustat is a new term for most of us. We have not heard of this compound, but most of us have heard and used the compounds which are here. And this gives you an example that these are the compounds, means the everyday drug which we are using in our life by prescription. These compounds can be repurposed to another, another indication. Means if something is antifungal, you can reuse for antiviral therapy, or if it is an antifungal which is given by oral route, you can make a topical, that is a cream or ointment or animal gel or whatever. If you can do that, then that drug stands to be repurposed. So if you want to define drug repurposing, it is searching a drug for new indication. For marketed medicine, that means, for example, if you are searching, means the best example which we will follow is aspirin. It was an analgesic or antipyretic agent, and nowadays its better uses to stop cardiac arrest and thrombosis. The second is, if you can have compounds, they have shown, means you don't need a drug to be a marketed drug to repurpose it. You can find a compound which is there in the clinic, means in a clinical study, and that company no more is developing that compound. You can still use that compound for a different indication, and then you can put an NDA, an application to FDA, that we want to market this compound for this indication, and they will approve it. Now, I'll give you one example of a single drug example which can be used for all the three indications. Now, this is a compound called fingolimod. It is a S1P, sphingosine 1 receptor functional antagonist. So the same compound has undergone all the 3 applications. One is NC, means at some time Gilenya, whatever, Novartis made this compound to treat relapsing forms of multiple sclerosis in adult patients. So this was an NC when this compound was synthesized and undergone say 12 or 15 years of discovery, and finally it was sent to clinic and then to the market. So it became a drug, an NC. After once it became generic, then another company, which is Honda Neuroscience, they made an orally disintegrating tablet with a slightly different dose of say 0.5 milligram and 0.25 milligram. For pediatric population, they conducted a study in a pediatric population. So that's a phase 3 clinical study. And they found that it is working there with similar safety. And they got a application to market a drug as a orally disintegrating tablet, both for adults as well as pediatric population. So this is an NC. This is a generic, or this is a repurposed application. So this is how you can change an NC, which is already there in the clinic, by some formulation tricks in some different population, so that it still remains a new product and you get a protection. That means you can sell the product, means this product. The compound is same, but the product is different. This was a different tablet or capsule. This is a different tablet or capsule. You only can sell because now it is your ownership up to certain limit. And what is that limit? We can see later. Another is say what typically we do with Indian companies. Fingolimod, it has become generic. That means the company has lost the patent protection. Anybody can use that drug. So the companies like HEC Pharma, Biocon, Sun, they have made the compound. They have seen whether it is bioavailable and bioequivalent as the innovator product, and they sold it. And they can sell it by different brand name. They may not use the same brand name. They can use if they buy that brand from the innovator company. So the first and last is something which means the first is most difficult. The last is easy, but it is very competitive. Everyone will make a generic product. So what is the advantage of your product? Why people will give you money? But this second part, that is 505(b)(2) application, is something which gives you money, which obviously requires more efforts than making a generic, but it requires very less or lesser efforts than making an NC, which obviously very few people and very few organizations and collaborations can do. Now we are going again, means the science is going again one step ahead than Fingolimod, NC, generic or repurposed. Now that is how a new NC can come out of an old NC, and that is how what Dr. James Black has taught us, the pharmacist, 50 years ago. For example, Fingolimod, this is Fingolimod, and the mechanism of action is it goes and binds to S1P receptor. It internalizes the receptor so that the receptor is no longer available for S1P when S1P is there, which is there when there is a inflammatory stimulus in terms of say IBD or CKD. What it does, it causes decrease in the lymphocyte count. So there is a general immunosuppression so that you reduce the inflammation. But that immunosuppression causes many other side effects like say cardiac bradycardia or say macular edema, because that immunosuppression causes changes in the endothelial barriers or other things which S1P does. Now, scientists, they have found out that there is a transporter, and that transporter is something which is SPNS2, that is sphingosine 1-phosphate transporter, which takes out sphingosine 1-phosphate, which is a stimulus which goes out and binds with the same cell S1P receptor and causes immunosuppression. Now this S1P can be taken out by the receptor, and it is means they found it that it is specific to lymphocytes. So what we can do is we can inhibit this transporter so that the S1P doesn't go out. So even if fingolimod is there, it doesn't have any job to do. But then what we need is we need a transporter inhibitor, because fingolimod, if it works there, it will have side effects, because it will go and bind with S1P1 in, say, even blood and cardiac tissues and eyes, and will cause, say, bradycardia, macular edema, or even in some cases very severe side effects leading to severe morbidities or death. But if we don't want fingolimod, why? Because there is a side effect. We can have another drug, and this is a tool compound. It's not yet a drug, so it is only a science, not yet got the fully applicable drug, but it can lead to that. And what they have done is they have made a compound which will inhibit only the transporter. So there is no stimulus going out of the body. So S1P1 will not be there, but it will not be there only in the lymphocyte, only in the lymphatic circulation. It will not affect the S1P1 signaling in, say, cardiac tissue or say ophthalmic tissues. So the side effects may not be there, but you will still have the effect. And that effect is going to cure, say, indications like CKD or IBD, which are a chronic indication. So you don't need a chronic side effect. Now, this is how a new indication can be found, or a new drug can be made from an existing drug, if you understand more about the science and the way. Means there are only two aspects which we need to understand: either pharmacology, means pharmacodynamics, or pharmacokinetics. Now, pharmacokinetics is this: when you make a new formulation for a pediatric population, which is equally useful, that's something called pharmacokinetics. And here you find a new avenue of using the drug or using the mechanism to reduce the side effect. That's called pharmacodynamics. And that is why if you want to repurpose a drug or make an NC out of an NC, you need a very sound and thorough understanding of pharmacokinetics and pharmacodynamics. And eventually you cannot have that unless and until you understand biology and chemistry to the fullest. So you can, means going to 505(b)(2) again, how can we repurpose a drug? This application applies when certain changes are made to the innovator drug. For example, here fingolimod, if you want to make a new formulation or can use a new indication. For example, a similar drug like ozanimod, it is a multiple sclerosis compound, but now it is repurposed to treat IBD. That's altogether a new indication. So that falls in the second use, that is a new indication. Or you can have a generic which you can improve. Like here, they have improved fingolimod, the original formulation, they have improved the formulation, which is a very different formulation, so that you can use it as an improved generic. It is no longer a generic, you still have the patent protection. And for that, you don't need to rely on the existing information on safety. Means you can rely on the existing information on the safety and efficacy of the original innovator's product. You don't need to reinvent the wheel. And that's the most important part of this application. Now, what is the incentive? Means we are doing that, but why it makes better than making a generic is because a new chemical entity will have the market exclusivity in typically European or American, and that these can change by 1 or 2 years with the recent modifications in the law. But typically it gives you a market exclusivity for 5 years. That means if we market, for example, desidustat in US, if we get a permission later, we can have the exclusive marketing rights for 5 years. But if we make a new formulation out of an existing product, say, for example, aspirin, if we make a new product or formulation, I'm giving only just crudely the existing example. Then we have 3 years of marketing right, which is still better. In another indication, if we get an orphan drug indication, for example, there is a disease called, for example, lactose intolerance. It is a disease for which there is no drug. In that case, that disease can be said an orphan disease. And if our drug gets a designation that this drug can treat that orphan disease, then you get an exclusivity for 7 years. And for pediatric add-on, you can also get 6 months. So this is how typically a market exclusivity is. What is the incentive for a repurposed drug over and above what you are already having? Now, when you get that, a drug repurposing may take 3 to 8 years, and still you have a limit. After you market the agent, you still have 3 years to market it and solely and make money out of it, so that you can use that money for, or you can recover the money which you have spent in developing that drug in 3 to 8 years. So it is still lesser than a typical new drug discovery, which requires at least 5 times or 6 to 10 times more money, and say 3 or 4 times more time. Now, how can we do that? One is that you can take a off-target, off-patent drug. You can enhance the formulation, and you can have a new indication. You get exclusivity. Or you get a drug which is off-target, means off-patent, which is no more patented. No one has the exclusivity. You change the form. Like, for example, people have used sitagliptin. They change the salt, and it is still unapproved. You do a bioequivalence trial, and then you get a new indication. Means the indication might be same, that is type 2 diabetes, but you get exclusivity. Another indication, another could be you get a drug which is off patent, you get another drug which is off patent, and it is a late stage developed drug. So it may not be a developed drug. You combine them, get a new indication, and get the exclusivity. I'll give you an example. The example, and it was again in the news, and not to just to reassert, but I have seen this news in a daily as simple as Rajasthan Patrika, a typical Hindi belt daily. So you don't expect those kind of dailies giving scientific informations to that level, but they have published a news that this compound or this drug Entresto, it has become generic. So this is how, as we have seen in very primary slides, that this is how you can make a formulation or a compound or a product which is discussed by people. A good brand is always discussed by people. So it's like that. So this was a compound, everyone knows. Valsartan is a compound which was initially approved by FDA for treatment of hypertension. Now, in 2002, it was also used for heart failure. Now, later in 2005 also, they extended some indications. Obviously, it is going to get expired in on a day or later. So they have another compound, and that another compound is called a natriuretic peptide modulator, which is called sacubitril. This is that compound. Now, sacubitril, this company, Novartis, they went up to phase 3 trials, and they couldn't get the approval. So what they did, using this principle, this is a compound that is valsartan, which was already approved. This was a compound which is sacubitril, which was, they went up to phase 3 trials, but they couldn't get enough efficacy so that that compound can be marketed alone. What they did, they combined these two agents as a co-crystal. That means make a crystal out of sacubitril and valsartan. So they make a kind of combination, a covalent bonded compound. So it becomes an NC. But when you put that NC into the body, that means once it comes into the contact of any esterase in the body, it will eventually split into sacubitril and valsartan. And for both these agents, there is enough data available up to phase 3. So what they made a co-crystal, and that made a new NC, but still it can be used as a repurposed drug, and they got exclusivity for 5 years, and they actually earned billions of dollars. So this is an example of how a smart repurposing can give life to your existing drug, as well as give life to the patients who are really needing a drug for, say, a disease like chronic heart failure. And what is the exclusivity we are talking about? They got an exclusivity up to that region, up to 2027. Otherwise, they were losing it in, say, 2005. So they got a large product exclusivity. So these are few examples by you can do that. I'll not go into the details. I think my time is reaching to the end. I'll briefly discuss 2 issues, which we are some examples like finasteride or butasteride. These are the examples which people have used to repurpose a drug for a new indication, from enlarged prostate to hair loss. But what are the issues which we need to take care when we want to repurpose a drug? The first thing is that you are still going to get off patent, because if you have a drug, you made a new drug, for example, sacubitril and valsartan, they made a new drug, but still people developed a similar drug. They made the bioavailability, and in economies like India, you can still sell a drug if it is really available. For example, if you want to sell aspirin for cardiac indication, say stopping thrombosis, you still get aspirin in the market for the indications like analgesic or antipyretic. So people can sell it and they can use it. So what you need is a formulation which can be sold only if you get a product protection only for that product. For example, if you have a tablet, and if you make a repurposed drug out of that tablet as a topical, then eventually no one can use a tablet, even if it is available in the market, for the same indication, because you need a kind of topical formulation for that. So these are some issues. Like if you have a generic version of a drug, then you have no opportunity to recoup your investment, because that drug can easily be sold by any other company, because they may not sell it for your indication, but it is already there in the market. Off label, they can still use it. So what people do is they can make a unique formulation or a dosage regimen. So currently there is a formulation which is available. I have given examples. So you can use it as a topical formulation. No one can make and sell it, even in India. Or a dosage regimen. That means you have sitagliptin and you repurpose it so that you can use sitagliptin with the same bioavailability, means same drug levels. Instead of giving it once a day, you can give it, say, once a week. No one can make that formulation, because you only know that trick and you have patent and exclusivity for at least 3 years. Or you develop new formulations, new derivatives with a similar therapeutic effect, like sitagliptin new salt, and obtain exclusive marketing approvals in new geographic regions. These are the few things which I think are going to get us to something which we are talking of new drugs. And these are few ways by which we can use or put a new drug to repurposing. If we are able to do that, there are many softwares which are free to use on the net available. And as I have told and stressed earlier, that anything which we discussed in this presentation has to be done. It needs collaboration of lot of efforts. Fortunately, you being in a teaching and research institute to the scale of SNJB, you can have that advantage because you have everything at your disposal, from a formulation scientist to a pharmacologist, and with your great marketing alumnus to a formulator, and also preclinical pharmacology or pharmacognosy people who can help you in isolating some compounds which you want to see. I think you're going to listen about it to a great and to a next level of scientific excellence by Dr. Thakur Desai, who happens to be again my senior as Dr. Upasni. So I'm really thankful to you for your patient listening. I'm sorry for being a bit faster in the later half, but in terms of the time and the lack of time, and also I don't want to bore you a lot. But if this has helped you to think on something which can lead to a better drug repurposing, I think this talk is worth. Thank you.
So thank you so much, Dr. Amit Jorapurkar, for the wonderful talk and enlightening the participants on significance and recent updates on drug repurposing, along with very significant details on how to go for drug repurposing. Drug repurposing is also termed as drug repositioning or therapeutic switching. And this method is very, very useful to identify the novel therapeutic agents from the existing FDA approved drugs. As Dr. Jorapurkar has also mentioned, that drug discovery is a Herculean task, and it involves a team of scientists from different disciplines to work together on a common platform. And we all know that it is a very time-consuming and very costly process. But if you work rationally and have a good understanding of the disease pathophysiology, targets involved, and the limitation of the existing therapeutics, you can actually come up with the new therapeutics from the existing pool of FDA-approved drugs. As Dr. Jorapurkar has also mentioned, you need to do the smart repurposing. So, sir, your talk was very informative, and I'm sure that participants would have benefited immensely with your lecture. Now the session is open for discussion. We can take, one or two quick questions from the participants. I think there are some questions, which has been put by the participants in the chat box. If you permit, I can take a few questions from here.
Yes, sir, please.
Ms. Charulata Nima has asked a question. If I found the marketed formulation can be used in particular disease by natural pharmacology, can I go for NDA, new drug application, without clinical trial? Possible, sir?
You require a kind of clinical trial, or what you can do is you can do a equivalence. You can do a proof of concept. Means what typically people do is they do a proof of concept here. Means you understand whether that particular drug is working in other indication, and you can directly go to FDA citing that data, that this is the data which we have, and we want to use this drug for that indication. And if you can show that your doses are not changing and you have a novelty that this is not disclosed anywhere, you have a patent to that effect. You may not have a patent, but you have an exclusivity that this is something which has not used anywhere, then you can surely do. There are examples, people have done that. And fortunately, the good thing is that FDA is so, I'm talking of US FDA, they are so open that people actually go to them. They tell them that we are going to develop this drug, whether it makes sense, and they do tell whether it is it makes sense and what studies you need to do so that we can give you permission. Now this is something we can't expect. No? It's like you are going to say election commission and asking, I want to stand for election, and what should I do to ethically so that I can get elected. But this is kind of work which FDA does, and that is why it makes a sense. And eventually we are seeing that that is also happening in India, and that's a very good terms. Means with the kind of changes which they have done in last 10 years in CDSCO and ICMR, we do believe that in near future, we will have that kind of free and still good regulation will be there in India. So it is very well possible. You can do a very quick preclinical studies using an existing formulation and go to FDA asking them that, can we do it? Can we take it further? Yes.
Thank you, sir, for answering the question. There's another question by one of the participant. In case of one drug is DCGI approved and the second drug is not approved by DCGI, so can we, how we can go for the BE studies?
Yes, there miss again, I repeat, in India that regulatory is not yet fully crystallized. So I'll talk in terms of US FDA regulations only. What happens is, again, the same compound, means I'm telling about sacubitril and valsartan. Now valsartan was approved by US FDA. Sacubitril was never approved, but sacubitril was approved, had gone up to phase 3. So there is a data which is there with US FDA, which they can make you available if you ask, and you can use that toxicity data to so that you don't need to repeat that toxicity again. Provided you have the similar compound made, you can show that these two compounds together are better than individual compound alone in our studies, and that can be even preclinical study, and then you want to take it further. Then what will happen? FDA would ask you, based on your data, some more studies to be done, say equivalence or a quick safety study or whatever. But that study cannot be phase 1 or phase 2 study. It will be only a phase 3 study. So only one clinical study will be required. And that is the beauty of drug repurposing. If you have a good idea, you can always come out with a great product. And again, I have seen that that you don't need to have a full company at your disposal. Means we are living in a startup age, right? You can quickly do some studies using your resources and go to a company. You can, means I'm saying you can even go to an Indian company who has presence in US. If you have merit in your idea, you have a kind of benefit which you have shown even at a pilot scale, even in a preclinical study, provided it is ethical and integral, then you will get enough funding to do that. So means I sit on many BIRAC boards, BIRAC teams who do fund startups, and I know that it is very well possible that even a small startup can get enough money to develop a drug even in US. So it is possible. You have to come out of that mindset that we don't have resources. If you have idea, I think that is good enough.
That's a very good point, sir, and a real motivation for all the participants, like, all these things are possible, if one really want to, pursue. So one, I think I would just, put one last question, from my side. So in our lab here, we are trying to develop some new formulation for the existing drug which are in clinic for the treatment of neuropathic pain, just to, avoid the side effects and the central toxicities like addiction, which are associated with these, like opioid-like drugs.
Yeah.
So as a layman, I want to understand from the industry perspective, do we have to go for all the toxicity related studies in animals at preclinical level if we want to, bring this formulation in the market?
You may not need toxicity because that's a new formulation, right? The drug remains the same.
Yes.
And if you're the toxicity is, and that's what I was saying, the beauty of repurposing is that the toxicity is already reported. Say, for example, if you have gabapentin, all the toxicity of gabapentin is already there in FDA documents, which are open for public. If you are increasing the dose, then that might be needed. But again, the limits, right? In toxicology, they have already told you what is the NOEL. So it changes from case to case. But I think if if it is a new formulation with lesser side effects, you don't need to repeat the toxicity.
Okay.
But again, I saying you off line, we are always open to opportunities like this. So you are welcome to discuss it with Zydus later.
That will be really great. Like I will get in touch with you and
Yeah,
to
sure.
discuss
You
more
can
about these
approach us.
ideas.
Yeah, yeah. You can
Yeah.
very well approach us and we can take it further.
Thank you, sir. Thank you. When I was in Napar, Ahmedabad, we were, talking to Dr. Mukul Jain, who used to visit Napar, Ahmedabad, regularly. I hope he's also doing well. So,
Yeah. Yes.
So yeah, I think that's a very good session, very informative talk, by Dr. Amit Johapurkar. So once again, on the behalf of the organizers, I would like to convey my heartfelt thanks to Dr. Amit Johapurkar for the wonderful and the very informative session. I would also like to thank Professor C.D. Upasne sir, who is the principal and also the convener of this conference. I also convey my thanks to Dr. Okalkar and all members of the organizing team for organizing this wonderful meeting and inviting me to chair this session. Now it's over to you, Aman sir, and thank you so much. Thank you.
Okay. Thank you, Amit. Thank you, Vinod. Now, I thanks Dr. Amit Johapurkar sir for sparing his valuable time with us and enlightening various aspects related to drug perfusing. I'm also thankful to Dr. Vinod Tiwari for chairing the session. Now I request our technical team to release a certificate of appreciation for our today's speaker, Dr. Amit Johapurkar sir and Dr. Vinod Tiwari sir. Sir, it is my request to please accept the certificate.
Thank you, sir. Thank you. It is our pleasure. Thank you.
My pleasure. Thank you.
Okay, thank you, sir. Over to you, Dr. Atish.
So we can leave now, sir, right? Okay. Thank you.
Thank you.
Thank you. Thank you very much.
I hope that we meet soon.
Yes, sir. Yes, sir. Thank you so much, sir.
It is
Upasne sir,
okay,
it is
thank you.
pleasure. It's really pleasure seeing you, Upasne sir. Thank you.
Thank you, Johapurkar sir. It is always pleasure to listen to you and the kind of insights you have given to the repurposing of drug. It is really useful and will be really thoughtful for our RS students as well. The pathways of 505B1 and 505B2 and how they are being used, you are very well explained. Thank you very much. Over to you, Swati ma'am.
Yes. May I request Dr. Manoj Mahajan to take this program further? Dr. Manoj Mahajan.
Thank you, ma'am. Good morning, one and all. I, Dr. Manoj Mahajan, welcome all the dignitaries and delegates for the second session of the day 3, the concluding day of this international conference, SNJB PharmaCon-2022. The session will be delivered by Dr. Prasad Thakur Desai, the well-known scientist for his work in the area of herbals, and this session will be chaired by Dr. Bipin Dhote. It's a great honor and privilege for me to introduce Dr. Bipin Dhote. He is the young and intelligent researcher in the field of pharmacology. He has completed his B.Pharmacy from Amravati University. He has completed his M.Pharmacy from the well-known JSS College of Ooty and doctoral research from the renowned MS University, Vadodara. He is currently working as the professor in the pharmacology at VNS Faculty of Pharmacy, Bhopal, Madhya Pradesh, India. He has vast academic and research experience with the areas of his expertise, including reproductive hormones, myocardial and cerebral reperfusion injury, inflammatory diseases, and several molecular techniques. He has published several research and review articles with journals of national and international repute, and also given talks in various national and international conferences. He has also published books and book chapters with international publishers. His major achievements include the prestigious UNVAS Prize for the best publication in Indian Journal of Pharmacology by Indian Pharmacological Society. He has also recipient of Best Podium Presentation Award by APTI. Dr. Bipin, during his tenure at Zydus Cadila, worked on oral antidiabetic GLP-1 agonist, which is now in the clinical trial. He has also recipient of several grants. Few to name are the ICMR grant, INSA and MPCST travel grants. With this brief introduction, I like to invite Dr. Bipin Dote to chair the session and request him to introduce our today's speaker for this session, Dr. Prasad Thakur Desai. Thank you, sir. Over to you, Bipin, sir.
Yes. Good morning, everyone. Thank you, Mahajan sir. Actually, I'm really humbled with this introduction because I do not know I'm worthy of it or not. But I'm really privileged to be in this conference, and I could hear one of my mentor, Dr. Amit Zohrapurkar, who guided me at Zydus Research Center, and everyone sitting out here will be really happy to know about what is repurposing. And I have been bestowed with the task to introduce one more important scientist who is having wonderful career altogether. I take the privilege in introducing today's speaker, Dr. Prasad Thakur Desai. He is CEO of Indus Biotech Limited, Pune. And he acquired his education right from bachelor's to master's and his PhD from Nagpur University. And he is an alumni of Nagpur University, having a great academic as well as scientific background. Besides, he holds many certification courses on patents from World Intellectual Property Right Organization. He has around 35 years of professional experience, including the academic activities as well as the research expertise. He worked with the institutions of national and international repute, especially BITS Pilani, Pune College of Pharmacy, Bharati Vidyapit, and he is spearheading the organization Indus Biotech from last few years with great success. He is involved in scientific research. The focused area seems to be preclinical. He is working on development of various model toxicological studies, safety studies. He has special expertise into pharmacokinetic and pharmacokinetic development. Apart from that, he is counseling and making many more researchers to know about regulatory affairs, filing patents, and making ANDAs for their new formulation. He has been involved into scientific activities with numerous presentations and publications. More than 100 peer-reviewed international publications are there. I feel that this itself is a most prestigious achievement. He has around 60 international conference presentations, many book chapters, and he has already written 2 books. He is involved into magazine article writing, especially for the laymen who are not actually associated with the scientific scenario, but he is actually involving into the social activities through this magazine writing. He has been involved into guiding many PhD students and MPharm students. He is member of many professional organizations in pharmacy, pharmacology, animal welfare, and medical writing. He is actively involved into community pharmacy, through which he is taking the onus of reaching to the section of society who is not directly connected with the scientific activities we all are aware of. So I feel really privileged and really happy to introduce the individual to whom I know very closely, and many of us regard him as one of the most spearheading pharmacologists. So I feel that it is everyone who is going to hear him and learn many things about the science today. I take this opportunity to introduce today's speaker, Dr. Prasad Thakur Desai, and request him to share us the insights about the ongoing research in his company and his overall experience. Thank you very much. So I hand over this session to Dr. Prasad Thakur Desai.
Yeah, thank you, Dr. Vipin. I am audible?
Yes, yes, you are.
Okay. So thank you very much for the very detailed introduction. Sometimes it is good to hear from others what we did in the past, and it's a feel good about it. Thank you very much. And I also thank Dr. Upasni, Dr. Vivek, and Dr. Aman for calling me and giving me opportunity to share some of the thoughts, our experiences about some global regulatory approvals, and this time or natural products. Last lecture we heard how difficult it is to get any regulatory approval anywhere, either in India or outside, for synthetic product. Our company and my whole research is basically related to natural products. So I hope that I will give some additional insight about these regulatory approval processes and opportunities and challenges. As a case, I will just talk about one of our product which we developed in past few years, and now it got already approval in some of the places. So so this is one landscape I just try to bring out specifically for a natural product. And naturally, when you talk about the approvals, new chemical entity is the highest level of evidence which is required, and highest level of complexity, which we already discussed in last lecture. But for natural product, you have number of opportunities from increasing complexity. Naturally, when you do some more research and more evidences are submitted, approval will be of that much of value. Naturally, there is a demand. So new chemical entity, as Amit is already mentioned, that you get a specific marketing monopoly, or you can say marketing exclusivity in the market. Such a exclusivity you won't get in some of the natural product categories. In some of the natural product categories, we can get it. So in the last or lowest level, food products which you and me, we can just purchase from market, fruits or vegetables, and they are also natural product which has medicinal value, particularly which we are using to prevent the diseases or to give us a nutrition. But if you can do some research and get some nutritional claims, you can market this product as a dietary supplement. There are many such products. You can get it from fruit or vegetable isolated from that, and they are in the market, but naturally you cannot make any specific medical claim or disease claim. To next level, if you can do some additional research, you do some clinical trial, specific clinical trial against in some disease population or some healthy population, you can claim as a complementary medicine. This is a place where you can take not only food product, but you can take some herbal, other natural herbs also, which are mentioned in the traditional literature. And in India, Ayush, which is a body which is related to this area, which can consider it to give you marketing rights or licenses. But more or less, it is a license for manufacturing as per good manufacturing practices, and they want some quality claims there. Do the disease claim. To higher level, if you have some of the specific health claims, and if you can prove it by naturally clinical trial, the body like FSSAI can give you food safety organization in India, they can give you some specific claim. In similar claims you can get in other countries for special medical purposes or medical food category in Europe. There are some similar regulations exist in other countries in Europe or even in other places. But in this case also, you won't be able to give claim any disease or specific claims. But if you can do a specific to go to that level, almost like new chemical entity or NDA or IND level of research for a natural product, in US FDA has a provision to give you a approval called as botanical drug approval, and by which you can enjoy the same advantages as that of synthetic products. Of course, you have to go to that level of research. And recently, Drug and Cosmetic Act, there is a proposal to amend and add to the phytopharmaceutical category, and they also have some provisions. Naturally, they are almost similar to the NDA or IND level of procedures. So this is what is possible. Now, naturally, increasing complexity, which I was talking about, I will try to tell you what are those complexities. This is just a list of the previous one and a very more specific information. Like dietary supplement, they should meet a dietary supplement definition as there is in US, there is a Dietary Supplement and Health Education Act. And there are some provisions which are mentioned in that act, by which if your compound is or your product is suitable for that, you can definitely market the product in US. This is not approval. This is basically you have to assess your own position and all the risk and benefits, and then you can market it in US without any approval as a diet supplement. Naturally, there is very, very important wording you have to note into the claims, and any disease claim you cannot write, and you have to write about this FDA is not warning about that this is not a FDA evaluated product. So there are certain regulatory norms which you have to follow on the label also and as a studies also. And in Australia, there is a category which is also non-prescription category proposed by TGA. There is a well-defined norms as a complementary medicine. Now, complementary medicine, there are two types of medicine they propose. One is listed complementary medicine, and there is a registered complementary medicine. So listed is basically a low risk indication you can include, and register you have to go for the high risk indication. In both the cases, naturally all the studies you have to do, clinical trial you have to do, which is evaluated by TGA, and they can give you a listing. This is also not approval. This is a listing again. So mostly this is you have to self-evaluate yourself against the risk. And if there is a problem, naturally TGA can take action on that. There is categories are again, some of the categories are prescription based only. So medical food in US and product with health claim in EU, which is basically targeted to give solutions against nutritional and dietary management of metabolic process. So the word metabolic process is very important. If the product fits into that category, you can definitely apply for those and you will get the approval. This is, of course, approval process is there. And botanical drug, as I told you, is almost like synthetic drug approval in US, IND and NDA process, which is intended to provide the solution to cure or treat the disease as synthetic products. In India, the parallel, not parallel, but some of the categories are there under which you can market the natural products. Like dietary supplement and food supplement, as I told you, like sports nutrition product, like proteins, vitamins, mineral, they can be dietary supplement. There are certain exercise physiology products like endurance product or testosterone boost products are also there. Recovery product, exercise recovery products are also there. So this is another very subcategory of food supplement, which is itself is quite a big one. Our companies mostly work in these areas. This category of product also you can naturally market to US as a dietary supplement under DISHA product. Then Ayush product, we already know about proprietary Ayurvedic medicine or homeopathy medicine, for which you can get an approval, license to manufacture. There is no product approval as such. Then herbal or natural cosmetic. This is a specific category, of course, for cosmetic purposes. Again, here state FDA gives licenses to manufacture. There is no specific license to market. And phytopharmaceutical is a recent category by in the Drug and Cosmetic Act, which is to be developed to the full extent. And there is a good opportunity in this area for drug. Now, in this slide, I would like to show you some of the challenges which can be, or at least some of the points which we should be taking care of. I'm just taking a point which are separate or different from synthetic product. Of course, what I am assuming that most of the science or the research which you have to do for the synthetic product approval, almost all of them we need to do, except some of the things are different. But these are the points which are specific to natural product, which has to be done and very important for the approval of natural product all over India or for any category most in the cases. Like first is botanical authentication. Naturally, natural products which are coming from plant origin, people tend to adulterate with many other similar looking products. So naturally, unless we do the analysis by sophisticated method, we won't be able to know whether there is a proper active compound in that. But that HPLC fingerprinting will not give us whether the product is by default, it was taken from authentic source. And that is why botanical authentication is almost compulsory, and which everybody of us know. That is a taxonomical classification which is to be done on the each sample of the raw source. And by the expert taxonomists, they have to do that. There are certain organizations in India as well as in the world, they are the only one who can do that. And we have to submit the voucher specimen for that. It's a very, very important procedure for as a part of the approval process. Then recently, FDA, particularly in US, they are insisting on DNA barcoding of the raw material or source material. The purpose is same, to prevent the adulteration. It was found that people are doing adulteration in a way that HPLC fingerprinting is same, but it is coming from some different source. The problem here is that the source can also contain some of the other toxic compound which can be problematic in future, although HPLC fingerprinting is same. So DNA barcoding is nowadays almost necessary for any dietary supplement product or any natural product in US. Then on-site vendor validation. This is also what happened is most of the time raw material is not grown in a controlled condition. It is done by somebody, and manufacturer is purchasing it as a raw material. Now that can contain lot of other things. Cleaning part is also important, and only we can take a sample. Any manufacturer can only take a sample, but ultimately vendor is going to send the whole lot or the batch for the production. So it is very important that what are the practices the vendor is following. In US, the vendors or a supplier verification program is there, which means if you want to export to US from your factory, you have to, your supplier, which is your supplier means your vendor, which is providing you solvent, which is providing you raw material, which is providing you other any other process material, they also should follow the GMP, good manufacturing practices. It's not only you, it's not it is not sufficient that you are following proper GMP practices, your supplier also should be following. So that is another point, so that you also has to involve into the quality assurance from vendor side or the supplier side. Then each of the material, naturally it should be quality control test has to be there, and food grade is also another important point, because we are anyway doing it a food supplement. So food grade compliance is also necessary for each and every item. In raw source is also there, but there are some process chemicals, if it is involved, they should be food grade. And naturally all the practices, the manufacturing premises has to be good as per good manufacturing practice. So for these things, like for example, good manufacturing practices, one has to audit, get your facility audited from third party, which is acceptable to the FDA of that country. So we are mostly concerned here about US FDA, because our business is for that. But if you follow those practices, almost all the regulatory norms of the world you can follow, with some additional points might be there, but you are almost ensured to follow almost 90-95%, because all our world now there is a uniformity of rules are going to be there, and efforts are also going on to make it more uniform. Now, second thing, for raw manufacturing, raw material, you have to also follow the in-process manufacturing checks. In this food business or food supplement business, it is called as HACCP, Hazard Analysis and Critical Control Point. So critical control is also very important. Hazard analysis means for your type of raw material, hazard can be different. Some places pesticide is a problem, some places solvent, residual solvent limit is a problem. Sometimes heavy metal may be the problem. So we have to understand that particular raw material, study it, and then set a HACCP. And all these things should be documented as per GMP. So which is not the thing which is not documented, it is not done. It is the same law applies to synthetic as well as natural. So after that, these are the problems. Actually, the specific impurity profile which you talk about in synthetic product, here this impurities in terms of heavy metal, pesticide, solvent, residual solvent limits should be maintained, and microbial, because these are natural products. They will contain all type of sugars and very good source for bacteria growth. So naturally, how you store those things, whether it is stored in the proper condition, hygienic practices are followed or not, all these things are extremely important. And regular audits by in specific quality control department, quality assurance department role is very important here. Not only final product, but in-process checks are also very important here. And these are the ones which are audited by the people who are going for the GMP audits. Finished product, of course, which you are going to supply to the people. Rigorous quality checks for final product. Most of them will be based on very sophisticated testing, HPLC or HPTLC nowadays. And then recently, the mechanism-based QC methods are under development. These are not yet established or accepted, but these are also sometimes people are now doing. It might come in future. So these methods are based on, say, for example, you are talking about COX-2 inhibitors from our natural product. So naturally, that efficacy study based on that COX enzyme inhibition study might be useful. So, but this area is still under development. Then another important point, so which people generally very difficult to comply with, is the California Proposition 65 standard for heavy metal. So this is generally done with the ICP-MS methods. And US has extremely critical limits, and for which you really has to put all the checks in process as well as raw material checks. Then only you will get this under limits. Then USP standards are available for residual solvent. With GC we can do it. Then purity testing, purity here which I mean by assay methods. So for natural product also, some of the natural product assay methods are available. Some of the natural product assay method has to be developed in-house. Sometime a reference standard also may not be available. So for that also you have to develop those reference standards. So additional research is involved here. But that is having very good advantage, because these are the things which will make your product more and more robust and strong for the approval. And stability studies, which are as per ICH guideline, we have to follow. These are the chemical part. Now I will try to come to the biological part for the claims. This is mainly related to the efficacy and safety claims, which you wanted to go for the approval. Like science part first, safety parts and second. So science means in vitro and ex vivo studies one can do. Some people can go for science first and then efficacy, clinical efficacy and safety. But in natural product, many times efficacy and safety is the first thing, and then people are interested to know science part of it, this mechanism part of it. So these studies are almost same. The methods are same, which is not a change. Like safety study and toxicology study. Generally, these 4 types of studies are sufficient to start with, but depending on the product, you have to go for additional studies, like acute oral toxicity or whatever route you want to follow. 28-day toxicity, 90-day toxicity, and 180-day toxicity study, mutagenesis study, and chromosomal aberration study. In specific study which I mentioned, for example, fenugreek, which we generally work a lot in the past, there are certain publications were there in the past which shows that it is not used in first trimester of pregnancy. Now, fenugreek seed has this prior art. So in that case, we had to prove that our products are safe in those conditions. So we had to do those studies, specific studies. Depending on the product, we have to go for that. If the product is intended for pregnant conditions, or one more point here, in the natural product is intended to be taken by everyone. It is not a specific population which can be taken. So the safety requirements is sometimes much more than the one which is for synthetic product, because synthetic product drug is to be taken in the supervision of a doctor. So naturally, people will not take excess amount of drug over and above the prescribed limits. So if we are already having safety of those levels, risk for toxicity is low in the synthetic product if you are following those limits. However, the natural product, the recommended dose is there, but because they are not meant for prescription product and they are may not be taken under medical supervision, people can take overages and above a double or triple dose in the day, and that might produce some toxicity. So that is why safety part is to be very, very critically assessed for the natural product. And based on the animals toxicology studies. And then naturally for the claims, you have to go for double-blind placebo-controlled studies, which is gold standard for claims. And relevant population, again, it is very important. What is your positioning of the product? That accordingly, you have to go for that. And naturally, you have to follow the regulatory norms of that country where you are doing the clinical trial and where you intend to do your marketing. So global standards are required here to follow. even if you are intended to market in only 1 or 2 countries. And GCP is already internationally recognized standard good clinical practices. One has to follow in India also. ICMR has specific rules. They are applicable for natural product also. Then regulation and market. Now this part is very important because the market is also growing for dietary supplement, and accordingly regulatory vigilance is also increasing, and rightly so, because these products are to be taken by consumer need to be aware, and there is no physician in between, so there is nobody is there to educate the consumer about the product and other things. So it is responsibility of the manufacturer or a marketing organization to do that awareness. And because of internet, people are also becoming more and more aware, and consumer acts are also applicable for this product also. So naturally, for that, another important problem in this area in market is there are a lot of copy product, because if there is no patent protection, anybody can copy those products and claim what you do or what you want to claim. There is no exclusivity for this product in the market, apart from if you have patent for manufacturing. So these are some of the challenges and the methods which can be, which needs to be taken care of. Now, we are doing this processes since long time, and we came out with some of the products which went up to the approval stage also. So that end-to-end experience we have. So I will try to tell you one such product which we developed based on cinnamon bark. Now, cinnamon bark is a very well-known product. It's a product which is used in household for, as a spice and flavoring agent in pulao and many other places. Is very, very common. Scientifically, if you see some of the literature, it has a broad spectrum antimicrobial activity. It has immunity improvement or immunostimulatory activity. Interesting thing about the cinnamon is it has both immunostimulatory and immunosuppression activity based on the dose. This is already documented. This is very, very typical and very unique about the cinnamon bark. Then the digestive problems also it is used. Because of that only we get good benefits from pulao, which contains cinnamon sticks. And recently, lot of studies been done on anti-diabetic activity of cinnamon, particularly polyphenol. And lot of studies with mechanisms also people are trying to come out. And different type of diabetes with combination with synthetic product. It is used as a, and lot of clinical trials are also done on this aspect. So this was the prior art which we had, and then one of our colleague, Dilip, he wrote one article, and when we did the lot of research, actually we did this literature long back, and we tried to give this review. What are the modern health application? So we were trying to see, can we do some studies and evidence-based science to see what areas cinnamon bark can be explored as a modern health application, which may go to the even botanical drug approval level. So while doing, one of our phytochemist, they did isolation of procyanidin polyphenol compound from cinnamon bark, which has pentamer, tetramer, and trimer, because these are polymers and linkage is there. So there are two types of linkages are possible, A type and B type, and particularly cinnamon contain type A procyanidin polyphenols. Now this type of compounds are known, like procyanidin polyphenols are present in grape seeds, wine, apple, So, and even berries, cranberry, blueberry, all these compounds are basically procyanidin, anthocyanidin polyphenols. What is special here is we tried to, this is first time we did from cinnamon bark, and type A, we characterize it as a type A procyanidin polyphenol, which is also patented as a PCT, and then other countries in US, which we got actually. And then while doing the things, we also found that it is very important as a dietary supplement from cinnamon that it should be free from coumarin. So we develop a process to make this cinnamon bark extract on the marker compound procyanidin polyphenol, which we gave them as an IND route. Naturally, we did efficacy study, safety study, and this was not having any coumarin content. That was another very, very challenging part of the process which we did. After that, we did naturally first thing, as I told you, safety is extremely important. So first thing we did is an extensive series of safety studies, which are basically guidelines are available, OECD guidelines. And in one more important thing is it should be third-party lab, which is GLP accredited. That is what is accepted by the community, regulatory community, scientific community, or even sometimes patient groups also, or community groups. So that is why, because this is to be done in unbiased conditions. So GLP accreditation is given to the lab which is having specific norms about the laboratory testing. And that is why it is accredited. So there we did acute oral toxicity study. We found that 1000 microgram per rat per day intranasal. Now here, the important point is we have done oral studies also, but this particular product which I was talking about, we are trying to go for intranasal route. Because which we found that cinnamon bark extract is explored for diabetes to the extent. A lot of people are selling the product for diabetes. But this immunostimulant activity of cinnamon bark, particularly this polyphenol, is not been explored. So we wanted to develop an intranasal formulation, particularly for allergic rhinitis application, which is a huge problem. So everybody of us has allergic rhinitis in some time or the other, but some patients, this disease is allergic in nature, and every season they have to suffer because of that. And if you see products, these are all corticosteroid products, which has huge side effects. People are habituated to it, habit forming, and there is no natural product specifically designed for allergic rhinitis and having strong evidence behind it. That is important. There are many natural products are also proposed and they are in market. Mostly they are oral, and nasal is not that much available. So these are some of the things which we found that it will be very good for the patient that if you can get some nasal spray out of cinnamon extract, which will be very cheap also for the patient to take. So that is why intranasal route we wanted to develop, and for that specific acute study, 28-day repeated dose toxicity study, and then we did even some in vitro toxicology studies to see some mutagenicity and genotoxicity study. This is a set of toxicity studies we did for this product. One more thing which is also done is most of the some of the marketed product, there is the product from zinc. In the US, they found that after intranasal application of zinc-based product for allergic rhinitis, the sensation of smell is lost after chronic use. And now that is a ban for chronic use. Basically, they can zinc-based product you cannot take more than 1 week or 3 days something. So we wanted to see that also as whether that side effect is there, and good that we have not found that side effect with our product. So our product is keeping or maintaining that smell sensation. So that part also we did in this 28-day repeated study. And we found that a very high dose also is very safe, 100 microgram per day. And this part is published also. All this work is already published, so you can go through individual papers and get details. The next study which we did is an efficacy study. This is a specific model for allergic rhinitis in animals. Bulb C mice, when you challenge with the oral albumin, it will produce allergic rhinitis like symptom, like nasal rubbing, sneezing, discharge. All these things you can easily see in the animals, bulb C animals, after induction of OVA. Basically, you have to give oral albumin daily for 14 days, and then you have to challenge dose you have to give after 7 days after the last day. So two times you have to give oral albumin. First time you have to actually doing the and you have to produce antibodies in the body of the animals, and then that symptoms will be seen after challenge. So this nasal symptom, we could see that this product in 100 microgram per rat, it could reduce the nasal symptom, serum IgE levels, serum histamine levels. Hematology are also quite prevention of oral albumin induced symptom in hematology also. And even we saw the histopathology of nasal mucosa and even spleen, because sometimes spleen is the place where after nasal administration product can reach. After we ensure that safety and in animals and efficacy, of course, in animal, we went for double-blind placebo-controlled trial in Indian population first, which is on 40 patients, 100 microgram per nostril twice a day for 4 weeks we use. And in a clinical trial of intranasal allergic rhinitis, RQLQ, which is the rhinoconjunctivitis quality of life questionnaire, which is a very standard questionnaire and well validated questionnaire, which is used. This is used for all trial, intranasal allergic rhinitis trial, including all corticosteroid third generation, antihistaminic, and even nasal decongestant trials also. So here we got extremely good result. Nasal symptom, eye symptom, no fever symptoms, RQLQ scores are reduced. Then nasal drainage and sneezing. Nasal symptom score, there is a specific parameters which we have to measure. The physician scored this, and that is also reduced. And work productivity, again, this is also questionnaire. So these are the three things which was very standard parameters for the efficacy of against allergic rhinitis, which we did. And we published this paper. Then we had to go to second study, because first study generally in this area, in a dietary supplement, we have to do some additional studies to confirm the activity in the different population. Here we wanted to market this product in Australia. There is a reason for that, because Australia, if you may know, it is a country where all the people resides in the sea area. Australia has around 7,000 beaches, and most of the population of Australia lives around 2 to 5 kilometers from seashore. A very, very typical country, and a huge problem of allergic rhinitis. It is called as hay fever in Australia. And we thought that we can have some good marketing possibility here. There is really need of solution for those people. So we did it in Australia. And this time, we in the first study, we found, if generally what happened, from the study you learn few things and you implement in the second study. So in the first study, we used this product for 4 weeks. But we found that after 3-4 days, people were telling that we are getting relief, why we should take your product. Now then we found that it is having very quick effect. And that's why we reduced the duration. And within actually in this trial, we found that within 1 day, some of the patients are getting benefit, which we measured it specifically in this trial. So here also RQLQ has to be included for the confirmation. So we did it and we got the similar result. Nasal symptom score also same, but in this case we did daytime and nighttime symptoms separately, and both the places we got good result. There is a clinical importance to daytime symptom and nighttime symptom. So daytime symptoms prevention is important because work productivity is important based on that. Nighttime symptoms are important because people don't get sleep and next day they will have problem. So if nighttime symptoms can be prevented, it is a good. And work productivity, of course, we got the positive result here also. So more or less, this trial also was very successful, and we published the second trial as well. Then in the market, people are really also interested to know the mechanisms. And naturally, regulatory bodies are also equally interested. So then we did series of mechanism studies. We found that mast cell degranulation inhibition effect is very, very prominent for this product. We did in vitro and ex vivo, both the type of studies we publish also. Then I will come to the mechanism in detail, but allergic rhinitis has two phases. One is allergic phase and inflammatory phase. So that inflammatory phase by leukocyte addition also inhibited by the product, which is also in vitro test, which we published separately. And if you know, this allergic rhinitis is also responsible or as a causative factor for many viral infection, because it makes it virus to enter easily into the nose and produce the problem. So HIV, HCV, of course, HIV is not having much direct relevance to the allergic rhinitis, but it's H1N1, which we saw in addition to that, and SARS-CoV last year we did, last year. So we found that it is having entry inhibition activity. So irrespective of virus, this procyanidin from cinnamon inhibits the entry of the virus because it has glycan binding properties, and this glycan binding is preventing the virus to bind to the related receptor in the nose and enter into the body. So that is another good. This is basically SARS-CoV ACE2 receptor binding we saw, and we found very strong effect there. So we believe that these are some of the mechanisms by which the product is effective. This is the diagrammatic representation. If you see, there is a sensitization and memory phase where allergen is trying to bind, and product IgE binding inhibition is prevented, which we saw in the animal study as well. And immediate reaction is basically mast cell degranulation, which will gives to rise to eosinophil and other interleukins, and they are the responsible for the symptoms, which generally comes as a later stage. So product IND02 is inhibiting mast cell degranulation inhibition, which we saw in ex vivo and in vivo, and also Ig mediated sensitization, which we did in preclinical study. And another separate experiment we did is a pan-selective antagonist activity of IND02, which blocks and recruit the eosinophil, which is rolling, adhesion, and diaphoresis. These are the typical chain of reaction in allergic rhinitis related inflammation, the second phase. So these are when we completed these activities, we thought that we are now ready as an ingredient to produce some product out of it. And naturally, we already have product in mind is a nasal product. So we produce a nasal spray, which is standardized to total polyphenol, which is generally, these are the 15 ml bottle and other things are there. Of course, we have to do shelf life studies, stability studies as per ICH guideline, which of course took 24 months. Now we completed. This is also important part because natural product based nasal spray is not much available in the market. science is not very new. Mast cell degranulation inhibition is known. Cinnamon bark-based polyphenols are also known. But nasal formulation was not much developed. Not only from cinnamon, from any other. The basic problem is procyanidin is basically, if you see stability wise, they are very active molecules. And when it comes in a contact with oxygen, it degrades. And in the nasal spray, particularly in liquid formulation, this is a huge problem. So what we did is we did a formulation research also and made sure that this is stable. And this is also very, very important work for any marketing purposes to be done. The product has to be stable. So this is the end result. This is just one prototype design. But these are the point here is what are the basic claims which we can make based on the product. Reduce runny, dripping nose, reduce, relieve nasal itching. If you see, we have not given any huge claim, which is, we are not saying about the viral antiviral effect, which is not given because which we don't have the evidence, clinical evidence. So after that, we went through those approval processes. They are different in different countries, which I already told you about. Listed complementary medicine category in TGA. It is licensed as a natural product number, that is NPN category in Health Canada. Health Canada has NPN category and then one more, I think it's a DPN category. It's a drug related category. NPN is for natural product, D is for drug. The procedure is same. My point is all the procedures are same and equally difficult as that of synthetic. But because we had done all those studies, we could get submit and got the license on that. And proprietary Ayurvedic medicine also, because it is from food chain and cinnamon is used in the Ayurveda for immune stimulation, we could get the proprietary Ayurvedic medicine approval in India. So the product reached to this level, I will say only because of the evidence-based science. The key here is ask the question why. And if you get answer, you can convince anybody. So this is all from my side. If you can have any question or query, we can discuss it further. Thank you.
Thank you, Dr. Thakur Desai. It is wonderful to hear you and get an insight about how the herbal products can be taken ahead. I'm sure that there will be a few questions from the students and the participants who are out there. But before giving them the opportunity, can I have one quick question? Because I'm aware that in the US market, heavy metal seems to be a major problem. And we are actually thinking about the inflammatory, anti-inflammatory agents by different foods. So as you are into industry, how you are dealing with the problem of reducing the content of heavy metals in our traditional formulations?
The first thing is the source, the farm itself. That we have to see whether in the soil, what is the thing. It starts from there. My point is, it is very, very difficult to remove heavy metal from the without using a strong solvent. The problem here is food supplement industry generally don't see any organic solvent in positive way. And naturally, if you use some organic solvent, you can able to remove heavy metal, but problem is you will introduce another problem of residual solvent. So source, if you can see the samples and select the source where heavy metal is not there or less, because it depends on weather condition, soil, basically soil and irrigation practices.
Yes, thank you, sir. The session is open for the participants to have questions, if any. If the participant, yeah, please.
I have one question.
Please.
Supposingly in India, major use of insecticide and organic pesticides, it is more in India. If at all a material from biological origin is obtained from India, and if it has some heavy metals, is there any process to remove those heavy metals, and whether regulatory bodies accept that?
So important thing is prevention. That is the only thing. So it is for heavy metal particularly, it is difficult to get removed. The problem here is you can see you can make an extraction process. See ultimately what solvent you use and what is the process, how much time you are giving. There is a temperature control also is possible. pH changes also possible. Sometimes you need to add, so these are the things you can manipulate and try to remove. The problem here is if particularly with solvent, other things are okay, but particularly with solvent, whatever you add as a direct contact with the in the process, we need to analyze it first every time, and there should not be residual of that. Say if you are using, say, except sometime alcohol limits are slightly higher, alcohol and water. So without all our processes, that is the tricky part actually. Extraction processes can be developed by, say, benzene, chloroform, acetone in the lab. But problem is if you practically want to use for the dietary supplement market, it will be difficult because there will be residual solvent and our product will go to more than that. Pesticide part, pesticide and insecticide. So that is another part which why we are doing the work only from fenugreek and cinnamon and some of the product. What is that separation? So separation is basically we saw that agricultural practices, fenugreek doesn't require much pesticides. So by default, farmer will not add it. So if you can select the raw material likewise, that is the only solution. So every time, see, first part is very, very important, selection of raw material. See how to make the extraction. See, extraction can be done with organic solvent, but problem is it is going to cause a problem and market, or it will not be regulatory compliant. Point is, scientifically you can do it, but regulatory compliance may not be achieved, or limits, it may be crossing the limits. Of course, all the limits which I told you are very, very stronger limit for US. Now, if you are not successful in keeping those limits and slightly upper limit you are having, but you can think of having this product outside other than US also. The limits for other countries slightly are less stringent. So maybe you can fit into that. But basic point is avoid organic solvent. Organic solvents are specifically avoided. Only water and alcohol.
Okay. Our one of our international expert, Dr. Professor Adiwale, wants to ask you some questions.
Yeah.
Yes, thank you. Thank you, sir. Thank you for your presentation. It was so enlightening. And I was following your presentation keenly. And then when you showed the structures of procyanidins, my thought went to, will it be good that Dr. Prasad just look at it in whether we test it for COVID, stuff. So by the time you move further, I now saw that actually you have even gone ahead to test the procyanidins for COVID strains. So my question is that even though you said your paper is still under under review or sort of. But during the COVID-19 that everybody were asked to sit back at home, why is it that the World Health Organization and all the other regulatory bodies prevented people from using natural products to at least to be as a supplement to to COVID-19? Why did we not use all these things as at then and then now? Although I don't know, or I can't predict, or I cannot preempt what your result has been, whether it really worked at a higher percentage, or it is still moderate. But please help us to throw more light. It is advisable that people should go towards taking more cinnamon form of supplements. Yes, thank you, sir. That is my question.
Yeah. So it's good to ask. In India, when the COVID started, the Ayush department has recommended some extracted compound called as SCADA, and this SCADA, one of the major ingredient was a cinnamon. So many organizations accept that natural product. These are some of the things which can be used. Apart from this particular product, which we already did a double-blind placebo-controlled clinical study, even on hospitalized patient. This was actually in the Ayush department came out with guideline also. If somebody wanted to test the product in COVID, there should be guideline, and they provided a guideline for clinical trial. And we followed those guidelines, and we also did. So we found that over the period of time, oxygen, SpO2 levels are improved drastically as compared with placebo. So this is what it is in development stage. Overall natural product, as you question, all the regulatory bodies now coming out and really helpful for the natural product based approvals. The problem with them is we have to give all these points, right from mechanism, quality assurance is very, very important. How we are going to assess the quality, that is a major challenge for quality control of the product in natural product. So if we convince them about the quality control, quality assurance, that is what I told you is not a final quality. It is also important for raw material quality, in-process checks, good manufacturing practice. These are processes also important, because the compound natural product has potential. Okay, we can find out. But if it is not handled in the hygienic condition, it will introduce something additional. Like they are worried about that. All the regulatory bodies in the world are worried first about the safety and then about the efficacy. So we have to follow those norms. And most of the time, if you can follow the international norms, provide a very, very detailed document which can be acceptable to the regulatory body. And they also have mechanism to evaluate by different ways. And that is why the example I showed you the example of 3 regulatory bodies where this product is got approval. We can officially market those products. In a country like Australia and Canada, they are very, very advanced countries. Their law is very strict. So my point is we should make the science experiment to the international level, understand what is their concern, and see the science also, what patient wants. See, we have to keep the balance. What patient want? They want good, safe, and some solution for some problem. We have to solve that. At the same time, regulatory body is worried about the quality, how you handle the product, whether you can consistently deliver the same product, batch to batch consistency. If you can follow these 2 things, I think we can we can market the product.
Okay,
Yes.
Thank you, doctor. Thank you.
Thank you, Dr.
I do
Thakur.
have one question.
Dr. Chattapalli
Prasad.
is also having a question. I think Dr. Chattapalli, you can put your question.
Yes, sir. Dr. Prasad, am I audible?
Yes, yes.
Yeah. This is with reference to the query raised by Dr. C.D. Upasni regarding toxicity of heavy metals, contamination of heavy metals in the products which you prepare, indigenous products, natural products, also nutraceuticals. I'm curious the use of, or how the given preference to use ion chromatography in order to remove specifically toxic metals and those conditions in products with reference to ion chromatography. And what will be the implications of use of ion exchange chromatography on the quality of the product? Am I connected?
I think Dr. Thakur Desai got disconnected. We'll wait for him to get reconnected.
Because what I feel is I get to read about ion exchange chromatography, much more about it in the literature regarding uses of it in laboratory itself, academic laboratories. But I'm not aware of what sort of ion exchange chromatography setup is made in industry.
Dr. Thakur Desai, you are on mute. You can unmute yourself.
Sorry, Vivek, I was lost because of my current was not there. Can you just repeat in the short question?
Okay, yeah, sure. Ion exchange chromatography is known to remove ionic compounds, ionized material from the elutes. So how far the industry bothered about the metallic impurities in the products, how far the industry is using it to get the product safe and free from metallic impurities?
It's very difficult to assess because everybody is using different methods. There are a lot of novel methods also are available. But the use of resins are, I think many people are using. It is a good way to remove. But depend on, say, ultimately you have to see chemistry part of it. Main important point is when you remove that impurities, what is your active material? It depends a lot on that structures of active material. What type of thing you are wanted to separate and give as a marker compound. So, but it is possible. This is a good idea.
Yes. Thank you, Dr. Thakur Desai. And I feel that if there are no more questions, we can thank our speaker for the day. And I wish that organizers should be organizing these sort of interactions more frequently so that we can get to interact with the industrialist as well as the academia, so that everybody can be benefited out of it. I thank everyone out here and today's speaker, Dr. Thakur Desai, and I hand over a session back to the organizers.
Thank you, Dr. Vipin Dote. Definitely, I want to add here that we participants and all over the world delegates should be keeping their eye during the month of December. We are always organizing these type of interactions for 3 or 4 days. We are calling experts all over the world to interact with the young budding scientists from this part of the country. So every year in the month of December, we are organizing SNJB PharmaCon, and this program spreads for at least 3 days. So I appeal everybody to keep an eye over that calendar, part of that calendar. Thank you.
Over to you, Manoj.
I think Dr. Manoj is on mute, so he needs to unmute himself.
Okay. Sorry. Thank you, Dr. Prasad Thakur Desai sir and Dr. Vipin Dote sir. We are so lucky that you both have really find the way to be with us today, despite of the hectic schedules and even it's on Sunday. Thank you very much. I request the technical team to present a certificate of Dr. Prasad Thakur Desai and Dr. Bipin Dote. Sir, kindly accept the certificate
Thank you.
from the organizing committee of SNJB PharmaCon-2022.
Thank you very much.
It is a unique way of appreciating everybody's effort. We are really happy.
This time you will find a official signature of Dr. Saranjit Singh, who is a mentor of this SNJB.
Wonderful, sir.
We are very honored to get a certificate from people like Dr. Saranjit Singh and others.
It is very important.
Yes.
Thank you.
He is a very renowned and his research is extremely good in pharmaceutical analysis. Of course, we heard his lot of lectures, technical sessions also. Amazing person.
Thank you, sir. Thank you very much. For the announcement, I would like to hand over the session to Dr. Swati Zaruna.
Is Dr. Atish Mundada there?
Yes, ma'am. I'm there.
So shall I go ahead or you?
Yes, ma'am. You can go for announcement.
Yes. Thank you, everyone. Thank you, the speakers. Thank you, the chairpersons. Thank you, everyone. And the delegates also. Let's take 15 minutes break for lunch. And we'll again join for oral paper presentation, scientific presentations at 1 o'clock, so that we can close the sessions, that session also before 4 o'clock. So have a nice time, have a nice lunch. Thank you very much. Thank you, Dr. Dote. Thank you, Dr. Thakur Desai.
Thank you.
Nice to see you with
All
us.
the best
Happy
to all
have you
presenters.
with us.
All the best to all presenters for
Thank you.
today's session.
Thank you.
Bye-bye.
Yes.
Bye.
Thank you.
Vivek is asking. Okay. Bye.
Good afternoon, everybody. Good afternoon, Sumit Sarkar, sir.
Hey.
Good afternoon from India.
I tried. Let me see.
Hello.
Yeah.
Am I audible, sir?
Let me. Can you hear me?
Yes, we are hearing you. We are getting your voice. Yes,
Okay,
there are
let's
some
see.
pictures. Yes, yes. The same person which I departed from 22 years ago.
Yep, you got it right.
Yeah.
Is
It's good to see you, sir. It's good
1:39
to see
morning
you.
here in US.
Yeah. Yeah, I am aware of. There must be early morning. So should I start?
Yeah, I'm
Yeah.
ready for evaluation of this presentation, right?
So, dear delegates, this is the third day of SNJB PharmaCon 2022. Our coordinators, Dr. Atish Mudada and Dr. Swati Jadhav has permitted me to start this session. We are about to start the third day of scientific oral presentation competition. For this today's session, we have got 10 presentations. And to evaluate, we have got Dr. Dhiman and Dr. Sumit Sarkar from FDA USA. He works in US FDA.
Hi, good afternoon, everybody.
Good afternoon. Good afternoon and welcome.
Welcome, sir. Dr. Munish Dhiman, sir.
Yeah, thank you.
Okay.
Good morning to Dr. Sumit Sarkar.
Thank you.
Okay. Now, I will ask Dr. S.B. Patil to introduce Dr. Dhiman and
Thank
Dr.
you, sir.
Sarkar to the delegates.
Very good afternoon to one and all.
Good afternoon.
First of all, I welcome both the judges for today's oral presentations. Dr. Sumit Sarkar, sir, from USA, and Dr.
Thank you.
Munish Dhiman, sir, from Mumbai, India. So it gives me an immense pleasure to introduce first judge, Dr. Munish Dhiman, who is presently working as General Manager and Head Formulations Research and Development at U Medica Laboratories Limited, Navi Mumbai, since 2021. Dr. Dhiman has completed his MPharm from Mumbai University and PhD from the Pharmacy Department, Maharaja Sahajar University of Baroda, Vadodara, Gujarat. Sir is having vast experience of almost 20 years in research and industry. Dr. Dhiman has worked in various industries at different positions. To cite few, he worked as Associate Director, Formulation Research and Development at Jubilant Pharma Limited, Noida, UP. He worked as Senior Manager, Formulation Research and Development, IPCA Labs Limited, Mumbai. Team Leader as Formulation Research and Development, Glenmark Generics Limited, and Deputy Manager at Dr. Laboratories in Formulation Research and Development. His research interest areas are modified release formulations and development of pharmaceutical product based on quality by design. As far as research contributions are concerned, Dr. Dhiman has published 15 international publications and 5 national publications, along with 25 presentations in national and international conferences. To his credit, has 6 patent applications, and he has guided 6 students for MPharm projects in industry for their project work. He is life member of IPA, and he has also a reviewer for various journals for reviewing the research articles and review articles. So on behalf of team SNJB PharmaCon, I welcome you, sir.
Thank you, sir. Thank you, Dr. Sanjay.
Another judge for today's competition is Dr. Sumit Sarkar, sir, who is presently a research scientist in US FDA. Dr. Sarkar received his doctorate in neuropharmacology and neuroscience from Nagpur University, Nagpur, India in 2001, where he acquired a thorough background and expert skills in neuropharmacology and toxicology. His research contributed to mechanistic understanding of neuropeptides, especially opiates, and their interactions with gonadotropin releasing hormones in the hypothalamus. In 2000, Dr. Sarkar received further training during a postdoctoral tenure in Dr. Donald Lachman's lab in the Division of Endocrinology and Metabolism at Tufts New England Medical Center, Boston, where he studied the role of neuropeptides in regulating thyrotropin releasing hormone and corticotropin releasing hormone in the rodent brain during episodes of fasting and bacterial infections. His postdoctoral research confirmed that the phosphorylation of CREB is an essential step in activating thyroid and stress hormones in the brain. This specific work earned him in 2002 Abbott Laboratory Thyroid Research Critical Fellowship Award for the best poster presentation at the 84th annual meeting of Endocrine Society. Later at Indian University, Dr. Sarkar studied emotional stress-induced cardiac regulation and fever to explore common hypothalamic origins and brainstem mechanisms, and found a common locus which is the key in the thermoregulation, locomotion, and heart rate. Thereafter, he worked as a clinical research fellow in the Simon Cancer Research Center in Indianapolis for 1 year and 2008. He joined Boston Children's Hospital, Harvard Medical School as a research scientist to study the role of ER stress in industry obesity in diabetes. So with this brief introduction, I welcome Dr. Sumit Sarkar, sir. Welcome to you, sir.
Thank you, Dr.
SNJB PharmaCon. So now I request Dr. Chatpalwar, sir, for further proceedings. Over to you,
Yes.
Dr. Chatpalwar, sir.
Thank you, Dr. S.B. Patel. I am thankful to Dr. Dhiman and Dr. Sarkar to be available to cooperate us and evaluate 10 presentations. Today we will be having 10 presentations. Those presentations are lined up. I will be calling one by one, reading out the titles sequentially. The list, the evaluation sheet is available with both the judges. Participants are requested to observe the timings. After 7, the timing will be counted after you change your title slide. From there, at the end of 7th minute, you will be warned once, and after 10th minute, you will be asked to stop. Dr. Sarkar and Dr. Dhiman are ready. The first candidate which we have today is Dr. Abdullah Sherikar. His presentation is entitled Pharmacological Inhibition of Calcium V2.2 Alpha 2 Delta 1 Beta 1 Subtype Calcium Channel on Peripheral Neuropathic Pain. Dr. Abdullah, are you ready?
Yes, sir.
Yes. Best of luck, Dr. Abdullah.
Thank you, sir.
Please put your slide.
Yes.
Over to you.
Sir, whether skin is visible to all of you, sir?
Yes. Is it? Yes,
Okay.
it is visible.
Okay. Thank you, sir. Very good afternoon to all of you, sir, and thank you for the organizing committee for considering my presentation for this
Excuse me,
oral.
Abdullah. Excuse me,
Yes, sir.
Abdullah.
Yes, sir.
It is a requirement that you should put yourself on camera.
Okay. Yes.
Yeah.
Okay. Thank you,
You may
sir.
start.
Thank you very much, sir, for kind attention to me, and thank you the organizing committee for considering my presentation for this oral presentation. So before just sir. Today, presently, I am working as assistant professor at Sri Villeparle Kilwani Mandar's Institute of Pharmacy, and I did my work under the mentorship or the guidance of the Dr. C.D. Upasni sir. And today I am going to present my oral presentation on the topic of pharmacological inhibition of CAV, that is calcium voltage gated calcium channel 2.2 alpha 2 delta 1 protein, particularly in the peripheral neuropathic pain. First, today I'm going to cover the points. First is the introduction. Second one is the objectives. Third one is the material method. Fourth one is result, and final is the conclusion. So first, introduction. First. Actually, the damage to the somatosensory nervous system, it is a major part or the major ailment or the disorder which is related to the neuropathic pain, and there is a disturbances in the homeostasis, particular homeostasis, and there is a disturbances in the communication between the somatosensory nervous system. Second point, the major contributor for this neuropathic pain are the diabetes, and there are some trauma, anti-cancer drugs, or the alcohol consumption, or the radiation. These are the major contributors that contribute to the development of the neuropathic pain. See, nowadays the advances are getting more and more in the field of the neuropathic pain, to find out to get the root cause, to find out the root cause of the neuropathic pain. So in this direction, to combat or to find a remedy for this chronic pain disorder, there are some challenges in front of the researchers. What are the challenges? Basically, the molecular and cellular complexity is the basic challenges in front of the pain researchers. Secondly, the neuropathic pain is majorly characterized by the hyperalgesia and allodynia. These are the two characteristic symptoms of the neuropathic pain, and the abnormalities in the different homeostasis of the different ionic channels is the major contributor to develop the neuropathic pain. So the propagation of the different biological signals is the major, what we can say, is majorly regulated by the ongoing activity of the major ion channels. Say examples, the activity of the sodium channels, or the potassium channels, or the calcium channels. Out of these, if you see the recent advancement in the neuropathic pain, the major contributors, that is the calcium channel. There are L type of the N type, or the O, different 3 to 4 types of the calcium channels are present. Particularly in that, the N type of the channel, calcium channel, that is the voltage gated channel, they are basically present on the nerve endings of the primary afferent fibers. Now the objective. Before going to the objective, just I want to drag your attention what is the hypothesis. The major model or the induction method used in this study is the chronic constriction injury of the sciatic nerve, particularly left sciatic nerve. So that causes the release of the calcium channel ion from the extracellular membrane to the interior of the cell. Basically, this calcium is entered through the voltage gated calcium channel. This release calcium channel, or the influx of the calcium channel, is going to activate the sarcoplasmic reticulum. In that the stored calcium is released into the cytoplasm of the cell. And this released intracellular calcium ion basically activate the macrophages to release the various type of the inflammatory mediators, say example, TNF alpha or the interleukin-1, 6, etc., etc. And secondly, that the intracellular calcium, this increased intracellular calcium exert a burden on the mitochondria to produce the, say example, increases the level of the reactive oxygen species. There is the inflammation as well as the burden of excessive burden of the oxidative stress that basically causes the Wallerian degeneration of the myelin sheet of the peripheral nerve. And this, after that, there is a development of the hyperalgesia and the allodynia. So considering these points, the main objective of this study to confirm that the chronic constriction injury to the sciatic nerve develop the peripheral neuropathic pain. Second one is to confirm the involvement of the calcium voltage gated calcium ion channels. The molecular docking studies have been studied. And third one is there are some behavioral parameters as well as the biochemical parameters that confirms the development of the neuropathic pain. So next point is the material methods. The study protocol involves the chronic constriction injury to the sciatic nerve, develops the behavioral parameters, say example, hyperalgesia and allodynia. After the confirmation of, after performing following the chronic constriction injury to the sciatic nerve, every week the behavioral parameters are studied to confirm the development of the neuropathic pain. So after 14 days, the neuropathy was confirmed, and in that case, the animals shows the characteristic symptoms of the neuropathic pain like hyperalgesia and allodynia. After confirming after 2 weeks, the treatment, the animals are treated with the intraperitoneal injection of the zonisamide at a dose of 20 mg per kg per day for further 2 weeks. And after 28 days, on 29 days, the animals were sacrificed and the sciatic nerve was isolated and homogenate was prepared, and in that homogenate, the various biochemical parameters for the oxidative stress as well as the inflammation were studied. Now the groupings, first group is considered as the normal, second group is the sham operated, in that the animals left without sciatic ligations, and third one is the group is considered as the chronic constriction injury where the left sciatic nerve was ligated with the suture. And the fourth group one is considered as the CCI with the gabapentin, that is a standard drug. And the fifth group is the CCI with the zonisamide at a dose of 20 mg per kg body weight for 2 weeks. Now what parameters were evaluated? First, molecular docking was done. Second, the allodynia is carried out by the Von-Frey filaments, and hyperalgesia is carried out by the Randall-Salito test apparatus. And third parameters, the biochemical parameters, say example, the nitric oxide, superoxide dismutase, and TNF alpha. And last, the total calcium and calcium ATPases were evaluated in the homogenate of the sciatic nerve. Now the result, first, the molecular docking study shows that the gabapentin
end of 7th minute, 3 minutes more.
Okay, gabapentin and zonisamide, after looking towards the binding energy in kilocalorie per mole, the gabapentin and zonisamide have near equal, that is the inhibiting properties, or that is the binding energy, that is minus 9.36 and minus 9.05. That is equivalent to the standard drug, that is the gabapentin. So the Von-Frey filaments, for the development of the allodynia, this red line indicates the graph, this is the point. This is the point where the hyperallodynia is developed, and this is the before 14 days. And after that, this is the normal animals, and this is the first week of the treatment. In that case, the zonisamide potentially attenuated the development of the allodynia as that of the what you can say the test drug, standard drug gabapentin. And second one is the hyperalgesia, and it was done by the Randall Salito. And this technique, the CCI potentially developed the what you can say the hyperalgesia, and the treatment with the zonisamide as well as the gabapentin potentially inhibited the development of this hyperalgesia. By looking this graph, it is concluded that the zonisamide is equally potent to the potent as an analgesic as that of the gabapentin. Now the nitric oxide level. The 2-week treatment with the zonisamide equally or the similar compared to the standard gabapentin, zonisamide also prevented the elevated level of the nitric oxide. And second one is the SOD. The treatment with the zonisamide significantly attenuated the or the reduced the increases the level of the superoxide dismutase. By confirming these two parameters, the what you can say the zonisamide also equally or the compared to the gabapentin also exert the antioxidant effect. Now the inflammation, for this inflammation, the TNF alpha parameter was evaluated, and as compared to the gabapentin, the zonisamide also potentially or the potentially exert the anti-inflammatory effect by reducing the elevated level of the TNF alpha. Now the total calcium as well as the calcium ATPases. So on these, by looking the study or by looking the result, the gabapentin compared to the gabapentin, zonisamide also inhibited the increase in the level of the both total calcium as well as the calcium ATPases. Now the conclusion. After performing the molecular docking study, it was confirmed that the zonisamide is impart the potential neuroprotection as that of the gabapentin. Secondly, after performing the behavioral study, it is also confirmed that the zonisamide also exert an analgesic, useful analgesic or the beneficial analgesic effect as that of the gabapentin. Third one is that the compared to the gabapentin, standard gabapentin, zonisamide also exert a useful or the beneficial antioxidant property. And last, after reducing the inflammatory mediators, say example TNF alpha level, the zonisamide also exert the useful anti-inflammatory effect. Now the references.
Thank you, Mr. Abdullah.
Thank you, sir.
You have actually exceeded the given time limit by 2 minutes. So the judges were quite lenient. It doesn't look good to stop you in between, but it would be very good if you could limit your talk in the given time.
Okay.
Thank you. Thank you.
We'll take note of that, sir.
Over to you, Dr. Sarkar, to question.
Hey, Dr. Abdullah, nice talk.
Sir. Yes, sir.
Couple of questions. So, is this particular drug is already been proven that it is neuroprotective in the central nervous system, or is it only for the patient?
No, it is already actually it is a atypical anti-epileptic drugs, because if you see the first line treatment for the neuropathic pain is the anti-epileptic drugs.
Okay. No, yeah, I mean I understand that part. Is it been used for any other neuroprotective, like anti-epileptic is a different mechanism, right?
Yes, sir. Yes, sir.
Then when you say neuroprotection, it's an umbrella term, right?
Yeah.
So is it been this particular one, do you think it's going to act in the similar fashion, which is not only you are talking at the calcium channel, you know, on the neuropathic, peripheral neuropathic pain, but the epileptic drug is different mechanism, right?
Yeah, basically that's why I choose the target site is the calcium channels, because if you see the ongoing activity is very important, particularly through the various ion channels. So my target is to focus only to control the ongoing activity by controlling the activity of the calcium channels, particularly voltage gated calcium ion channels.
So do you have a plan to going forward to look all the cytokines and chemokines, or only?
Yes, sir. Yes, sir. Yes, sir.
Oh. Okay.
Thank you, sir.
May I now invite Dr. Dhiman?
Yeah. I don't have any further technical question. Just one small question to Dr. Abdullah. Would like to have his comment in terms of novelty or innovativeness for the methodology and the topic he has chosen.
Yeah. Yes, sir. Actually, if you see the first line drug in the market is the gabapentin. So we are using as a first line drug for the treatment of neuropathic pain. But nowadays, the zonisamide also, it is equivalent potent as that of the gabapentin. If you see the molecular docking study, the binding energy for the gabapentin as well as the zonisamide, it is coming towards the equal. For the gabapentin, it is a 9.36, and for the zonisamide, it is a 9.05. So why should we limit ourselves for the gabapentin? There should be a alternative for this gabapentin also. So I was I was thinking to take such a molecule that will be alternative to the gabapentin.
Agreed. So are you sure that there is no prior art existing for the use of the zonisamide?
No, sir. No, sir. It is. No, sir.
Thank you.
Thank you, sir.
Yeah, I'm done.
Thank you, sir.
Thank you, Dr. Dhiman.
Yeah.
May I now declare the name of another presenter?
Yeah.
Is the other presenter ready? Encoded P22.
Yes, sir.
Thank you, Dr. Abdullah.
Yes, sir.
Encoded P22. The presentation is entitled Formulation of Natural Gum Facilitated Ethyl Cellulose Microsponges of Antifungal Drug. Mr. Anup Thakre.
Yes, sir. Sir, am I audible?
Yeah, you are audible.
Yeah. Are my screen is visible?
Yeah, the slides are visible. Will you please put them to slideshow?
Sure, sir.
Yeah. Okay.
Okay. Good afternoon to all the dignitaries present, the evaluators, organizers, and the co-participants. I am Anup Thakre, working as assistant professor at Priyadarshini Jewel College of Pharmacy, presenting my oral presentation on the topic formulation of natural gum facilitated ethyl cellulose microsponges of antifungal drugs. The content of my presentation includes introduction, materials and methods, result and discussion, conclusion, future studies, and references. Now, starting with what these microsponges are, I am having very short thing on these microsponges. Microsponges basically are the spherical shaped particles having the pores and mydrites interconnected voids of the particle size that ranges from 5 to 300 micrometers. So ideal characteristics of this microsponges drug delivery system that these are stable over the pH range from 1 to 11. Then they are stable at a temperature up to 130 degrees centigrade. They are compatible with many of the ingredients and the excipients. The average pore size onto the microsponges pores, it is 0.25 microns, so that during the process, there is no need of the sterilization. Then about 50 to 60% of drug may be entrapped successfully into the microsponges, and it shows free-flowing properties. Regarding the various advantages of these microsponges drug delivery systems, so they can act as a controlled drug delivery system. Drug directly applied onto the target organs, then it increases the stability of drug. The drug loading capacity is higher compared with the other topical formulation. These are capable of absorbing skin secretions and reduces the oiliness. What are the benefits of this drug delivery system? Improves the formulation flexibility, also improves the thermo, physical and chemical stability, that is flexible to develop the novel product form, enhance product performance. It also been used for the extended release of the APIs, reduces irritation and hence improve the patient compliance, effective product elegance, and improves the oil control, and it can absorb up to 6 times of the weight of the drying of that microsponges. Then these are the certain advantages of the natural gum that I had incorporated during the formulation of microsponges. As these naturally gums, these are biodegradable. So this is the major advantage for the uses of the natural gum. Then these are biocompatible and the non-toxic mostly. Then they are having the low cost, then environmental friendly processing. Then locally they are available. Then in developing countries, government, they are promoting for the production of plant like the guar gums and the tragacan, so that their applications in the variety of industries. So it can be developed in this way. Then better patient tolerance as well as the public acceptance for the natural product. And mostly these are from the edible sources. So these are the advantages. Then moving to the next, that is the materials and methods used during this process. The material that is drug used is the clotrimazole, that is the antifungal drug, that is very popular and most common drug. Then excipients used, that is the first gum is being extracted from the leaves of Onosma bracteatum wall belonging to family Borangaceae. Apart from that, the major ethyl cellulose polymer it is used, polyvinyl alcohol, dichloromethane, they are being used during this formulation process. Then the methodology adopted during the formulation of these microsponges, first of all, the extraction of Onosma bracteatum gum, then followed by its characterization, pre-formulation study of the drug material, excipient compatibility studies, then preliminary evaluations of the formulated batches, then preparation of the microsponges by quasi-emulsion solvent diffusion method. First, the extraction of the gum, that is the very popular and common method employed for the extraction process. Whatever the plant leaves they are having, they have been first collected, they have been washed. First of all, they are been authenticated. Then they have been washed with the water in order to remove the dirt, debris, whatever the water soluble impurities present in that. Then after they have been dried, after the drying, the powder was been made, and then it has been soaked into the water, boiling water, for 5 to 6 hours. Then later on, it has been allowed to stand for 1 hour, so that whatever the mucilages they have been present onto the plant, their material was then squeezed from the 8-folded muslin cloth so as to remove the mucilage. The mucilage was then precipitated by taking the several portions of the acetone to precipitate that particular mucilage gum. Then later on, it has been dried into the oven at a temperature of 50 degrees centigrade. Further, it has been examined for the particular parameters like the carbohydrate test, protein test, starch test, amino acid test, then the steroids, various glycosides, alkaloids, tannins, saponins, and the confirmatory test for the gums, they have been done. After that, there is a method for the formulation of blank microsponges was first done by this solvent, by this method. In this, the two phases they have been made, that is the internal organic phase and the outer phase they have been made. So internal phase, the gum that is been separated, it is along with the ethyl cellulose, it has been taken into the consideration along with the trichloromethane, and it has been sonicated into the sonicator for 35 degrees centigrade. Later on, the another phase, that is the polyvinyl alcohol and the water in a ratio 1:1 was been taken and stirred for 60 minutes. Both of these solutions they have been stirred and allowed for the formation of the microsponges. So the microsponges structures whatsoever been developed, they have been tried at 40 degrees centigrade for 12 hours and waited for the determination of yield. So this is how the various preliminary selections of the drug polymer ratio, then the internal phase, then the internal phase volume, external phase volume, stirring speed, and the stirring time was being evaluated by using the design of experiment characterizations were being done. Then next one is the formulation of these gum facilitated clotrimazole microsponges. So this is how the internal and external phase they have been made, and only the drug component is being incorporated into the organic solvent, that is the internal phase. Later on, by the application of central composite design, so central composite design it is been employed over here. Here the independent variable, that is the stirring speed and the concentration of ethyl cellulose, they have been taken as the most important or the quality attributing parameters. They have been selected, and the dependent variables like the particle size and the entrapment efficiency, they have been selected. And applying the DOE central composite design, the number of 11 batches they have been designed in this fashion. Then the results and discussions, the FTIR studies, spectroscopy studies of the clotrimazole, then Onosma bractetum gum, ethyl cellulose, polyvinyl alcohol, and its physical mixture, they have been compared. And it was found that there is no typical changes in the structural peaks in the FTIR spectra. Then after that, the characterization and evaluation of drug-loaded microsponges were being done in order to determine the particle size. Then the percentage yield and the encapsulation efficiency was been done. And later on, the formulations, all these 11 formulations, they have been subjected for the optimization process. The software suggested this optimization design for the particle size and the entrapment efficiency. In this case, the dependent variables of a particle size, it is in a direct relationship with the concentration of ethyl cellulose and the stirring speed, so as to decrease in the particle size with the increase in the stirring speed, and it indicates the positive sign in the independent variables. Then the next one, in terms of the entrapment efficiency, the entrapment efficiency again shows the positive signs onto the dependent variables, shows the direct relationship, because increase in the concentration of ethyl cellulose leads to the increase in the entrapment efficiency and has extended drug release ability also. So these are the surface response graph obtained by this DOE. Then this DOE suggested the validation to be done, and on that basis, the suggested solutions by the design expert software, the two suggestions that have been given, and the first one, it has been selected whereby the stirring speed and the ethyl cellulose concentration, they have been taken in a ratio that is 2000 RPM stirring speed and the ethyl cellulose concentration 0.5 gram, and it has been selected, and the results that have been obtained, they are far closer to the expected one. So this is how the surface topography of the microsponges, they have been obtained from the optimized batch, whereby we can very easily see the that is magnified image. It shows us a very perforated and the void spaces that can be observed onto the surface of the microsponges as compared to the images that can be obtained only with the formulation.
Excuse me, Mr. Anu, 3 minutes more.
The scanning microscope images, they shows without gum, they shows somehow less pores, so that the drug is more entrapped in that, and the void spaces, they have not been observed in that. So this is how one can conclude that with the help of these gums, we can perform more perforations to the microsponges, that migrate spaces, they have been out of the white spaces, they have been created, and the drug can be loaded or incorporated in that particular space. So from the present work, I can conclude that the clotrimazole loaded microsponges with the natural gum can be prepared. The extracted Onosma bractetum leaves were extracted by using the ethanol by maceration methods. Then the physiochemical, phytochemical and the identification of gum was been done. Then pre-formulation studies of clotrimazole were been done. FTIR study shows compatibility of drug and polymer, and there is no characteristic peaks change observed over there. So the formulation of microsponges done by this quasi-emulsion solvent diffusion or the double emulsification method using central composite factorial design was applied with the help of design expert software 13 trial version. The dependent and independent variables were been selected, and total 11 batches they have been prepared, and their effect on each other was been studied. With the help of the solutions provided by software, optimized batch was selected for the further studies. Hence, it can be said that the optimized optimization applied to the formulation of clotrimazole loaded microsponges using natural gum was successful to reduce the error and was a time-saving method. Future studies regarding these microsponges, that is the stability studies and the formulation of these particular microsponges into the suitable dosage form, for the further studies. These are the key references that I had used during this research work. Thank you, sir.
Thank you, Mr. Anup. May I invite Dr. Sarkar to have first comments?
Nice presentation, Anup. I have only one question. So, which part of the, like, what are the areas of the work you guys did by yourself?
Pardon, sir, please repeat the question.
What are the work, like, did you do all of this work by yourself, or did you take some help from other?
Oh, sir, this is mind work.
Oh, so you also know how to analyze the SEM data?
So no, the same that has been procured from the outside. In our college, we don't have that same facility. So it has been most of the work that is performed in our laboratory.
So is this particular formulation being used in other area of research, or you're the first time doing it?
No, sir, it is not presented anywhere else. So I had not mentioned any of the other values over here.
No, this particular formulation, I'm talking about this this particular drug using these microsponge and then applying in other disease or any of these. Has it been done, or you're the first time doing it?
No, sir, it is already available. The clotrimazole formulations, they have been available, but I'm just trying to extend the use of clotrimazole for over a period of time, so that once it is applied overnight, it can prolong its activity throughout the night. So that's what I am targeting. That study is still remaining, sir.
Oh, thank you.
Thank you.
May I ask Dr. Dhiman to comment?
Yeah, Anup, I have a couple of questions. Like, how good is your formulation as compared to the existing formulations which are of mucoadhesive in nature for this particular drug, first of all?
Okay, sir, the clotrimazole, it is having a side effect that it shows a bumpiness or a certain kind of irritation. So if we avoid the excessive exposure to the affected area, so we can minimize the side effect over there. So the gums that is being used over here, these are mostly the polysaccharides one, and they are not going to affect the affected area in a pharmacological point of view. So that's why they are being incorporated over here. Also, it is being used to, as the structural modification occurring over there, it can absorb the skinny secretions and it can adhere to the surface for the furthermore times, so that it should be incorporated into the certain gels. So that was the next thing that I'm going to perform.
Okay, so the research work you have carried out is limited to topical application only. Are there any other application of microsponges? Like, are they compressible?
Yes, sir. They can be compressible one. They can be used in the capsules mainly. They can be used in the tablet also.
Okay. And what about efficacy studies? Have you performed any efficacy studies, in vitro release profile?
Sir, at present, the in vitro release profile is not given. So I'm studying that at present.
Okay. And did you ever check that whether your drug is not changing its polymorph while you are incorporating it in microsponges through chemical reactions, treating with polymers and solvents? It is very likely that when you treat it with solvents, most of the drugs, they may change their form, they get converted to other crystalline form or convert to amorphous form. So that may impact on their efficacy as well as absorption, if it is limited to absorption. In case of clotrimazole, although it is related to topical application, only drug won't absorb, but did you check for polymorph conversion?
Sir, it is reported in some cases, if we are using other method for the formulation of microsponges, that is polymerization reaction, where the temperature is being increased. So there are chances that polymorphs may get formed. But in this double emulsification process, as there is not temperature involvement is there, so the chances are it is to the least one. So there are no polymorph, they are going to be changed. And FTR shows there is no particular affected area or the peaks that.
But FTR is not the appropriate technique for characterization. It should be XRD and DSC.
Yes, sir.
They give more accurate idea of form conversion. Okay. And my last question, like you have utilized the natural gums, which are prone to microbial growth.
Yes, sir.
So in your formulation, how you are taking care of it, and you are most applying it to the most sensitive area of the body on the mucosa. So how you will be ensuring that your formulation doesn't carry any microbial load?
Yes, sir. The laboratory level, there are the microbial examinations were been done onto these gums. They have been extracted, and they are showing they are totally the sterile one. So during the incorporation, they are also been in a sterile conditions. So this is how the care I'm taking care of the microbial contaminants to be avoided during the formulation.
Okay. Yeah. Thank you so much.
Thank you, sir.
Thank you, Dr. Dhiman. I personally do have one question towards Anup Thackeray.
Yes, sir.
Okay, so do you believe that there is no interaction between the excipients taken and the drug? On what basis do you declare that there is no interaction?
Sir, prima facie, what the FTIR peaks that I am getting, they are not significantly showing any kind of interaction, sir. But later on, as sir says, the XRD studies is still remaining and DSC has to be performed. So
yeah,
that's
coming to IR, particularly to the conclusion drawn from the IR spectra, how do you say that there is no interaction?
Sir, there are no significant peak changes that have been observed in the physical mixture that has been obtained after these particular combinations, sir.
If there is IR is a physically is based on physical principles. It's a physical interaction between radiation and the molecules, the bonds of molecules. If there is no bonding, if there is no intermolecular interaction between excipient and the drug molecule, there won't be the carriage capacity between excipient and the drug. The excipient won't be carrying or binding with the drugs if there is no interaction. And this interaction is studied definitely in IR spectra and it is reflected.
Sir, actually, this is an emulsification technique, sir. During the emulsification techniques, these gums, they are forming a mitrite spaces over there, and the drug is going to be entrapped in that structure, sir. So that's why we can say there might be a possibility of the weak forces or the cohesive adhesive forces that might be acting over there during the entrapment process. But
yeah, and these cohesive forces, yeah, and these cohesive
or
yes, very much. These cohesive forces, they make higher frequency change.
Yes,
a change from lower frequency to higher frequency. And this is what we have to read in IR spectrum.
Yes,
I'm sorry that you are not displayed it here, but still overall, it was a very good presentation. Thank you. May I now call upon other candidate with your kind permission, Dr. Dhiman and Dr. Sakar.
Yep.
P23.
Yes, sir. Please.
Miss Aris Zehra. P23.
Hello.
Is the candidate available?
Good afternoon, sir.
Good afternoon. The presentation is entitled Elizartan Transdermal Hydrogel Accelerates the Recovery from Volumetric Muscle Loss Injury in a Rat Model. So, best of luck, Ms. Aris Zehra.
Should I share my presentation, sir?
Yes, yes.
Is it visible?
Yes. Will you please put it on slide mode? Yeah.
Yeah, it's good.
Start it.
A very warm good afternoon to everyone. My name is Arizara, and today I am going to give an oral presentation on Elizartan Transdermal Hydrogel accelerates the recovery from volumetric muscle loss in the rat model. As we talk about the skeletal muscle, they are very vital components of our body, and it contributes about 30 to 40% in females and 40 to 50% in males. So what we have observed, that the percentage of injury in skeletal muscle is around 10 to 55% of the total sports injury. The main reason of muscle injuries are contusion, laceration, strain, volumetric muscle loss, motor vehicle injury, burn and freeze, chemical ischemia, and contusion injury. If we see what is the healing pattern of this muscle, it takes around 21 days to heal the muscles. So we thought that to make a drug for something which which cure this disease. So we use a model which is called volumetric muscle loss or surgical traumatic muscle loss, which occurs in battlefield, in which a large part of muscles is shifted away. Previously, many drugs are used for treatment of volumetric muscle loss. They are NSAIDs and glucocorticoids. But on a long duration of run, NSAID causes loss of muscles and glucocorticoids causes irreversible destruction. So we thought to make a drug for volumetric muscle loss. So this is our target disease. Now the next part is that which drug should we use for treatment of the volumetric muscle loss. So if we look to the ARB class of drug, in that losartan has an effective against the contusion injury. So the thinking is that if losartan is effective in treatment of contusion injury, then the other drug, that is azilsartan, will also be effective in the treatment of muscle loss, that is volumetric muscle loss. And the other thing of azilsartan is that causes phosphorylation of AKT in the rat skeleton muscles. So we chooses azilsartan as our target drug, which we repurposed in volumetric muscle loss. So if we talk about the azilsartan belongs to Bacillus second class drug, which means it has poor aqueous solubility but high permeability. And if we talk about the bioavailability of that drug, that it has 60% bioavailability and T half is around 30 to T half is around 3 hours. So the next, if we talk about the route of administration, the next question arises in our brain: to which route should we administer this drug? So we chooses topical route. The main reason to choose topical route and give and make a transdermal hydrogel for it, because if we talk about the hydrogels, they are non-sticky and easily applicable, non-greasy, and do not cause any irritation and any side effects. So we chooses our formulation as higher. We should make a hydrogel for azilsartan. Then by findings, different researchers, we observed that there is no topical preparation is available for azilsartan. So the second thought arises in our brain: so how will we make a topical preparation, or which dose we should use for the administration of the azilsartan in that hydrogel? So for that, we compare different ARBs drug and what percentages of that drugs are loaded into the topical preparation. So we found that there are preparations for valsartan, losartan, telmisartan, irbesartan, and candesartan, whose topical preparations are already available in market, and the percentage of drug loading of valsartan is 1% and losartan is 0.5 and 1%, and so on. So if we observe this table, we found that 1% drug is loaded mostly in majority of the formulations. So we choose a one formulation that is 1%. The second dose which we choose is 0.5% because it is also used in most of the formula, and the lower dose which we have chosen is 0.1%. So we have three doses: 0.1%, 0.5%, and 1%. 1% of dot drug which we have loaded into our 1% carbopol gel, carbopol hydrogel. So the next thing is how we made that hydrogel. For preparation of the hydrogel, we use 1 gram of carbopol, and 1 gram of carbopol is dispersed in 100 ml of distilled water and kept for 24 hours so that it gets swelled. After that, we added dropwise triethanolamine to neutralize the pH and get a gel consistency. After that, we loaded our drug. As we know, azilsartan is not aqueous soluble. We have to load it into ethanol because it has a good solubility in ethanol. So we chooses 15% ethanol for loading of our drug. After dissolving the drug in ethanol, we loaded it into our.
It is
Yes,
3 minutes more.
Okay, sir. So after that, polyethylene glycol and is added, and we pack that formulation into airtight containers. Now next we move on to the how we induces the volumetric muscle loss. For that, we have divided the animals into 5 groups, 6 animals per group. In that, first group is uninjured group, second group is volumetric group, third group is ethanolic group, in which we made ethanol hydrogel and give it for 28 days. Fourth group is azilsartan 0.5%, in which we have given hydrogel for 28 days, and in fifth group 1% of azilsartan is loaded. Then for making of the injury, in that we first anesthetize the animal, then cut that gastrocnemius muscles of right leg so that the excision is 60% of its length, 75% of width, and 50% of the thickness of that muscle. After the creating of the injury like this, we sutured it and apply our hydrogel for 28 days. If we talk about the plan of work, my plan of work is divided into 2 parts: hydrogel evaluation and a muscle injury evaluations. If we talk about the formulation of azilsartan, we have observed that that azilsartan 0.1%, 5%, and 1% formulation has excellent homogeneity, extrudability, and spreadability, and has no grittiness. And when we increase the percentage of drug, the viscosity is also increases. Then we move forward onto the flux and permeability coefficient. Permeability coefficient and flux is good in 0.5% of drug in comparison to other formulation. When we talk about the drug release, the pattern of drug release of 0.1 is gooder in comparison to 0.5 and 1, but we chooses 0.5 and 1% drug for loading for continuing into the in vitro studies, because it shows a continuous pattern and follows zero order kinetics. Then we perform the skeleton race test and found that skin irritation, there is no irritation was found. Then we move into the in vitro result. In vitro result, we found that percentage weight we observed the percentage weight and there's significant difference in injury and VML. Then we move on to the gastrocnemius weight. Gastrocnemius weight, what is the difference in the gastrocnemius? We found a significant increase in gastrocnemius weight of 1%, 0.5%, ethanolic loaded formulation. Then we move on to the what is the gastrocnemius weight and length ratio. In that ratio, there's significant difference is found in 0.5 and 1% of hydrogel, and this diagram pictorially represented here that there is increase in gastrocnemius weight in 1% in comparison to other. Then we move upon what will be the effect on the muscle strength. When we talk about the muscle strength, there is significant increase in 1% while azilsartan in comparison to VML. and the force needed to break is increased, and the time required for its breaking is significantly increased in 1% and 0.5%. Then we move on to the what will be the effect of these formulation on the into the injury side. We observed injury on 0 day and 29 days and observed a significant decrease in injury areas in all these formulation. And if we talk about the injury width, there is significant decrease in 0.5 and 1% hydrogel. Then we move on to the protein estimation, and we found that an increase in protein is observed in all the formulation. Then we move on to the histology. In this, we found that there is a more number of infiltrations are shown in VMN in comparison to ethanol as a certain 0.5% and 1% formulation, and hypertrophy occur in 1%, and observe that there is significant increase in cross-sectional area of 1% ethanol in comparison to VML group, and a ferret diameter of all the three formulations increase in computer VML. Then we move on to the amount of MyoD. MyoD is a molecule which facilitate satellite cell formation. So in that we observe a significant decrease in MyoD synthesis by our formulation. We have literature review showed that MyoD level increases in initial phases of healing. After that, when we move further, then around 7 to 14 days, the amount of MyoD decreases, and after 20 years, it will disappear completely. So that pattern is shown by our study when we observe its amount after 28 days. Then we move on to the STEM and found.
I'm sorry to
that.
interrupt, but you are exceeding your permitted time.
Okay, sir.
I'm sorry to interrupt you, but the time has exceeded. You exceeded by 1 minute.
Okay, sir. Should I stop it now?
In further 1 minute.
Okay, sir. Thank you, sir. Thank you so much, sir. If we talk about the SAM study, that what will be the effect on its surface, we found that there is breakage in the superficial layer, but when we give treatment, their surface get healed, and smooth surface is observed in 1% hydrogel. Then we thought what will be the effect of our formulation on the fibrosis percentage. She is shown in VML when we perform massive screening and serious red screening, and when we give our formulation, there is decrease in the redness in serious red, and bluish content which depicts the fibrosis is decreased in 1%. So there's significant decrease in 1% VML if you compare the percentage fibrosis in masking factor. Significant decrease in 1%, and ethanol in 0.5 if you observe the serious red staining. Then we perform the antioxidant studies. In antioxidant studies, there is significant increase in GSH and SOD level, and significant decrease is observed in MDA and H2O2 level. Then we move on to the what will be the percentage of myostatin. Myostatin is the inhibitors of the myogenic factor. It shows that our formulation decreases myostatin level in the formulation. Next, we move on to the creatine kinase level. Creatine kinase is released in the blood when muscle injuries occur, and our formulation shows a significant decrease in creatine kinase level. So, in conclusion, we concluded that 1% of hydrogel is more effective in comparison to 0.5%. And if you talk about the pharmaceutical result, and when we talk about the mechanism by which azacitidine inhibits, it shows that it inhibits myostatin, which is responsible for protein degradation and fibrosis. So by inhibiting myostatin, it inhibits the process of protein degradation and fibrotic scar formation, and the decrease in MyoD levels shows that the muscle is reached to the last phase of remodeling phase, and concluded that azilsartan involves the down regulation of fibrosis and up regulation of muscle growth. And if you talk about both the formulation, the amount of positive results in 1% azilsartan is more in comparison to 0.5%. So concluded 1% azilsartan is more effective in comparison to 0.5%. Thank you, sir.
Thank you. Before I invite the respected judges to question you, I may appeal once again to all the candidates to stick to the given timings, 7 minutes before warning and 3 minutes further, and conclude your presentation in 10 minutes. Next candidate is Ms. Julie Jack.
Yes, sir.
I
Yes.
appeal her to be ready till the question answer session for this. Now may I ask
Sorry for
Dr. Sarkar to
delaying.
have queries.
You want me to ask Arish?
Yes, sir.
Okay. You could have reduced the number of slides first, and second, more practice necessary. You know.
Yes, sir.
Arish?
Yes, sir.
Yeah, more practice is necessary for this kind of presentation because you may not have practiced enough for this kind of conference. So yeah, I mean, a lot of work, and you wanted to put it all together in the one, like 10 minutes. It's not an easy job, no matter what. Okay.
Yes, sir.
So you have done quite a bit of work. So if you say like, okay, this azilsartan hydrogel accelerating this, particular muscle loss. So if you have to say in one sentence, how you reconcile, okay, what exactly the mechanism and how you can take it forward, this whole result.
Sir, azilsartan shows an inhibition on myostatin, which is responsible for protein degradation and fibrotic scar formation. And there is which if you talk about the mechanism in that the protein, by that protein estimation, that there is increase in protein formation. So we concluded that there is upregulation in muscle growth and down regulation of fibrotic scar formation and protein degradation by inhibition of myostatin.
Okay, thank you.
Over to you, Ms. Diman.
Yeah, I have one question. Like, what was the hydrogel retention time on the skin when you applied it?
Hydrogel retention time is around 1 hour.
Okay. And when your response was expected or observed?
Sir, in this we daily applied that hydrogel, and after completion of the study of 28 days, we observe what is the effect of those hydrogels.
What I want to ask, the pharmacological effect when it was observed post how many hours after the application of the hydrogel? Like suppose if you... Yes, please.
Sir, we haven't haven't taken any reading after the sacrifice of that animal. Then only we observe all the changes.
Okay, no issues. Thank you so much.
Are the judges satisfied? Should we move on to next candidate?
Please.
Yes. Thank you, Ms. Zera. Thank you, Ms. Zera. You have to restrict your talk to 10 minutes. Your work was very good, but you have to learn how to present it. I wish you all the best.
Okay,
I
sir.
wish you all the best for your personality.
I'm sorry, sir.
Next candidate. Next candidate, Ms. Julie Jagtap.
Yes, sir.
Yeah.
Shall I share this?
Her presentation is entitled as P24 and entitled Biotin Enriched Oral Dissolving Film as Nutraceuticals. Ms. Jagtap, are you ready with your presentation?
Yes, sir.
Yeah, put your slide. All the best.
Yes, sir. Thank you so much. So is my presentation visible?
Yeah, it is visible. Please start the slideshow.
Yeah, yes. A very good afternoon to all. I am Ms. Zoili Jagtap. Today I'll be presenting my topic on biotin enriched oral dissolving film as a nutraceutical. So according to survey which was made by International Society of Hair Restoration Surgery, approximately about 35 million men and 21 million women, they are affected by the hair loss in US itself, and 20% of them are from the age of 30. So to combat this problem or to look after this, we have formulated a dosage form, that is biotin loaded oral dissolving film. So basic reasons or the normal reasons for hair loss, it is vitamin B7 deficiency. Mostly in women, there is hormonal changes. It can be a hereditary issue. There can be stress-induced hair loss. Also, because of lifestyle changes, you can see it is affecting our hair quality as well, and some medical conditions. Why oral dissolving film? So basically, oral dissolving film, it is a thin polymeric strip which incorporating various pharmaceutical active ingredients, and once it is placed into the mouth cavity, within a very short period of time, it dissolves and it disintegrates without ingestion of water or chewing. So this is when administered on the tongue. Because of saliva, as this oral dissolving film has a saliva stimulating agent, so it starts to disintegrate, and through the oral mucosa blood lining, it directly enters into the systemic circulation, and hence it avoids first pass metabolism. So
there is no slide.
Sorry, sir.
There are no slides.
Shall I reshare, sir?
Yes.
Can you make it in the presentation mode?
Yes, sir. Is it visible?
Yes, but it is not in the presentation mode.
I have made it in a
Put it
presentation.
in slideshow. Yes.
Yeah, thank you, sir. The special features for the fast dissolving films are the films are very thin and elegant. They are available in various sizes and shapes. It's unobstructive. It adheres onto the oral cavity very easily. It has a very fast disintegration time without ingestion of water, and it also shows a very rapid release. There are
I'm
advantages.
sorry, sir, but is this slide visible?
I cannot see any slide.
No.
Yeah, this end also we are not able to see any slide.
So can I present in this mode?
Yeah.
Okay.
Okay.
Yeah.
Okay.
Yes.
Just increase the percentage.
Yes, sir. Yes.
Few more. Few percentage more.
Is it fine, sir?
Yeah. Okay.
The advantages of oral dissolving films are this dosing is very simple and it is very convenient. There is no risk of choking. It can be used anywhere without water. It is suitable for geriatric as well as pediatric. It has enhanced patient compliance if compared to the tablet dosage forms, and they are flexible, portable, convenient to handle, to store, and also to transport. The patients with dysphagia can also have these dosage forms. Why biotin? Because it's a water-soluble vitamin, and it is also to be proven as a hair conditioning agent. So it possesses rejuvenation characteristics, which helps in production of new cells. It also helps in preventing hair loss by rejuvenating it, both hair follicle as well as the scalp, and it is highly beneficial for preventing shedding and also helps in hair regeneration. Along with promoting hair advantages, it is also useful in conditioning nails and skin as well. It speeds up the metabolism. It is also helpful in balancing the blood sugar level and also the heart health. So this is the procedure and the formulation table. So in this, the water-soluble polymer, which I have used, we have tried with the two grades, HPMC E5 and HPMC E15. So these polymers were soaked overnight into the water and distilled water, and it was stirred for 2 hours with the magnetic stirrer, and after 24 hours, it was mixed with the excipients, which were also dissolved into the distilled water, And after complete dissolution and mixing of the two solvents, they were finally casted by the Petri plate method, which formed a very thin film when kept in hot air oven for 60 degrees Celsius for 1 hour. And after once we get the film, it was peeled off from Petri plates and later it was cut into the desired sizes. So the excipients which we used, it was HPMC E5 this, as a polymer, which is a very good polymer, water-soluble polymer. Glycerol was used as a plasticizer. Sorbitol was used as a sweetening agent. Citric acid as a saliva stimulating agent. Cross-povidone was used as a super disintegration agent. And we added some flavors and ascorbic acid, which is used or which is giving vitamin C as well. And here you can see these are 4 different flavors by which we had made the films. Among all these formulations which we had tried, F3 shown a very good evaluation parameters in which the thickness was around 0.59. The folding endurance was also the highest one. The percent elongation, it was also better than all of the other. The best quality or the best thing in these 9 formulations for F3 it was having a very short disintegration time, that is 20 seconds. Within 20 seconds, the film was completely disintegrated and dissolved. The weight variation for it was also acceptable, and the drug content was measured by UV. So these are the observations for all the these formulations. So from F1 to F3, HPMC E5 was used as a polymer. It was very clear and transparent film. The thickness was also very uniform. It had a very good flexibility, and it has shown a very good mechanical property. So by dose, this was the calculation for the dose. Label claimed or typically 20 to 30 microgram dose is expected. So we had kept 25 microgram as the ideal dose. So the conclusion is oral dissolving films, they are preferable because as you know, it requires less or no water, and it can also eliminate the unpleasant pleasant flavors from the drug, and it allows for rapid breakdown and disintegration. The person who is having dysphagia can also take oral dissolving film. In a situation where quick response is required, like motion sickness, allergic reactions, coughing, and acute pain, this type of dosage form can be a good help. And from these 9 formulation evaluated, F3 was showing the best, and it had appropriate thickness, the required plasticity, the drug content as per the label claim, and also it had good mouthfeel. That's it. Hello.
Yes.
Yes.
You have 3 minutes more. Have you concluded?
Yes, she has done with it.
Yes.
Okay. So now may I invite Dr. Dhiman to ask the questions?
Yeah. It was a nice presentation as far as application of oral films as a dosage form is considered. But I have a question in terms of why you have chosen biotin as a drug when the tablets, immediate release tablets are already available in 10 mg strength in the market. And as such, the Cmax of drug is achieved within 1 hour. And this drug is not used for disease management of a disease like pain management, where immediate action is required. It is used for a long-term treatment for 1 to 3 months for the hair fall related problems. Okay, so you need to justify your motive in the presentation why you have chosen the drug. And moreover, this drug, as far as I know, this is not sensitive to first pass metabolism also. So in that case, and as far as vitamins are concerned, they have a lot of stability challenges. So in terms of formulation as a film, these challenges will be more versatile, and lot of scalability issues are there.
Yes, sir.
So yeah, so what are your comments on that part?
Sir, I had seen that there is fast dissolving tablets as well. But then what happens with that is you need water. And what my motto to converting this into oral dissolving film was that it should be very convenient and as per the lifestyle changes, or it should be very easy to have it. Now, normally whatever this vitamin supplements we have, they are mostly in the this tablet form, or sometimes it is in the powder form as well. But then it happens like some people, they don't or the patient compliance issues may come. So I converted this into oral dissolving film, and as why biotin, because it has been shown that it is promoting hair growth as well as skin issues and nail conditioning as well.
Okay, and the dose which you have chosen is totally different from currently marketable packs. So how will you ensure the dose equivalency and frequency of administration?
So the frequency of the administration, the daily dose which is recommended for adults above 18 years, it is 20 to 30 microgram per day. So I had chosen as a 25 microgram we can have it. And as this is a nutraceutical product, so optimization of dose, I didn't give much emphasis on that.
Okay.
Okay. Should I take it that you're over?
Yeah, I'm done. I'm done. Yeah.
Okay. So Dr. Sakar?
I think Dr. Manish already asked most of the questions. So yeah, very nice presentation. I don't have any extra questions.
So thank you, Dr. Sakar. To move over, the next candidate in line is Mr. Kunal Bakul. His presentation encoded P25 is entitled Ball Milling Approach to Enhance the Dissolution Rate of Enzalutamide using Water-Soluble Polymers. Mr. Kunal Bakul, are you ready? Yeah.
Yes, sir. I'm audible,
best
sir.
of luck. Yeah, best of luck.
Yes, sir. Thank you.
Put your slides, share them.
The slides are visible, sir?
Slideshow. Yes.
Yes.
Start. Your time will start as soon as you shift from your title slide.
Okay. Good afternoon, sir. My name is Kunal Bagul. I am doing research work under Dr. Atish Kumar Mundada, sir. My topic of today's oral presentation is ball milling approach to enhance the dissolution rate of enzalutamide using water-soluble polymers. These are the contents of my presentation: rational and objective, material methods, result and discussion, conclusion, and finally references. I have selected enzalutamide, which is a anti-cancer drug used in the treatment of prostate cancer. Enzalutamide is a BCS class 2 drug having a rapid crystallizing property. This is a BCS class 2 drug having solubility limited absorption in vivo. Increasing the dissolution rate of enzalutamide ultimately increases its bioavailability. The objective of this study to enhance the dissolution rate of enzalutamide using ball milling and the water-soluble polymers, that is Soluplus and Poloxamer P188. These are my materials: enzalutamide, Soluplus, and Poloxamer. Soluplus and Poloxamer supplied by BSF, and enzalutamide supplied by Shilpa Medicare. These are my steps, materials and methods. Firstly, I have done a drug excipient compatibility by mixing the polymers and API and kept at 40-75 degrees Celsius for 1 month in closed glass vials and evaluated for assay. The physical mixtures were prepared by mixing in the mortar and pestle and sifting through the 60 sieve. After that, I had developed analytical method and characterization of the drug co-mixture. This analytical method were developed using UV visible spectroscopic method for the analyze, assay, and dissolution. After that, I have prepared drug co-mixture using a ball mill. Firstly, I have tried binary mixture, that is Soluplus and enzalutamide and Poloxamer and enzalutamide. In that, I have observed that initially it shows enhancing the dissolution rate But after keeping at 75 degrees Celsius for 1 month in closed HDP containers, it is showing a significant decrease in the dissolution rate of the enzalutamide. After that, I have prepared the ternary solid dispersions with Soluplus, poloxamer, and enzalutamide by ball milling technique. Initially, it was showing enhancement in the dissolution rate, and there is no significant decrease in the dissolution when kept at 40-75 degrees Celsius for 1 month in HDP container. I have used this ball mill. This ball mill is working on the principle of impact and attrition. I have used around 50 to 60% occupancy in the ball mill of material and balls. After that, result and discussion, the drug excipient compatibility study analyzed for the assay, the initial results and the 1 month 40-75 drug excipient compatibility results showing no significant change from the initial values. The second one is the percent yield and drug content. The percent yield of ternary solid dispersion prepared by ball mill showing percent yield in the range of 82% to 86%, and the percent drug content in the range of 98 to around 100%. After that, I have analyzed the it for a dissolution profile in 0.1 normal HCl plus 0.5% SLS. These are the initial results of binary and ternary solid dispersions, whereas after keeping for 1 month at 40-75 degrees decrease in the dissolution rate except the batch number T004. After that, these are evaluated for the FTIR analysis to check the chemical any chemical reaction. The initial the enzalutamide characteristics peaks of the enzalutamide are present in the batch T004. It is showing no chemical interaction. After that, it is evaluated for the DSC. This is the DSC of enzalutamide showing a sharp endothermic peak at around 200 degrees Celsius, and this is the DSC of physical mixture prepared. It is
Mr.
showing,
Bagul, 3 minutes more.
yes, sir. It is showing enzalutamide peak at 200 degrees Celsius and polymer peak at around 54 degrees Celsius. After the evaluation of T004 for DSC, the disappearance of enzalutamide peak was observed, and a shift in the endothermic peak at around 60 to 65 degrees Celsius. It is showing the conversion of crystalline enzalutamide to amorphous enzalutamide. It is also confirmed with the PXRD diffractogram. This first one is the crystalline enzalutamide. This is polymer XRD. This is physical mixture XRD, and this is drug co-mixture XRD, which is showing the hump. That is no sharp peak of the enzalutamide, which is in the amorphous form. After that, these T004 evaluated for flow properties and PSD evaluation. It is showing compressibility index around 20%, that is showing a fair flow, and a particle size distribution showing a D90 at 16 microgram. The initial particle size of enzalutamide as per the COA is around 140 microgram. D90. After that, these T004 and enzalutamide API evaluated for ex vivo intestinal drug absorption study using this modified apparatus. It is having arm A and arm B. On arm A, we are hanging the goat freshly procured goat intestine, and it is kept in the dissolution media, and the samples were taken from the arm A. It is showing the enhancement in the dissolution at enzalutamide API. It is showing 19.25% at 60 minutes, while the T004 showing 98% at 60 minutes. After that, this batch kept at stability at 40-75 degrees Celsius in HDP container. It is showing no significant decrease from the initial values of dissolution as well as the assay. The conclusion, the in vitro dissolution studies and ex vivo intestinal absorption studies clearly shows that prepared drug co-mixture enhance the dissolution rate of enzalutamide. The future prospect of the study, we can use this drug co-mixture to form the solid dosage forms such as capsules, tablets, etc. These are the references. Thank you.
Yeah. Thank you, Mr. Bagul, for completing in time.
Yes, sir. Thank you, sir.
May I now ask Dr. Sarkar to have his queries?
Yeah.
Dr. Sarkar?
So, is this method new for you, or is it the first time you're using, and how does it correlate with the existing methodologies?
Sir, in the market, there are many formulations available. In the US market, it is a soft gel capsule of enzalutamide present, and in the India market, it is a hard gelatin capsule present. But they are using a solubilizer, that is poloxamer. Sorry, not poloxamer. Labrazole ALF they are using. That is an oily substances they are using as a solubilizer. And we are formulating with the newly combination of polymers. It is in the patent they have used HPMC CP as a polymer to enhance the dissolution of enzalutamide, and we are using the combination of Soluplus and poloxamer.
So
and the
according
method is also they are using by spray drying, and my method is by using ball mill. It is compared to spray drying, ball mill is a slightly time consuming method, but they have mentioned this, they have claimed the spray drying technique in the patent.
So your formulation has more intestinal absorption?
Yes, sir. But compared to enzalutamide API, but I have not procured the samples from market. And not evaluated for means my formulation versus marketed formulation.
Thank you.
Yes, sir.
Dr. Sarkar, are you over?
No,
Over with Mr. Bagul? Okay. May I now invite Dr. Dhiman?
Yeah, Kunal, this was in fact a nice presentation.
Thank you, sir.
You covered almost all the aspects, starting from pre-formulation to covering the in vivo, in vitro, in vitro assessment, intestinal permeability, as well as stability.
Yes,
so during this course of presentation, I came across through doubts, which are like, you have done the ball milling, right? At 50% occupancy, which includes your API plus co-solvents or solubilizers.
Yes.
It works on the principle of impact and attrition.
Yes,
so,
sir.
like, which balls you have used, and what was the particle size of those balls?
Sir, we have used steel balls having a 316 SS of 316 SS, and the diameter of our balls are 9.80 mm balls we have used.
And which mill you used?
Ball mill. Actually, it is a locally made mill, sir. Not.
But, did you optimize this ball size? How long it took for you to reduce particle size from 140 to 20 micron?
Sir, it took around 12 hours at 100 RPM we have run. This is the maximum speed of a ball mill. That's why I have.
If you could reduce ball time, ball size from 9.8 mm to further, you can reduce.
Okay,
you will get more efficiency, and maybe in half time or whatever you have currently done, you can achieve your milestone.
Yes, sir.
Since this is a BCS class 2, a challenging molecule, as per my understanding and work I have done on this molecule.
Yes, sir.
And you should have evaluated the available sample in the market for intestinal permeability, and that could have added value to your presentation.
Yes, sir. This has been it is come in future prospect, and also one thing, the cost of the Indian formulation is around 28,000 for around 14 capsules, sir.
Okay.
That's why I'm not.
I know innovator is very costly.
Yes, sir. And US innovator is around 6 to 7 lakhs for around 50 or 60 capsules, sir.
Yeah, I do. Extendi you must be talking about.
Yes, Extendi soft-gel capsules.
Yeah. No issues. It was a good presentation.
Thank
Keep
you,
up the good work. Yeah.
Yes, sir. Thank you, sir. Sir, can I stop the sharing?
Yeah, please. Thank you.
Thank you, sir. Thank you, sir.
Thank you, Mr. Bakul. And thank you, Dr. Diman, for your expert comments.
Thank you, sir.
Should I now move to next presentation entitled P26? Mr. Mahesh Shantaram Patil.
Yes, sir.
Yes, you
Can
are
I
available?
Yes,
Yeah.
sir.
Okay, share your slides. Your topic is entitled A Comprehensive Study on Regulatory Requirements for the Submission of a Post-Approval Changes,
Yes, sir.
specifically in Brazil, USA, and Europe.
Sir, is the
So,
slide visible?
we will hear from Mr. Mahesh. No,
Yes,
your
sir.
slide is not visible yet.
Sir, now?
Not
No.
yet.
Hello, sir.
Yes, you are not visible. Please put yourself on camera.
Sir, 1 minute.
Meanwhile, I will be declaring the name of next candidate. I will be
No, sir, no,
appealing
sir.
him to be available.
Wait for a minute, sir.
Yeah. I will call on next candidate to be ready. Mr. Nirmal Sharma. Sorry, Mr. Nirmal Shah.
Sir,
Is
now
Mr. Nirmal Shah available?
Sir, screen is visible now?
Yeah, screen is visible. Your presentation is not yet.
Yes, sir. Wait for a minute.
It's an appeal for Mr. Nirmal Shah to be ready with his presentation.
Sir, is visible?
Yeah, it's visible, but it's, yeah.
Okay.
Yeah, very much. Best of luck, Mr. Mahesh.
Okay, sir. Thank you, sir, for compromisation. Today I am going to deliver oral presentation on the comprehensive study on regulatory requirement for the submission of post-approval changes, specifically in Brazil, USA, and
Excuse
Europe.
me, Mr. Mahesh.
Yes,
You
sir.
should be on the camera.
Yes, sir. There is problem regarding gadget. Can I deliver like this? Hello, sir.
Yeah. I may ask both the judges if they are comfortable with.
Yes,
Yeah.
yes.
Let's see.
No
Okay.
issues.
You may continue.
Okay, sir. Thank you, sir. Basically, the table of content of the this oral presentation is introduction, aim and object, and discussion. Basically, the post-approval changes. It's post-approval changes is the any change to any aspect of pharmaceutical product, including but not limited to formulation, method, and packaging, but also include the manufacturing side, finished product specification, container product, ingredient product labeling, and information. Sir, means basically post-approval changes is unavoidable part of the organization that includes the overall the product life cycle management. Whenever the any changes related to as part of the quality or regulatory guidance, it may affect to the quality, safety, efficacy of effectiveness of the product. That's why we need to submit the post-approval changes to the regulatory authority that our product in the approved manner, that is will not affect to the safety and effectiveness of the product. Basically, there is diverse mechanism from for various reasons of regulatory authorities. Then I have decided to study only few reasons, that is Brazil, USA, and Europe. That the regulatory authority for Brazil is ANVISA, and USA for USFDA, and Europe is EMA. Sir, I have studied only. I have concluded only superficially about the post-approval changes, not detailed, because it is very theoretical and it cannot be concluded all data in 8 slide of the presentation. Then discussion about the post-approval changes as per ANVISA. The UNVISA have the post-approval changes referred as the impact on the quality, safety, and efficacy of the product. They are classified as follow. Being for immediate means if no prior notification required to the regulatory authority, it will be implemented and submitted to the authority after the 1 hour or that period. And with or without individual protocol means there is protocol required, or without protocol we submit post approval changes and subject to prior approval means that we can submit the post approval changes before the changes implemented, and the authority review and analyze that change will affect effectively, or any adverse effect regarding to that change will affect to the product. Then UNVISA give us the approval to the product. Then next is post approval changes as per US FDA. Mainly US FDA referred as the scale up and post approval changes, and they are classified into 3 categories, that is major changes, moderate changes, and minor changes. The major changes means that is required a prior approval supplement, means that the major changes occur, if many changes occur, that will affect to the product effectiveness, quality, or any adverse effect occur due to this major change. Then we need to submit prior approval supplement that will affect to the product effectiveness. And then next is moderate change, that is CB0 and CB30. CB0 means that the changes being affected within 0, that means we not clarify that only implement the change and they will we will submit the post approval change. And CB30 means the wait for 30 days after the submission of post approval changes. If any query, if not query occur for the post approval changes, then the product is in the distribution for the market recording. And then next is a minor change. Minor change is annual report. Basically annual report is the minor changes. All changes are implemented during the 1 year of duration. Then we submit that changes in annual report. The next one is post approval changes for EMA. The EMA, European Medicine Agency, referred to as a variation. The variation has 2 types, type 1 and type 2. That mentioned the type 1 is the type 1A is do and tell. Type 1A notification that do and tell, and type 1B tell, wait and do. Basically, the type 1A do and tell means first do and tell to the authority after 12 months, and do and tell that is require immediate notification after the implementation of our post approval change. Then type 1B, that is tell, wait and do, that requires wait for 30 days after submission of procedure. Then next one, type 2, that is tell and do, that requires usually 60-day review, range for 30 to 90 days. Okay, thank you, sir.
Okay, thank you, Mr. Mahesh.
Thank you, sir. Sir, actually, I have not concluded all the data specifically for which post approval changes, because it is very theoretical.
Yeah, but it is sufficient. You have concluded. Okay, you have concluded.
Okay.
Okay, you have concluded well in time. Within at the end of 7th minute, you have concluded. Now, may I invite Dr. Sarkar?
Yeah. Hey, Mahesh, can you hear me?
Yes, sir. Yes, sir.
So, first question: what prompted you to have this particular study?
What's the? I am not audible. What question?
Hello.
Can you hear me?
Hello. Yes, sir.
I said, what prompted you to do this study?
Hello.
Hello. First, Dr. Sarkar.
Hello. Yes, sir.
In the meantime,
I think he's having connection issues.
yeah, ask Dr. Dhiman to ask.
Yeah, Mahesh.
Yes, sir.
I'm audible. Yeah,
yes,
see,
sir.
This was a nice presentation. Most of the things, though, you have taken from literature, but were nicely compiled and explained to the audience. I have a couple of questions. Like when you were comparing USA, Europe, and Brazil, why did you keep Brazil as a first slide?
Sir, actually, I am not comparing. I had
Yeah.
only reviewed it. That why that.
Okay. Because as per the industry trend, people always keep USA
Yes.
on the top priority.
Yes, sir. Yes,
Right?
sir.
Okay, so you explained it nicely about CB0, CB30, and fast approvals.
Yes.
But yeah, you could additionally value addition you could have done is like the estimated time for all the approvals you could have incorporated in the slide, so the audience could have well understood. Anyways, it was a nice presentation, so I have nothing more to ask. Yeah, thank you.
Okay, sir. Thank you, sir. Sir, any suggestion?
No, you can
Yeah,
incorporate more
he has
details.
already suggested. He has already suggested. You have, if you would have presented the time span spent to
Yes, sir.
seek the approval.
Yes, sir. There are limited
For all
slide
three.
and time also. That why I concluded in very less.
Okay. I think Dr. Sarkar is back.
Okay, sir.
Are you here, Dr. Sarkar?
Yeah.
Yeah. Your comments, please.
Yeah, what I'm saying, so I think you already said, what prompted this particular study, like you must have some kind of intrigue, like you must be intrigued to this particular thing. So what prompted you to do this particular company? It's a nicely presented, information for, so what exactly prompted you? That's what I wanted to know.
Sir, I can't understand.
Mahesh, Dr. Sarkar is asking, like, what prompted you to choose this topic?
Okay, sir. Sir, there is a organization have the same product has a different means that various post approval changes we need to submit in various region. Then it's difficult to the organization that submit the changes to various regulatory authorities. Then I am reviewing that what all requirement for that to submit the post approval changes in one place. Okay, and I am further studying about the specifically for the composition change, but I did not mention that in this slide.
Okay. Yeah, I mean, because more often than not, people wanted to know how the whole approval process goes, like phase 1, phase 2, phase 3, phase 4. Then only the regulatory authorities, it can US FDA or any other authorities, any other country will be following, right?
Yes, sir. Yes, sir.
So what you presented is the post approval, like even after approval, after approval,
yes,
the
sir.
market for a long time, it can be taken away because there's too much toxicity.
Yes, sir. Yes.
Okay.
Okay, sir.
Okay.
Thank you,
Okay,
sir.
are you done with Dr. Sarkar? Okay.
Yep.
So, thank you, Mr. Mahesh. We'll be shifting to next candidate, Mr. Nirmal Shah. Mr. Nirmal Shah, are you there? Hello. I think Mr. Nirmal Shah is not available. He was seen in the chat room few minutes ago. May I have another candidate, Mr. Pawan Kumar Domeja? Mr. Pawan Kumar Domeja, are you available? Not yet. May I have another candidate, Ms. Shilpa Borate? Ms. Shilpa Borate?
I see her on.
Yes, sir.
Yeah. This is to the respected evaluators. If these two candidates appear in between, we will be calling them after the end of the session. At the end of session means when it concludes, P30 presentation is concluded, we'll be calling them, we'll be giving them a chance. So, Ms. Shilpa Borate, are you ready with your presentation?
Yes, sir. Sir, should I share my screen?
Yeah, you should share your screen as well as you should put yourself on camera.
Okay, sir. Okay.
The presentation is encoded P29 for the judges to mark. The title is Formulation and Evolution of Curcumin-Loaded Nano Sponges.
Hello, am I audible, sir?
Hello. Now Ms. Shilpa Borate is presenting.
Am I audible, sir?
Hello.
Sir, is my screen visible to you?
Yes. I think Mr. Nirmal Shah is calling. It should be noted that Mr. Nirmal Shah will be presenting after the conclusion, after the presentation of Ms. Simona, the last presentation.
Sir, is it visible?
Yeah. Yeah, it is.
Okay, yes. Okay, sir. Thank you, sir. Sir, very good afternoon, one and all. I, Shilpa Borate, presenting here on a title Formulation Evaluation of Curcumin-Loaded Nanosponges. So these are the contents of my presentation: introduction, objective, methodology, formulation and evaluation of nanosponges, conclusion and references. Introduction. So we are very well known about this herb, that is Curcuma longa, obtained from the Gingiveraceae family. So from this.
Excuse me, Ms. Shilpa.
Yes,
you are
sir.
supposed to put yourself on camera.
Just a minute, sir. Now, is it okay, sir?
Yep. Yep.
Yes, sir.
Is she visible?
Yes.
Okay. Continue.
Okay, sir. Thank you. So, phenolic content present in this Curcuma longa, that is curcumin, des-methoxycurcumin, and dimethoxycurcumin, sorry, and bis-dimethoxycurcumin. So, from this, I have selected curcumin, but this curcumin is having applications like antibacterial, antifungal, anti-inflammatory, antioxidant, and anti-psoriatic activity, along with wound healing property. But the limitation of this compound is it is having very low bioavailability, then having hepatic metabolism, then poor pharmacokinetic properties. So because of this, the best dosage form I have selected, that is curcumin nanosponges, nanosponges dosage form, sorry. So this dosage form may offer sustained release delivery of this curcumin and can enhance bioavailability of this curcumin. Methodology: pre-formulation study and formulation and evaluation of nanosponges. In pre-formulation study, I have evaluated this curcumin for this parameters: physical state, appearance, color, odor, and melting point. And all these experimental findings are matching with this reported values. Then I have analyzed it for UV spectroscopy in the range 200 to 800 nanometer. And in observation, I found that the curcumin is showing lambda max 424 nanometer, along with the linearity range 1 to 7 microgram per ml with the R square value 9982. Then I performed FTIR analysis of curcumin along with all excipients. Okay. This drug and excipient ratio is 1:1. So there is no significant change in the peak of drug and physical mixture. So there, it has shown to me that there is no chemical incompatibility in this physical mixture. And then these are the formulations N1 to N7. So in this formulation, I have varied the concentration of ethyl cellulose from 12
12,000 milligram to 1,000 milligram, and prepared these 5 batches. I have prepared these nanosponges by using solvent diffusion method. In this solvent diffusion method, I have taken ethyl cellulose as a polymer, then drug curcumin, and dissolve it into this 20 ml dichloromethane. It is used as a dispersed phase. Then fixed amount of polyvinyl alcohol, that is 1% polyvinyl alcohol, prepared with the help of magnetic stirrer, and it is used as a continuous phase. Then this dispersed phase added in this continuous phase dropwise with a continuous stirring for 4 hours with the help of magnetic stirrer at 1000 rpm, and then mixture is filtered and dried in oven at 40 degrees centigrade. Then prepared nanosponges evaluated for these 3 parameters: particle size, then production yield, and entrapment efficiency. So I found that the batch N4, which is having a concentration of ethyl cellulose up to the 300 milligram, and N nanosponges batch N4 and N5. So these 2 batches are showing me the particle size in the range of nano size, whereas this N1, N2, and N3, these 3 batches showing me the particle size in the micrometer. Then production yield for this all batches calculated. And then third parameter is curcumin entrapment efficiency. So again, this entrapment efficiency is also high in N4 and N5 batch as compared with other 3 batches. This is the particle size obtained by using optical microscope for N4 and N5 batch. Then conclusion, a drug excipient interaction was not observed. Curcumin loaded nanosponges were formulated by solvent emulsion diffusion method by using ethyl cellulose and polyvinyl alcohol. Then this formulated batches evaluated for these 3 parameters: production yield, entrapment efficiency, and size analysis by using optical microscopy. And curcumin and ethyl cellulose ratio influences the size of nanosponge. As we are increasing the concentration of this ethyl cellulose in the nanosponge, the size of nanosponge increases. So drug and ethyl cellulose ratio in between 1:1 to 1:3 is useful for the preparation of nano-sized formulation, or especially nanosponges. Then future scope, optimization of nanosponges required to be do. Then formulation and optimization of this nanosponge loaded topical gel. These are the references. Yes, sir.
Thank you, Ms. Shilpa.
Thank you, sir.
You have completed well in time.
Thank you,
May
sir.
I ask Dr. Sarkar to have his comments?
Nice presentations. I have few questions. Well,
Thank you, sir.
Curcumin, or nowadays, there are so many companies making nano curcumin. I think you are pretty much well aware of that, right?
Yeah. Yeah.
Still, the bioavailability or the distribution of curcumin where it's supposed to go is not as good as it's supposed to, right? So in that context, so do you think your new formulation will give an answer?
Sorry, sir.
Do you think your formulation in the nanosponge, specifically for this curcumin delivery or curcumin availability in the targeted organ or targeted region of the full body, is have better? better reach?
Sorry, sir, but I am not getting your question.
Putting into other words, Dr. Sarkar is asking how will you float your formulation, your laboratory formulation into the market and compete and make it compete with the marketed batches, make it marketed formulation, sorry.
Okay, okay. Sir, optimization is required to do, first of all, for this nanosponge formulation. And then I'll compare it with the marketed formulation.
Thank you.
Okay, sir. Thank you.
Dr. Dhiman, please.
Yeah. So Shilpa, like curcumin is having low oral bioavailability, and your research work is oriented towards topical gel formulation.
Yeah, yes.
Okay. So I would like to have your insight on stability aspects of your formulation.
Stability aspects of.
Yeah. So what parameters you are going to evaluate, and did you have you established any of the CQAs, critical quality attributes, during your development for your formulation?
So if I say from a methodology point of view,
no, testing point of view, I'm asking. CQAs means like testing, end product testing or testing during stability.
Okay. So the content estimation is one of the parameter in stability study. How much percent of content is being retained during this stability study?
Okay.
Then dissolution profile. Or you
okay. Hello.
Yes, sir. Yes, sir.
Yeah. So apart from this,
so in case of nanosponges only, the release profile, then dissolution study, then drug content,
viscosity.
Yes. When I incorporate in the gel, then viscosity is also one of the parameters for the stability testing. But
what is dosage frequency for your formulation? How many times a day you need to apply this gel?
Dosing. So basically, sir, I am preparing these nanosponges for the cosmeceutical use.
Okay.
So I am going to prepare this gel along with a few more vitamins. So it is used as a cosmeceutical.
Okay. Okay. It was a nice presentation. Thank you so much.
Thank you, sir. Thank you.
I think Dr. Dhiman is satisfied with the replies presented by Mr. Shilpa.
Yeah.
Thank you, sir.
Shall we move over to the next presentation entitled Assessment of Hepatoprotective and Nephroprotective Potential of Nerolidol on Vistar rats by Ms. Simona D'Souza.
Yes.
Yes, sir. Am I audible, sir?
You're audible. Will you please put yourself on camera and put your slides?
Yes, sir.
The remaining two candidates, take note that the two candidates, Mr. Nirmal Shah and Mr. Pawan Kumar, entitled P27, P28, will be given a chance after this presentation. Okay. Okay, Ms. Simona?
Yes, sir.
Over to you.
So is my screen visible?
Yes.
Good afternoon to one and all.
Slide
My
is
name is
not-
Simona D'Souza. Yes, sir.
Okay, start.
Good afternoon to one and all. My name is Simona D'Souza, and I'm from MET Institute of Pharmacy in Nashik, and I'm here to present on my research topic, assessment of hepatoprotective and nephroprotective potential of nerolidol on Vistar rats. So there's a prevalence of toxicity which is caused to the liver as well as the kidneys. One of the main reasons as to why this happens is because of oxidative stress. In worst case scenario, if you want to treat such a condition, we could one of the possibility is organ transplantation, and in worst to worst cases, it can also lead to morbidity. So hence, we have tried and focused our concern towards hepato and nephrotoxicity. For that, we have taken a drug, nerolidol. Nerolidol is a sesquiterpene alcohol which shows antioxidant property. So the basic hypothesis which we have laid for our project is that we would be inducing oxidative stress into our animals, which with the help of an inducer, which would create reactive oxygen species and in turn cause oxidative stress to the cells, causing lipid peroxidation and various decrease in various antioxidant enzymes, leading to hepatotoxicity and nephrotoxicity. Our drug, being an antioxidant in nature, will have free radical scavenging activity, which will reduce the oxidative stress and will restore all the antioxidant levels and will improve improve the cells of the liver and the kidney. So the material and methods for the research work was the chemicals which we had used was alloxane. Alloxane is an inducer which we have used to create oxidative stress into our animals. Nerolidol is our drug of choice, that is our test drug, which would help to cure and protect the liver and the kidney from oxidative stress. Ascorbic acid is our standard drug, which we will compare nerolidol to. The experimental animals used is albino Vistar rats. We have conducted the study for a period of 14 days. Now, the grouping of animals, we grouped our rats, into 6 groups containing 6 animals each. So the first group was a control group, which we did not induce any oxidative stress, nor did we cure by giving any of the drugs. We just administered plain distilled water orally to the rats. The second group was a negative control group where we induced oxidative stress with the help of alloxane. We have used 120 mg per kg IP. Group 3, 4, 5, and 6, we have induced oxidative stress with the help of alloxane. And after a period of 72 hours, we have started our treatment period. Group 3, which is the standard group, we have given ascorbic acid in the range of 250 mg per kg orally, and group 4, 5, and 6 have given our test drug nerolidol in the range of three different doses, that is 100, 200, and 300 mg per kg. So the experimental design is, first we conducted the in vitro antioxidant assay to see how much of a good antioxidant our drug is. We compared it with a standard, that is ascorbic acid. We conducted the DPPH radical scavenging assay, wherein we got the graph for ascorbic acid. Our drug showed good antioxidant activity. The next was a dose selection for alloxane and the drug. It was done through literature survey. The drug we chose, the dose of the drug nerolidol was taken by the LD50 of nerolidol, which was reported as more than 2000 mg per kg. So taking the 1/10 of the dose, we came to a conclusion of taking 200 mg per kg as the dose of the drug. And to get a range of doses, we chose 100, 200, and 300 as low, medium, and high doses, and was given orally. So we dosed the animals for a period of 14 days. Alloxane was given intraperitoneally, and our drugs, ascorbic acid and nerolidol, was given orally. Next, we conducted the biochemical estimation of the serum samples. We collected the blood from the retroorbital plexus of the rats. We centrifuged it and got the serum, and we conducted the biochemical estimation of aspartate aminotransferase, alanine aminotransferase, alkaline phosphatase, lactate dehydrogenase, cholesterol, triglyceride, total bilirubin, urea, total protein, and creatinine. These were the biomarkers which we had estimated. Then next, we conducted the in vivo antioxidant enzyme level estimation, where we isolated the liver and the kidney by sacrificing our rats. We homogenized the liver and the kidney, and we conducted tests like lipid peroxidation, reduced glutathione, and catalase. And finally, we sent the samples of the liver and the kidney. Yes, sir. Liver and the kidney for histopathological estimation. The results of the biochemical estimation, as we can see, we got the normal readings. For the negative control, which was shown in red, there was a high increase in the level of the biomarkers of the liver and the kidney, showing increased oxidative stress. The standard drug decreased to a very good extent. As well as nerolidol, our drug, also decreased it to a good extent. Looking at the graphs, we can see nerolidol 300 mg per kg showed good results as compared to 100 and 200 mg per kg. These are the results of the antioxidant activity. This is for lipid peroxidation. There was increase in lipid peroxidation when we induced only alloxin, whereas our drug decreased the lipid peroxidation to a good extent. Even our drug nerolidol showed good decrease in the levels of the lipid peroxidation. Glutathione catalase, which are necessary for removal of the hydrogen peroxide and the breakdown of hydrogen peroxide into oxygen and water. There was decrease in the oxidative stress created animals, whereas our drug showed an increase in these levels of the antioxidant at the end of the study. This is the histopathological examination. As we can see in the for the liver and the kidney, the normal control showed good architecture of the cells, whereas no disruption of the cells, whereas the negative control showed infiltrations, vacuoles of the hepatocytes and the nephrocytes. Standard drug, there was restoration of the cells, whereas our drug nerolidol 100 and 200 mg per kg showed good results, not as much as the standard, but our drug in the range of 300 mg per kg showed better results as compared to 100 and 200 mg per kg. Hence, we could say from the biochemical estimation and the antioxidant activities, as well as a histopathological examination, that alloxin-induced rats were attenuated considerably in nerolidol-treated animals. So this could lay as a further research work to be conducted, further studies. These are the references. Thank you.
Thank you, Ms. Simona. May I invite Dr. Sarkar to have his comments?
Yeah, nice presentation.
Thank you.
Nerolidol. So one technical question, when you injected this to the gavage, do you recall exactly the gauge of those gavage?
So could you please repeat your question?
Do you remember the gauge, like what size of the gavage needle you used?
For rats, we have used 14 for oral administration.
So, and I saw you used ascorbic acid, right? Vitamin C, right?
Yes.
That itself is a scavenger for oxidative stress, like specifically ROS, reactive oxygen species, right?
Yes. Yes, sir.
So if you compare that with the nerolidol in the different stages, how much it differs?
Yes. Like, sir, our aim was not to show that our drug is better than that, but there are other drugs which could show antioxidant activity and reduce the radical have radical scavenging activity and reduce oxidative stress in rats. And it could further be used as hepatoprotective and nephroprotective agents.
Good. By any chance, do you know whether this particular drug also affects in the brain at all? Does it cross the blood-brain barrier? Anything like that? Any idea?
So could you please repeat again?
I said this particular drug, I don't know, I'm just asking you, can you please shed some light whether this particular drug can be used as a neuroprotective agent as well?
No, so till now there has not been any research being conducted on neuroprotection, but there has been other activities which has been conducted, like antimicrobial or skin penetrating agents. So these are the research which has been conducted on this particular drug.
Thank you.
Thank you, sir.
Okay, Dr. Dhiman.
Yeah, it was a nice presentation. She has well presented the research work. No further questions.
Thank you, sir.
Okay, both the evaluators are well satisfied with the presentation. May I thank Mr. Simona for her good presentation. May I now call upon Mr. Nirmal Shah if he is available for presenting his presentation on solubility and dissolution rate enhancement of poorly soluble telmisartan using hydrotropy method. Mr. Nirmal Shah, I think he has not reported. Another candidate, Mr. Pawan Kumar Dhumeja, is available. His presentation is encoded P28.
Yes, sir.
Okay, no problem.
Yeah. Will you please put yourself on camera?
Yes, sir. I'm on camera, sir. May I share the presentation, sir?
Yes, yes.
Just a second, sir. I'm unable to do so.
Yeah, will be allowed few seconds.
I think in the background.
The screen is visible, sir.
Yeah, but not your presentation yet. Where is your presentation?
Is it visible?
Yes.
Yes. Yeah.
So, good
best,
afternoon,
Mr. Pawan.
myself, Pawan Dhumeja. Thank you so much, sir. Thank you so much. Good afternoon, everyone. Myself, Pawan Dhumeja. I am tab means is the on
Poor
the
connection.
still. Hello.
We barely hear you.
So first of all, we need to understand that why
Hello. Hello. Hello.
I think he lost his connection.
Yeah, I think we lost him. Yeah, he has reconnected.
He's coming back.
Yeah, he has come. Mr. Pawan? Yeah. Will you please put yourself on camera? You're not audible, Mr. Pawan. Please put on your mic. Mr. Pawan, you are still inaudible. Dr. Sarkar and Dr. Dhiman.
Yes, sir.
Is he audible?
No.
He is seen to be speaking. Mr. Pawan, do you require some more minutes?
Am I audible, sir?
Yeah.
Yeah, for this first time you are audible.
Yes.
Speak out something.
Yeah. We need to understand before starting the presentation that why cleaning validation is required, and for that we need to understand there may be the possibility that two and more formulations may be prepared on the same equipment. So before starting, so we need to check whether the cleaning procedure done is effective or not, and for that we have developed the method of cleaning validation. By that we will assure that the cleaning procedure is the effective to use for the next dosage form, and this is very important, especially when the drug product is the worst case for the cleaning procedures regarding the solubility, therapeutic potency, toxicity, and the surface adherence of the API. So first of all, we need to understand the limits, how much carryover of the first formulation to the next formulation is allowed, and we have put some guidelines of the US FDA PICs and etc., which you can see. So according to the US FDA, 10 ppm of the first formulation is maximum limit allowed for the next formulation. So after that only the cleaning procedure will be we can use on, or other criteria are 1 by 100 thousands of the normal therapeutic dose of the previous drug, first drug. Second is of the PICs, and then ICH Q7A and Q7 Q9 guidelines. And according to that, based on the minimum known pharmacological or toxicological or physiological activity of the API or its most deleterious component. And other guidelines or limits you can see on the screen. So first of all, what we have done is we have prepared the 50 mg equivalent to the favipiravir drug formulation, as we have taken the formulation and we have prepared the 1000 ppm of the drug solution, and after that we have taken the linearity using the UV visible spectrophotometry. So what we have found is the analytical wavelength was found to be 323 nanometer, and the linearity range of 2 to 10 ppm. And we have found the correlation coefficient of 0.999 and LOQ was 0.8 ppm. So 0.8 ppm concentration was then we prepared the 6 aliquots of 0.8 ppm, and we measured whether the 0.8 ppm is giving the constant results using the same using the UV visible spectrophotometer, and which can give the constant results over the time. So you can see the absorbance value which is a constant over the time. And what we have done is first of all we need to spike the concentration of the LOQ onto the stainless steel 316 plate. Why we have taken the stainless steel 316 plate is because the same standard stainless steel is used for the in the pharmaceutical industry. So what we can do is we can spike the 0.8 ppm, which is the LOQ of the method, on the stainless steel plate, and after that we can use the swab, which is of the TechSwab company, which the swab will be able after using the cleaning solvent, which we have taken as the methanol water ratio. So after drying of the surface, we can collect the residue from the surface by using the swab. So how we need to swab the surface, that you can see on the third picture, that by using the first swab, what we can do is we can swab the surface by horizontally and then vertically on the second side, and then we can put that swab into the a glass container, and we can cut off the surface of the swab, and we can put it in the container, and then next swab will be taken, and that will be swiped on the as a cross-sectional area, and using the both sides of the swab, and both the solutions will be mixed together, and we can use all vortex also, and after that we can check the absorbance of the that particular solution, and we need to do the recovery studies of the 80%, 100% and 120% of the LOQ, and you can see the absorbance values, and we have also calculated the percentage recovery, and according to the ICS guideline, it is greater than or equal to 80% recovery is allowable by using the to verify the cleaning procedure is effective or not. And you can see that this is more than 80%. So this method can be used in the industry, especially when the same equipment is used for two different drug formulations. This we can do by HPLC also, but UV visible spectrophotometry is used because this is very convenient, and this will take only less time to do the study, as HPLC will take more time to verify that the cleaning the surface is having not the residue of the previous drug. So that's why we have used the UV visible spectrophotometer, so that we can do the studies in less time, because the main purpose of the industry is to do the production, and they cannot invest more time on the cleaning procedure. Thank you so much, sir.
Thank you. So should we take that you are completed?
Yes, sir.
Yeah.
So much,
May I now invite Dr. Sarkar to have his queries, please?
Hello, can you hear me?
Yeah.
Very nice presentation. Yeah, I had a little bit of difficulty in the beginning to understand what you're saying. So what I understood from your presentation, this particular formulation or film coated tablet on this one, you have validated, right? This method.
Yes. Yes, sir.
Oh. What is the next step?
Sorry, sir.
What is the next step? What exactly are going to take this result? What are you going to do with this result?
We have completed the study on the UV visible spectrophotometer, and what next we can do is we can try to develop the method on the HPLC that we are trying. If we can say lower the LOQ of the method, so that we can detect the less amount of the minimum amount of the drug present on the surface by using HPLC. But here the UV visible spectrophotometer is used because industry cannot invest more time on the cleaning validation, as they have to focus more on the production side.
Got it. Thank you.
Okay, now let us listen what Dr. Dhiman has to say about this.
Pawan, this was a nice presentation with the industrial application. So one of my doubt, which you have answered it slightly, so I'll go more specifically into that. UV versus HPLC. Like you told that if you take UV technique, you would be able to save time and save man hours. But as far as HPLC is considered, it is always more sensitive, and the traces of the API or active present in the cleaning solution can be traced out through HPLC in a better way than what your UV is going to help you. The first thing is that.
So here
And,
I can, yes, sir.
Pardon?
Please, sir.
Yeah,
Continue,
and
sir.
what you did not mention is the surface area which you covered, because while calculating the while performing the cleaning method verification and validation, you need to study the area of the equipment exposed to the active substance. Okay, the surface area is that. Yeah, so I think that was missed out in the presentation.
Yeah, sorry, I'm really sorry for that. We have used the 5 by 5 centimeter square area for that, and according to the guideline, we can use maximum 10 by 10 centimeter square area. And to answer your first question, is that you must have seen the limits, how much limit of the carryover of the previous drug is allowed for the next drug formulation is less than 10 ppm or 1 by 1000 of the dose of the drug. So here we are getting the LOP of 0.8 ppm, which is less than the guideline. So that's how I can answer. We are getting the lowest amount according to the US FDA limits or any other limits. So that's how I can answer. And industry cannot afford to say the to invest the whole day for the cleaning validation to start over the next production. So that's why UV will take less time, and they can use this method, and they can develop the method for other formulations also, and they can use the equipment.
Okay, which other information related to the API you have used for your method finalization?
Sir, here we will use the how a drug is soluble in the particular solvent, especially in the cleaning solvents. First thing. Second, whether the drug is adhering to the surface or not. If the drug is adhering to the surface of the equipment, there is specially a requirement of the cleaning validation procedure we need to develop. So these two things we need to understand before going for the next step.
Okay.
And third thing I missed is third point I can add is the toxicity of the drug, whether how much dose of the drug is toxic to the patient taking the next formulation, because the patient do not want to take the to admit the dose of the first drug.
Okay. So, as per my understanding, the maximum daily dose and AD 50 are of consideration.
No, sir. It is the here they have not mentioned the LD50, but they are seeing that the 1 by 1000 of the dose or less than 10 ppm of the previous drug. So that's they have considered. And one thing I can add is cleaning validation is not the guidelines developed in nearly 8 to 9 years ago. So there is not much you can say understanding to the more of our you can say developing country. That's why we need to understand this validation procedure, and we need to carry out this procedure for all the drug formulation.
Okay, thank you so much.
Thank you so much, sir.
Thank you, Mr. Pawan. Thank you, Dr. Dhiman. I think we being a developing country and we being a strongest economy, definitely we have to concentrate on the economic part of the pharmaceutical industries in our country, because most of the methods are developed abroad. They are based HPLC, and HPLC is less economic than that of UV spectroscopy. So spectroscopic methods are more invited to be developed in all aspects of industrial work. Thank you, Mr. Pawan, for focusing on this aspect. I may declare now that there were the 10 presentations scheduled for today, out of which 9 presentations have been made. Respected judges should be satisfied with the presentations, and they will have sufficient time till we have a tea break now. And after this tea break, we will have the results. The result, I hope Dr. Dhiman and Dr. Sarkar will tabulate the results in time and provide to us. I wholeheartedly thank Dr. Sarkar to spare his time, and Dr. Dhiman to spare his Sunday with us. To evaluate, 3 hours is not a lesser time. They were with us for near to 3 hours now. Now my colleague Dr. Sanjay Patil will take over from here and will sum up. I request Dr. Sanjay Patil.
Thank you, Dr. Chatpalwar sir. I am very thankful to both the judges, Dr. Sumit Sarkar sir as well as Dr. Munish Dhiman sir, for accepting our invitation for evaluating today's oral presentations at International Virtual Conference SNJB PharmaCon 2022. Both of you have provided a strong input, suggestions whenever required for the research work carried out by today's presenter, so that will be benefited to the researchers for improving their research work. So I am again very thankful to both of you. So I request our technical team to display the certificates as a token of love, gratitude, and affection towards both of you.
Thank you.
Kindly accept the certificate.
Thank you so much.
Thank you, Dr. Sanjay.
Thank you, sir. Thank you very much.
Dr. Sarkar, I hope to meet you sometime, maybe next year when you visit India.
Yeah, let me send you the evaluators thing.
Yeah. Now, it's my request to you both that please complete the evaluation so that we may declare. We are waiting for the evaluations. Send it on the official email address. You may print out the sheet, fill up the marks, sign it, and mail it to us so that we can tabulate it with yesterday's and day before yesterday's presentations.
You will receive it in next 5 minutes, Doctor.
Yeah. We may have a tea break, and we will be rejoining up at 4 p.m. sharp for the valedictory function. Take it as an open invitation to you people, you both, Dr. Sarkar and Dr. Dhiman, to be available with us during the valedictory function.
Sure.
I can't promise because it's for 4:15 here.
Oh, yeah, it's too late. I won't pressurize, sir. Thank you, sir, for your time.
Yeah, I'm going to send it within few minutes.
Yes, sir. Yes, sir. Thank you.
Vaibhav sir?
Yes, shortly we'll be starting with the valedictory ceremony. Just a couple of minutes more. I am requesting all the delegates, dignitaries to be on camera. Though it's a post-lunch session, but I'm able to see all those fresh faces over the screens. I can see Mr. Tanmayji Mandloy. Welcome to you, sir. Shortly, we'll be starting with the function.
Welcome all guests. Welcome.
It's exactly 4 p.m. I request the convener to permit me to start with the valedictory function. I need the permission from convener, Dr. C.D. Upasni, sir. May I start with the session?
Let sir join.
Yes, I'm waiting for sir to join.
Yes, he is there.
I need permission from the convener to start this vital program of valedictory function.
Yes.
Thank you so much.
If you are ready with the results, you can start.
Yes, sir. Meanwhile, the results for the oral presentations are getting on. We'll be starting up with our valedictory ceremony. I'm very consciously using this word as ceremony because no doubt we have made it a grandeur of conducting this 3 days international conference, now popularly called as SNJB PharmaCon-2022. Here, I'm not using word welcome, but I'm taking a cognizance with an appreciation to all of you because major of the list I'm seeing are the same candidates who were there right from the first day. So I must really appreciate all of them for their presence, for their interest and their keen approach towards taking all this knowledge from the whole ocean of pharmaceutical updations and resources and researches. I need to say good morning or good afternoon or good evening to each and every part of the world wherein all of the people have joined us. Maybe some of the dignitaries are also on the line and all those specific delegates who have participated in the oral presentations as well as in the approach of, you can say, presentation C. Now, after this cognizance, I welcome everyone and move up with the thanks from our side to all of you because it is said that Vakta Nacha Vakta Tochi Shrote Vina. So you all were here. So all our speakers, all the oral presenters were also having an enthusiasm to share their views. So to getting into the sharing aspect, we have definitely taken it as the concept of together we grow. Very latestly, Ayushman Bharat as a national health program was launched from the PMO. And in that launching, respected Prime Minister had said one notion that Sal Ek Ayushman Anek. That means every year will be celebrated as Ayushman Bharat. That means taking your healthcare system on a higher side, and every healthcare system will be very keen to take every individual from the country to the most healthiest person. And this is what we are now sharing in this international platform. As Bharat always says, Vasudhaiva Kutumbakam. I'm very happy to now come to the concluding sessions. And before moving ahead, I need to just announce that we are having our chief guest, who is a venerated senior pharmaceutical quality control and quality assurance, as well as regulatory affairs professional in the field of pharmacy. His name is Shri Tanmay R. Mandloi. Mr. Mandloi is not at all a name who is not known in the pharma field, especially in India, because he's a very, very senior and experienced consultant for a number of pharmaceutical industries. Shortly, I'll be introducing him before of his chief guest address. Moving ahead towards the presidency speech by our convener, over to Dr. C.D. Upasni, Principal and Convener for SNJB PharmaCon 2022.
So good afternoon, one and all. Respected Tanmayji, who is our support to run our regulatory affairs course in pharmacy. Coordinators of today's function, Dr. Ratish and Dr. Swati. Those who have planned this activity nicely and moderated nicely by Dr. V.A. Chakpalliwar, Dr. S.B. Patil, and Dr. Raman. Then present delegates on Zoom as well as on YouTube. This is one occasion for all of us, those who wait for this particular SNJB PharmaCon. And this PharmaCon is nowadays a awaited event in the field of pharmaceutical education, especially. Many delegates from various colleges and in India as well as in Nigeria and other countries, they wait for this particular event, and the standard and quality of this event is increasing every year, and we are trying to focus on quality pharmaceutical research to be displayed in this particular conference, and the knowledge sharing by experts from various fields. I should not forget the name of our mentor, Dr. Saranjit Singh, who himself is a stalwart. He is a name which is used as a reference standard for whether it is pharmaceutical education or pharmaceutical research or even regulatory aspects. So in all the sector, Saranjit Singh sir has proved to be a reference standard, and he is our mentor, and he is driving us to these activities. The theme for this SNJB PharmaCon 2022 is given by him, and all our delegates were benefited by the theme which is given by Dr. Saranjit Singh, and various resource persons, those who deliberated on various aspects of regulatory, whether it is in terms of filing ANDA, or it is a herbal, or it is a biomedical device, or whatsoever may be a form of pharmaceutical research. Because documentation is very important, because filing is very important, and we should know the standards, we should know the references, and we should know the profiling, how to do the profiling of our documents and before presenting it to a certain apex body. Tanmayji, you will be happy to know that our college has started regulatory affairs and pharmacy course from last year, and this year those students are looking forward to your knowledge and your sources, that they should learn the technique, they should learn the documentation, and they should learn many things from you. And therefore, I specifically, it was my idea and Dr. Basarkar sir's idea to call you for the valedictory function. So, I welcome you on this particular occasion, and I thank all the participants, all the resource persons, those who contributed to this SNJB PharmaCon 22 this year. Thank you. Thank you very much. Over to you, Atish.
Thank you, sir. Thank you so much, sir. It was a nice insight towards the topic, which was been as entitled for SNJB PharmaCon 2022, and the respective orations, whatever we have taken up in this particular regard. Sincere thanks, sir. Regarding chief guest, for the first day of this inaugural ceremony of SNJB PharmaCon 2022, we had Padma Shri Dr. Raman Ganga Khedkar as our chief guest. I learned that Ganga Khedkar sir has born in Ganga Khed, one of the town in Maharashtra. And we are taking this conference from Nashik. Nashik is the first place where Godavari is incepted. And the most holiest part of Godavari is at Ganga Khed. That means to its highest point of holiness and to the highest point of know-how and knowledge towards pharmaceutical and medical field, we have Dr. Raman Ganga Khedkar. He has given an insight towards the epidemiology as well as being as the most efficient and you can say the most senapati towards the war of COVID. We sincerely thank Dr. Raman Ganga Khedkar ji for his first day presence. Our mentor, rather I must say consciously our beloved mentor, because unless his driving force and unless until he had given us proper guidelines towards the conduction of this SNJB PharmaCon 2022, an international forum, we wouldn't have been made it possible. So as per his guidelines, we have taken it on the right path. This is the 6th year of conductance of this SNJB PharmaCon 2022 International Forum. I think with very rightly, as his name is Dr. Saranjit, we are also having such a mentor where we all are feeling that we are Saranjit towards you as under your shadow to attain the most shelter in this knowledgeable world for updates in resourceful, researchful pharma industry happenings. Today at the valedictory function, I am very glad to announce about today's chief guest, Shri Tanmay Mandloi. I've got a very big biodata and resume of his, wherein I was just counting down these accolades and it was not less than 20. So that means to count and to say about all of them will be very difficult as we are literally having a happy approach towards having him. A chemistry grad with very well versed analytical chemistry knowledge from Mumbai University and almost having 35 years of experience in the field of quality control, quality assurance, and regulatory affairs. As our convener, Dr. Upasni sir has told that we have launched this particular theme of SNJB PharmaCon 2022 with the regulatory affairs aspect in very specific. We are very glad that we are here to conclude with this conference with the person who is having very well versed with regulatory affairs and profession. Some of the accolades what he is having and his expertise, let me also support it about analytical method validation, process validation, area validation, validation of water system, prepared various documents like validation master plans. He's having thorough knowledge of stability studies as per ICH guidelines. He's also having his proper hand in vendor development and the audits. He's also there as a trainer for a number of staffs to be trained in various domains of pharmaceutical industries. He's having a very big experience in quality assurance of solid orals, liquid orals, ophthalmics, injectables, and ointments. His experience, he include almost experience from 7 different industries, or very specifically 7 different domain what has worked since 1987 and presently is working.
I think there is a technical glitch with Professor Vaibhav's side. He'll join us soon. Kindly be patient.
Am I audible?
Yes.
Am I audible?
Yes. Yes, sir, go ahead.
And with respect to the accomplishment, what Mr. Tanmayji have, he has worked as team leader for successful grant of certification from NSF from its nutraceutical plant at Mission Viva Care Limited.
Since 1987 and presently is for
Am I audible?
Yes, now you are audible.
Thank you. So with this bigger introduction as such, I will request Mr. Tanmayji Mandloy to grace the occasion by his words of wisdom, proper focus on regulatory affairs, and his view towards SNJB PharmaCon 2022. As such, we consider him as one of our family members, as is associated with us with his proper guidelines towards our post-grad program for regulatory affairs. Over to you, Mr.
Hello. Hello.
Yes, you are audible, sir.
Yeah.
Yes.
Hello.
Yes,
Can
Yes, sir,
you
you are
hear?
audible.
Yeah. Honorable Principal, sir, all faculty members, I thank you very much for inviting me on this as a chief guest on this platform. I would like to brief overview of regulatory affairs. As you all know, basic role of RA, that is regulatory affairs, is preparation and filing of drug master file for market authorization in various countries for product registration in MOH of that country. Regulatory affairs in a pharma industry is chain between regulators, industry professionals, and consumers to help make them make sure that the marketed products are safe and effective. People who work in regulatory affairs interact between regulators, that is government, the regulated market, that is regulated industry, and the market consumers, market that is consumers. It is a chain. After product registration, in any country, it is the prime responsibility of regulatory affairs to file annual updates and intimate any changes in drug master file to regulators, which has happened in due course of time. Because time to time, the guideline and compendial requirements, compendial specifications, they change. So we have to intimate to the regulatory bodies. The range of products covered in regulatory affairs for filing is huge. That is drug product, foods, agriculture products, veterinary products, surgical equipment, medical devices, diagnostic tools, herbal and nutraceuticals. Recently, I had filed an herbal drug product for marketing authorization for WHO certification. It is the responsibility of regulatory affairs to keep updated of current legislations, guidelines. Hello. Hello.
Yes, we can hear you.
Yeah. People who work in regulatory affairs interact between various bodies like technical teams of plant, technical teams of marketing persons, and the agencies, various agencies are there. Like if some legalization of document is there, so for that you have to regulatory person has to talk to various embassies also, countries embassies also. It is a wide role of regulatory affairs. After product registration in any country, it is the responsibility for regulatory person to keep abreast of current regulations, guidelines, rules, and because if the regulatory person is not updated, it is very difficult to interact with the regulatory bodies. The regulatory affairs professional plays a vital role in advising on what will be realistic information, prescribing information, that is label for intended product. The regulatory department reviews all documents from a regulatory perspective, because reviewing a document is a major task of regulatory affairs in spite of making a document. And it is very much required that the document is consistent and complete, and that its conclusions are explicit. The documentation includes clinical trial applications as well as regulatory submissions for new product and for changes to approved products. Once again, I thank everybody for inviting me here as a chief guest. I wish all students a very bright future. Thank you.
Thank you so much for the insight, sir. Now I request one of our coordinators, Dr. Swati Zadav, to ahead with the concluding remark. We have to spare some time later for the announcement of prizes, first, second, and third for the oral presenters. Over to you, Dr. Swati Zadav.
Thank you, Vaibhav sir. Thank you very much. So good late afternoon, or you can say good early evening. Dear delegates, here we come to an end to this 3 days international conference organized by SNJB's Shri Suresh Dada Jain College of Pharmacy, Chandwad. The theme of the conference this year was well thought of by the team, it being global regulatory challenges to seek approvals for ANDAs, herbal medical devices, herbals, and nutraceuticals. The conference began on a fresh, beautiful morning of 2nd of December with the presence of 2 stalwarts in the area of health and medicine of India. Padma Shri Dr. Raman Gangakhedkar, former head ICMR and director in charge National AIDS Research Institute, Pune, graced the occasion and inaugural function as chief guest, and Dr. Saranjit Singh, former professor, dean, Department of Pharmaceutical Analysis, Naipur, Chandigarh, honored us by being the first speaker of scientific session. The honorable management and trust board members of SNJB chaired the function. Honorable Zumbarlalji Bhandari, coordinator of SSDJ College of Pharmacy, and Advocate Shri Prakash Chandji Bokadia, coordinator of SDFL College of Pharmacy, blessed the function with their presence. Campus administrator, respected Pramodji Garankar, conveyed his greetings to the program. Principal Dr. C.D. Upasni welcomed the guests and delegates. Dr. Raman Gangakhedkar enlightened the audience with his decades of experience in medicine and clinical trials, especially his close remarks on development of COVID-19 vaccine, regulatory challenges in developing newer drugs and vaccine through clinical trials, and overcoming the hurdles of regulatory challenges globally for approval of new drugs and vaccines. Dr. Gangakhedkar motivated budding researchers to work hard and sincere in the area of drug discovery and regulatory affairs. It was really a great intellectual feast listening to both the stalwarts, Dr. Gangakhedkar and Dr. Saranjit Singh, in the beginning of inaugural function of SNJB PharmaCon-2022. The inaugural function was very well and beautifully compared by Vaibhav Butske. The scientific lecture session started with grand opening by Dr. Saranjit Singh sir, whose name is taken with utmost respect by everyone in pharmacy field. He enlightened the audience on the topic current global regulatory challenges, control of cohorts of concern in pharmaceuticals. Dr. Singh emphasized on heightened focus at pharmaceuticals, including synthetic impurities, residual solvents, elemental impurities, genotoxic impurities. His presentation was meant to create awareness among the participants on development with respect to testing and control of cohort concern. The second scientific session of day 1 was deliberated by Dr. Prashant Nikam, Vice President and MD, Merck, Sharp and Dome, Australia and New Zealand. He discussed the impact of COVID-19 pandemic on global pharmaceutical sector, need for greater regulatory collaboration and harmonization for addressing future public health crisis. Dr. Prashant, after his technical session, also took out some time to reflect on the lessons learned in his life during the journey of success as well as failures. The post-lunch session was started with oral presentations. Well, then the day 2 of SNJB PharmaCon 2022 morning brought in a huge inflow of knowledge and discussions. The first lecture on day 2 of development of in vitro diagnostics was very well and extensively discussed by a very able and knowledgeable speaker, Dr. Vivek Borse, who has an expertise in the field of medical device development technology, nanobiotechnology, formulation development, drug delivery, etc. His lecture involved some insights on regulatory processes associated with development of in vitro diagnostics. The PG and PhD students must have been hugely benefited by the insights given by the speaker. The day 2 and session 2 of SNJB PharmaCon was conducted by Mr. Amit Bhopre, an Assistant Manager, Siddha Ayurvedic Research Foundation Private Limited, Nagpur, India. Mr. Bhopre discussed in depth the regulation of herbal medicine in India, Europe and South Africa region, emphasizing on current scenario. He threw light on need of harmonization of herbal medicinal products registration regulations across various countries across the globe. The question answer session by the delegates further threw light on the topic. Day 2, session 3 by Dr. Pramod Agarwal, a technopreneur from Netherlands, spoke about cross-technology innovation, building ecosystem and delivering holistic health and care. His talk covered several important aspects of healthcare, such as need for young entrepreneurship for translating ideas into profitable products and services. This lecture was followed by oral paper presentation session on day 2. And now, today, the day 3 of SNJB PharmaCon 2022 started in the morning with first session by Dr. Amit Jorapurkar, General Manager, Zydus Life Sciences Limited, Ahmedabad. Dr. Amit enlightened the delegates with his topic, regulatory pathways for repurposing drugs. He explained the interesting approach of repurposing of the drugs to optimize the drug development. The plenary session 7, that is the session 2 of last day 3, was engaged by Chief Scientific Officer, Indus Biotech Private Limited, Pune, Dr. Prasad Thakur Desai. He discussed the global regulatory approvals for natural products with the case of cinnamon. The talk focused on the evidence-based development process with examples of internationally approved natural ingredients and products from cinnamon bark. All these 3 days of PharmaCon, the scientific sessions were held in pre-lunch session and oral paper presentation was held in post-lunch session. We, the organizing committee of SNJB PharmaCon 2022, wholeheartedly appreciate rigorous work and sincere efforts by scientific committee to read, observe, scrutinize, analyze, and evaluate more than 120 abstracts and to shortlist 30 abstracts out of them. Dr. V. A. Chatpalliwar, Dr. S. B. Patil, Dr. Manoj Mahajan, and Dr. C. T. Nemade, we appreciate you as our in-house scientific committee. We still appreciate more to our external evaluators of scientific oral paper presentation evaluators. We are indebted to Dr. Professor Adeloye O. Adewale from South Africa, Dr. Shailendra Gurav, Goa, Dr. Pramod Agarwal, Netherlands, Dr. Sandeep Chavan, Glenmark, Mumbai, Dr. Munish Dhiman, U Medica Labs, Mumbai, and Dr. Sumit Sarkar, Research Scientist, US FDA, USA. We are really fortunate to have all these evaluators, and we are really fortunate to have today's guest, Mr. Tanmay Mandloi, like they are from all over the globe and who added to our knowledge or the knowledge of delegates to empower everyone intellectually and academically. Dear guests and delegates, the objective and motive behind arranging this international conference is to continue the noble work of delivering healthcare to mankind by pharmacy professionals by all possible means, so that this baton of teaching learning process never extend. The healthcare research should continue and the process of acquiring newer knowledge and enthusiasm towards it should never ever extinct. I would like to conclude with my concluding remarks by requesting the technical team to take us through a beautiful video of 1 minute, which gives us the message to the delegates, especially most of them are students. The delegates most of them are students. So I would like to give the message through that video that don't choose to extend. Sampunaka. Stop making excuses and start making changes. The future is in your hands. Dear all, please note that this video being now, it will be played shortly. It is taken from some social media, but the message in it is apt. May I request the technical team to run the video for 1 minute?
You okay? You need a minute? Cool. Listen up, people. I know a thing or two about extinction. And let me tell you, and you'd kind of think this would be obvious, going extinct is a bad thing. And driving yourselves extinct? In 70 million years, that's the most ridiculous thing I've ever heard. At least we had an asteroid. What's your excuse? You're headed for a climate disaster, and yet every year governments spend hundreds of billions of public funds on fossil fuel subsidies. Imagine if we had spent hundreds of billions per year subsidizing giant meteors. That's what you're doing right now. Think of all the other things you could do with that money. Around the world, people are living in poverty. Don't you think helping them would make more sense than, I don't know, paying for the demise of your entire species? Let me be real for a second. You've got a huge opportunity right now. As you rebuild your economies and bounce back from this pandemic, this is humanity's big chance. So here's my wild idea. Don't choose extinction. Save your species before it's too late. It's time for you humans to stop making excuses and start making changes. Thank you.
Thank you, technical team. And thank you, dear delegates. This was the message. Don't choose extinction. Don't stop making excuses and start making changes. The future is in our hand. With this brief concluding remark, I would like to hand over the mic again to compare with Vaibhav Buchke, sir. May I please request, sir, to take this program ahead? Vaibhav,
Great,
sir.
Dr. Swati. In a nutshell, you have concluded with all the happenings of these 3 days and a proper know-how about each and every stepwise happening was very well discussed. Very specifically, I really want to thank you that the video was wonderful and it was definitely taking home message about really what we need to know and where we need to stand. We need to stand all together. Now it's the right time to move ahead with the prize distribution, and we are now ready with the prizes. I will hand over the voice to Dr. Vivek Chatpalliwal, wherein he will give up the prize distribution, and then we can congratulate all the prize seekers and those who have made their attempts to get these prizes. Over to you, Dr. Vivek Chatpalliwal.
Thank you, sir. Thank you, Mr. Bhaskar.
Vivek sir, you are not audible. You are muted.
Hello.
Yes.
Hello.
Yes, yes. So there is an echo. There is an echo.
You switch off the second device.
Yes.
Yes, I have. I must thank my scientific committee team as well as coordinators, Dr. Atish Mundara, Dr. Swati Jadhav, and above all, the convener of this program, Dr. C.D. Upasni, for allowing us to have, helping us, assisting us, and guiding us all together to conduct this conference successfully. I will be falling short in my duties if I forget to mention the guidance provided by Dr. Saranjit Singh sir. Yesterday, I had requested him to be available at this program, valedictory program, so that he can encourage the presenters, he can encourage the budding scientists and to do well. But he has expressed his inability and has typed a message. I will read out the message. According to him, no human being can replace science as a greater ally, as the greatest teacher in all time. So we must always respect the scientific principles. We must always respect the science and should always take it as our ally to proceed further. So thank you, Dr. Saranjit Singh sir, for this beautiful message. Now I know that everybody is waiting for the declaration of prizes. Our evaluators have worked for days. Before that, the team of scientific committee has struggled a lot to compile and select only 30 presentations. To our surprise, this year we have received an overwhelming response. As many as 122 abstracts were received from different corners of the world, out of which we had to select only 30 for presentation. That was a very daunting task. Those whose presentations were not selected shouldn't lose heart. They fell short by few points. They should take this into their stride and improve further. There are many chances you people get nowadays to present in several conferences. Coming to the result. Yes, I should appreciate the efforts and the swiftness in action shown by Dr. Sarkar and Dr. Deemak to quickly calculate the results of today's presentations and provide us with it so that we could tabulate it in time. For the conclusion, for the prizes, we have in all 8 prizes for almost all 3 positions. We have discussed a lot within these 15 minutes amongst the team, and we have come up with 8 prizes. So let me first declare the consolidated prizes. There are 3 consolidated prizes. The first consolidated prize goes to the presentation entitled P30, that is assessment of hepatoprotective and nephroprotective potential of nerolidol on Vistar rats, presented by Ms. Simona D'Souza. Congratulations. Congratulations, many congratulations, Simona D'Souza. Next consolidated prize goes to presentation P25 presented by Mr. Kunal Bagul. Ball milling approach to enhance the dissolution rate of enzalutamide using water-soluble polymer. The third consolidated prize, just a minute. The third consolidated prize goes to presentation number P2, synthesis of novel thiosemicarbazide analogs for potential applications in molecular recognition by Ms. Harsha Narkade. Congratulations. Now coming to third prize. There are two people on this position. Third prize goes to Mr. Siddheshwar Jadhav, P17, entitled Targeted Delivery of 5-fluorouracil in Breast Cancer Using Hollow Mesoporous Alumina Nanoparticles. Congratulations, Mr. Siddheshwar Jadhav. Next prize 3 goes to presentation P21, Pharmacological Inhibitions of Calcium V2.2 Alpha 2 Delta 1 Beta 1 subtype calcium channel on peripheral neuropathic pain. Presented by Mr. Abdullah Sherikar. Congratulations, Abdullah. There is only one candidate for second prize. Presentation entitled P18, entitled In Silico Modeling of Dual Inhibition of Dipeptidyl peptidase 4 and xanthine oxidase in Diabetes mellitus. Presented by Ms. Sneha Duff. There are two presenters on first position. I'll declare the first position prizes. Encoded P16, QBD based approach on RP-HPLC method development and validation of bupivacaine hydrochloride in bulk and in-house developed nanostructure lipid carriers by Mr. Sarfaraz Khan. The second first prize goes to P12, entitled Stability Testing of Hydrochlorothiazide and Quinapril HCl in bulk and tablet formulation by Ms. Deepali Bhandari. I congratulate these all 8 winners to having been adjudged as the winners of the presentation. Congratulations, one and all. Thank you. Now, I congratulate all the presenters as well as all the prize winners. At the end, I would like to invite Dr. A.S. Mundala, the co-coordinator of this program, to present his vote of thanks.
Thank you, sir. Thank you very much. First of all, I would like to congratulate all the winners of this oral presentation competition. It's my immense pleasure to propose vote of thanks for this 3 days virtual international conference on global regulatory challenges to seek approvals for ANDAs, medical devices, herbals, and nutraceuticals. We call this conference as SNJB PharmaCon, which stands for Scientific and Juvenilia Baton of Pharmacy Conference, and it rightly goes with the name of our trust, that is SNJB, which stands for Sri Nemina Jain Brahmacharya Ashram. So it was an idea which came in our mind in 2017. Dr. Saranjit Singh sir was behind us to start this kind of baton, which will be passed on every year to each and every faculty who will be handling the responsibility to be the coordinator of each year's SNJB PharmaCon. I'm really fortunate to tell you that this is my 3rd time I'm able to coordinate this conference, and I was ably supported by Dr. Swati Jadhav. So big thank you to Swati Jadhav first, who could make these things done by us in a short period of time. Let me tell you, it is being said, well begun is half done. And getting a person like Dr. Raman Ganga Khedkar, who is having the 4th highest civilian award at the hands of President of India as a chief guest for inaugural function, that was epitome of this year's conference. And let me tell you, he's such a down-to-earth person that when Swati ma'am met him in one small conference in Pune, she just insisted him to be the chief guest, and immediately he accepted our invitation. So I can say that this year, because of the presence of Dr. Raman Ganga Khedkar, we could escalate to a new height with this SNJB PharmaCon 2022. My heartfelt thanks to our mentor, Dr. Saranjit Singh sir, without whose lecture we will be never able to start our proceedings or plenary sessions. The very first lecture, he is our opening batsman, and the way he scores the run, I don't think anybody else is required to compensate. And that's why we always ensure that if he is available, then and then only we will schedule our PharmaCon. Mostly in the first week of December, we plan SNJB PharmaCon. This year, the theme of the conference was being given by our mentor on regulatory affairs. And let me tell you that we have started regulatory affairs postgraduate course in 2021. So it was a nice gesture of Dr. Saranjit Singh sir, wherein we could highlight our first batch of regulatory affairs. And for sure, these batches will flourish under the able guidance of Dr. Saranjit Singh sir and our chief guest of the valedictory function, Sri Tanmay Mandloiji. Tanmay is instrumental in helping us in regulatory affairs issues. He is keen to provide us the details and enlighten our students as and when required. The second speaker of day one was from, and let me tell you, arranging or organizing international conference is always a daunting task, to be very honest. And reason behind this is to manage and accommodate the international speakers. We are really indebted to all our resource person, but I would like to put a special mention of Dr. Pramod Agrawal, who was sitting since dawn. He is in Netherlands. He was sitting since dawn and he didn't change his position. He was a resource person for third session. And at the same time, he evaluated all the presentation. So hats off to you, sir, for your dedication. We are really indebted to you. No doubt all other speakers also, they provided insights into different regulatory arenas, whether it was related to medical device by Dr. Vivek Borse, whether it was related to herbals by Amit Bhople, whether it was being related to the natural products by Thakur Desai, or it was related to repurposing of the drug by Dr. Amit Jorapurkar. Sir, we are really indebted and grateful to you all that we, as and when we call you, you immediately accept our invitation. Keep blessing us, keep giving that love to us. Because of you only, we are able to conduct such a nice conference. Let me tell you, we could have 6 evaluators for 3 days of this oral presentation competition, and they were also meticulous enough to evaluate each and every presentation, whether it was Dr. Shailendra Gurav, whether it was Dr. Adiloy, whether it was Dr. Sandeep Chavan, Dr. Pramod Agrawal, Dr. Manish Dhuman, or Dr. Sanjay Sarkar. We are really happy and pleased to have you all with us during these 3 days of international conference SNJB PharmaCon 2022. I'll be committing crime if I forget to acknowledge the support and able guidance given by our mentor, by the principal of our college, and at the same time by the honorary management, Shri Jumbarlalji Bhandari, Shri Prakash Chandji Bokadiya, and Shri Dinesh Bhauji Loda. We won't be able to conduct these kind of conferences, and we won't be able to make it at such a grand level unless and until we are being supported by you all, sir. So we are, from the bottom of my heart, as one of the coordinators, along with Swati Jadhav, I take this opportunity to thank you, Upasni sir, for putting faith into us and allowing us to conduct this conference. Though we couldn't get much time, but I hope we could give justice to the theme which was being given to us by Dr. Saranjit Singh sir. No doubt, we are also grateful to all our delegates. Let me tell you, Tanmay sir, this is the first time that we could cross the number of delegates beyond 1,000. 1K participants in an international conference and attending the conference wholeheartedly for last 3 days. Though it is Sunday today, there were n number of delegates which were there listening to our eminent speakers on the regulatory affairs. I will be grateful to thank our scientific committee members for keen interest in evaluating each and every abstract and selecting the important abstract and doing a lot of brainstorming sessions for subjecting only 30 abstract to be selected out of 120. I know, sir, it is a daunting task and you have done it. So we are really thankful to you. No doubt, registration committee was really behind each and every delegate. They provided kind support to each and every delegate, provided attendance link, provided feedback form in time, and they could maintain the record of each and every delegate properly and nicely. No doubt, how can I forget to say big thank you to our technical team who made sure that this conference go uninterrupted. So their support, their backup to us for conducting this international conference virtually was only possible because of the support given to us by our technical team. At the end, last but not the least, our food committee, Lokesh Kothari and Hitesh Share, they made sure that in last 2 days, even today, they have provided the stump us food to the members. And in last 2 days also, we were being served by delicious breakfast and lunch. So thank you, a big thank you to each and everyone for making this conference a grand success. Thank you, Tanmay sir, for sparing your valuable time and accepting our invitation in one go and sparing your Sunday. I know it is really crucial that you are sparing your Sunday with us. We are really indebted to you, sir. Thank you. Thank you one and all.
Thank you, Dr.
You,
Atish. Thank you, Dr. Atish. May I request our chief guest, Mr. Tanmay ji, to witness one ceremony. I am proud to announce that our team of abstract book, that is Dr. Aman Upaganlawar and team, they are
I'm sorry, I couldn't mention. Thanks to Souvenir team as well, Dr. Aman Upaganlawar and his team for making abstract book available in time. This is the first page of the abstract book of SNJB PharmaCon-2022, wherein the abstracts of all resource persons along with the abstracts of all presenters who have presented their findings have been included in this abstract book. And all the delegates will be receiving the e-copy of the abstract book soon from the organizing committee. Thank you, Aman and team, for making it happen within a given stipulated period of time. A big thank you. Swati ma'am, are you there?
Yes, sir, I'm there. Sorry, I was dropped out.
Okay.
Is unveiling of abstract book over?
Yeah, we could see the first page of the abstract book. Thank you to the technical team.
Atish sir,
Yes, ma'am.
is unveiling of abstract book over?
Yes, ma'am. We could see the first page of the abstract book. We are really thankful to Souvenir team for making it happen within time.
Yes, yes. Just before the vote of thanks and everything, concluding session is over. We really appreciate.
We really appreciate all the delegates for their august presence in last 3 days, and we expect the same support and cooperation from our delegates. आज तो कहीं जाने की इच्छा नहीं हो रही है, शायद इसीलिए आसमान भी रो रहा है. SNJB PharmaCon is coming to the conclusion, and even sky is blessing us by pouring its blessings. Dear delegates, you will be receiving your certificate within a week's time, because we'll need some time to understand and to get the idea of your attendance from the attendance link which was being provided every day. As mentioned earlier, minimum 80% attendance will be taken into account for disbursing the certificate. So be patient, keep in touch. We'll be providing you the certificate as early as possible. Over to you, Swati ma'am. Are you there? Shall we close the ceremony here?
Yes, if nothing is remaining, then
yes,
we should say goodbye to everyone and close it.
And see you soon in the month of December of 2023 again for SNJB PharmaCon 2023, which will that baton will be taken by someone else next year by one of our professors, and you will be delighted to join us with us again for this kind of brainstorming session. Again, it will be of 3 or 4 days. Thank you. Goodbye. Sayonara. And take very good care of yourself.